Good morning, all. If your actions create a legacy that inspires others to dream more, learn more, do more, and become more, then you are an excellent leader. As it's rightly said that leaders are not born, they are made. Leaders are the role models who inspires the followers and motivate them to reach the target of success. Good morning, everyone. I am Ajita and my co-host Jovita take this privilege to welcome you all to the most awaited event, the joint of Third International Conference on Bioinformatics and Data Science 2022 and Ninth International Conference on Public Mental Health Neuroscience 2022.
The conference has been organized by Sarvasumana Association Bengaluru, an association that aims to nurture an interdisciplinary association uniting public health, mental health, medical sciences, biological sciences, information sciences, and neurosciences. In this area, previously too, we have organized many such scientific conferences, workshops, and training programs, and we will continue to do so in the future. It's a mark of our tradition to evoke the Almighty God at the beginning of an important event. Like a great philosopher once said, prayer delights God's ear and it melts his heart. Now, I would like to invite Ms. Anusha B.S. to sing an inaugural prayer song.
महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि महागणपतिं महादेवसुतं गुरुगुहनुतं महादेवसुतं गुरुगुहनुतं मारकोटि प्रकाशं शान्तम् मारकोटि प्रकाशं शान्तं महाकाव्यनाटकादि प्रियं मूषिकवाहनमोदकप्रियं महागणपतिं मनसा स्मरामि महागणपतिं मनसा स्मरामि महागणपतिं. Thank you.
Thank you, Ms. Anusha. We all feel blessed indeed. Over to you, Ms. Ajita.
Achievement seems to be connected with actions. Successful men and women keep trying and achieve it. As an achievement chaser, I gladly welcome Mrs. Padmashri Murthy, President of Sarvasumana Association, Bengaluru. Mrs. Padmashri Murthy is a women scientist under the prestigious Department of Science and Technology, Government of India. She is a research scholar under Career Point University, Kota, India. She is a gold medalist from Morarji Desai National Institute of Yoga, New Delhi, under Ministry of Health and Family Welfare. She has over 10 manuscripts published in different journals and magazines. So I welcome Mrs. Padmashri ma'am to deliver the welcome speech. Good morning, ma'am. You are on mute.
Can you hear me now?
Yes, ma'am. Yes, ma'am. Carry on. Yes, ma'am.
Yeah, thank you. Swami Vivekananda says, a ship in harbor is safe, but that is not what ships are built for. Good morning, one and all. Distinguished guests, dear students and friends. It's my great honor and pleasure to invite you to participate in the third International Conference on Bioinformatics and Data Sciences that is ICBDS 2022, and 9th International Conference on Public Mental Health and Neurosciences, ICPMN 2022, scheduled virtually during December 22nd and 23rd, 2022. We had a plan of conducting this conference offline, but due to some unavoidable circumstances, it's happening online. Thank you all for joining, and I would especially like to thank Padma Shri Institute of Life Sciences, Bengaluru, Sanatana Yoga Vijnana Kendra, Bengaluru, for continuing their collaboration with Sarvasumana Association. And for the first time, MGM University, Mahatma Gandhi Mission University, Aurangabad, Maharashtra, also joining with us. I thank them also. We create this platform every year to get better understanding, opportunities, and systems to support mental health needs and services. I would like to welcome young and brilliant researchers, talented student communities to this platform. WHO says in recent years, there has been increasing acknowledgement of the important role mental health plays in achieving global development goals, as illustrated by the inclusion of mental health in the Sustainable Development Goals. Many mental health conditions can be effectively treated at relatively low cost, yet the gap between people needing care and those with access to care remains substantial, which is very fundamental. Effective treatment coverage remains extremely low. Affordable mental health care was far too limited even before the pandemic, particularly in humanitarian emergencies. The COVID-19 pandemic has made us to see the gaps in mental health services more deeply. According to the World Health Organization, WHO, at least 20% of young people are likely to experience some form of mental illness, such as depression, mood disturbances, substance abuse, suicidal behaviors, or eating disorders. We have to work to build resilient health systems and services that meet the mental as well as physical health needs of the community. We must try to reframe and advance the mental health agenda into response and recovery plans, because ultimately there is no health without mental health. Many NGOs, educational institutions, psychologists, counselors, all of them have joined their hands to improve the lives of hundreds of people. And it is important that we continue to build on it to ensure that those suffering from mental illness do not have to suffer all alone. The richness of ancient Indian literature on mental health and illness since prehistoric era is well known. Many literatures, especially Bhagavad Gita, Bhagavad Gita is quoted often in modern psychology articles for various reasons, including understanding Indian psychology and psychotherapeutic relevance. Ancient literatures help us understand the cultural attitudes, belief systems, and group philosophies which has shaped, influenced Indian psyche for thousands of years. These influences have given Indianness manifest in the social life, social support systems, and coping strategies. These are relevant issues for mental health and illness. The richness of indigenous knowledge, methods, and techniques which may be more relevant, easily applicable, and which may bring the desired change more easily and quickly, are not used adequately during psychotherapeutic interactions. Similarly, a lot of methods which are of Indian origin, for example, yoga and meditation, which are widely practiced around the globe, are used nowadays in therapeutic practice in India. It is also strongly suggested by many researchers that the ancient Indian knowledge on mental health should get proper representation and be integrated with modern treatment. There is a tendency among people to take the state of mental health for granted. But there is a dilemma in the form of an assertion by Indian philosophy. In human life, miseries and illness are inevitable. This thesis is a logical consequence of samskara theory. A soul takes birth only for the purpose of undergoing the fruition, the realization of its accumulated lot of samskaras, that is, impression of actions. There is no such thing as exclusive happiness or sorrow. There is never one without the other. Even though people pretend as if the contrary is true, that is, there is only happiness or only sorrow. No, it's not true. In this context, it is interesting to note that the meaning of the Sanskrit word Ayurveda, that is ancient Indian system of medicine, the Ayurveda is not like science of, it's a science of longevity. Ayurveda is not a science of disease-free life. If miseries and illnesses are inevitable in human life, what then is mental health? State of mental health should not be dependent on the presence or absence of illnesses or difficulties and should transcend both. It should also involve sincere management of illness and public or difficulties to the best of one's abilities and circumstances. In such an attempt, the issue of success or failure has already been dealt with while explaining about contentment. Thus, state of mental health is a set of attitudes and coping skills. Today, the conference is on bioinformatics and data sciences also. Bioinformatics as related to genetics and genomics is a scientific subdiscipline that involves using computer technology to collect, store, analyze, and disseminate biological data and information, such as DNA and amino acid sequences or annotations about those sequences. In bioinformatics, nearly every task can be done with one or two programming languages, R and Python. How data science is used in bioinformatics? Bioinformatics is a field of data science that focuses on analyzing biological data at the genomic and protein levels through software. The findings of bioinformatics can benefit healthcare, crops, and biodiversity. I'm coming to the end of this welcome speech. According to the schedule of the conference, many interesting keynote topics are there. For example, frontiers of nanoinformatics in nanomedicines and decision-making in the era of big data and machine learning. Many wonderful topics are there for oral presentation also. For example, neuropsychological disorders in Koragas. It's a tribal group of Dakshina Kannada, exploring hub genes in lung cancer, mutated genes and binding sites of DNA associated with proteins in Alzheimer's disease, enhancing crop production with application of nanoparticles in the agriculture, graphology, 2 papers are there on graphology, study of yoga in Indian psychology, family conflict and financial stressors among COVID-survived newlyweds, parenting stress and resilience with ASD and ADHD children. So I would welcome once again all the delegates to make this conference fruitful. All the best. Thank you.
Thank you so much, ma'am. Thank you for sharing the meaningful message. Over to you, Ms. Jovita.
Louis Pasteur once said, "Science knows no country because knowledge belongs to humanity and is a torch which illuminates the world." As a representation of the court, I take the privilege to welcome Dr. R. Somashekar. He is a chairman of an international conference on public mental health and neurosciences 2022. He is a research director at Vashisht Academy of Advanced Studies and Research, Bengaluru. His research interest lies in biotechnology, neuroscience, and medicinal chemistry. He has over 50 publications on his name. He's been awarded with many honors and recognitions like the late Shri K.P. Deshmukh Memorial Prize for the Best Research Paper Presentation at the Deccan Sugar Technologies, Pune, in the year 1989. He has also secured 4th rank in MPhil degree from the Gulbarga University during the period of 1997 to 1998. He's been given scientific talks on various topics in All India Radio, which has been widely admired. So I humbly welcome Dr. R. Somashekar to present the inaugural message. Sir, you're on mute.
Now, okay? Am I audible?
Yes, sir. Yes, sir.
Good morning, all the delegates, and warm welcomes to all of you for this 2 days third international conference on bioinformatics and data science, followed by 9th international conference on public mental health and neurosciences. It gives me immense pleasure to deliver inaugural speech on this occasion. It is indeed a great pleasure to state that very distinguished delegates internationally, 8 members, and nationally 50 members, followed by some 37 to 40 research papers are presenting in this conference. It is really a great pleasure to state that a unique platform to share and exchange the knowledge on these occasions. See, as we know, bioinformatics mostly serves academic research and medical fields, whereas our data science perform experiments and draw conclusions about their effects on industry products and consumers. So these two branches, that is bioinformatics and data science, go hand in hand because they are parallelly moving with the same objective. Because communication is an important skill for both data scientists and bioinformatics employees. But they can communicate directly depending on the circumstances. In data science, there is more teamwork and interactions with the clients than there is in bioinformatics. In bioinformatics, employees can work directly with biologists to conduct experiments and report findings. Career-wise, job outlook opportunities for both groups align with those for computer and information technology specialists. Before 2030, the government expects a 22% growth in job opportunities. Anyway, we have a wide range of job titles, people's working in the bioinformatics as well as in data science. For example, in case of bioinformatics, a wide field, so we can call them as bioinformatician, bioinformatics specialist, bioinformatics engineer, and biostatistician, so on, like this way. Similarly, the data science peoples designate as data scientists, data architects, database administrator, data engineer. Like this, we have some distinguished designations which are there in bioinformatics and data science. Whether you pursue bioinformatics or data science, you will perform work oriented around data analysis. Choosing an academic and career path that you find interesting is best, as both fields have challenging work. In most cases, if you choose bioinformatics and later wish to transition to data science or vice versa, you will find it easy to accomplish this. So overall, we can say bioinformatics and data science like a two faces of a coin. They are living together. That's what we can say. This platform to exchange, share, which makes lot of knowledge to be acquired. With these few words, I wish all of you great success and get more knowledge and dissipate this knowledge into your research work, and also make some good contributions, because we are in the challenging world, because the current world is a digital world. We can do many things in the interest of nation, in the interest of our public health. So with these few words, I wish all of you a grand success and make this 2 days conference, international conference, more successful, and go with a good knowledge and make use for your fruitful results in the coming days. With these, I thank all the organizers and Dr. Bagchi, Padmashri, so on, so on. I extend my good wishes to all of them on this occasion. All the best, all of you. Thank you. Thank you so much.
Thank you, sir, for your motivational and influential message.
Thank
Over
you.
to you, Ms. Ajita.
Thank you, Ms. Jovita. I am profusely elated to take an opportunity to introduce our chief guest of the day, Dr. Sanjay N. He is a multidisciplinary innovative scientist with expertise in metabolic engineering, molecular biology, molecular diagnostic, cell biology, infective diseases, microbiology, genetics, sociobiology, evolution, and ecology. He has an extensive experience in designing and management of metabolic engineering, molecular biology, molecular diagnostics, biochemistry projects, and has established research laboratory and assay development. He has worked on CRISPR-Cas9 based genome editing for the development of genetic engineered cell or microorganism. His research work lies in cloning, expression, and purification of recombinant protein in E. coli, Pichia, Saccharomyces, insect, and mammalian expression system and functional characteristic of expressed protein. He has even developed several molecular diagnostic kit or assay for infectious diseases. He have very deep knowledge in NGS metagenomics. We take the privilege to welcome you, sir.
Yeah. Nandini, thank you for introducing me. And thank you, Prenon, for giving me opportunity to put my thought on this platform. When I got invitation from Prenon for talking on this platform, then I got in my mind in what way I should speak on this platform. And then in front of me, I think 10 to 15 days back, I got one slide. And that slide is perspective in big data. Then I went in that presentation and different research papers also. Then I started to think how data is dominating. Instead of only data, how biological data is dominating in coming days. See, there are 4 domains of big data are coming. First domain is astronomy. Every year, near about 25 zettabyte data is coming. That much big data in astronomy. And every year, 1 exabyte data is coming for storage. And when you are doing analysis, there are pipelines and procedures are very tricky day to day in astronomy. At the same time, when you are comparing astronomy data with Twitter, every year 0.5 to 15 billion tweets comes, and storage of that data will be near about 1 to 17 petabyte per year. And here, if you want to analyze data, then you need to do metadata analysis. Means there is a huge data types present on Twitter platform. So you need to do metadata analysis. Next domain is I have taken next domain for discussion here. That domain is YouTube data. Near about 500 to 900 million hours videos are uploading. People are uploading on this platform, and the storage pace of storage increases every year 1 to 2 exabyte. And this need a very limited requirement for analysis because there are videos are there. But at the same time, if you are comparing these 3 platforms with genomics, I have told you astronomy data every year 25 zettabyte, Twitter data 0.5 to 15 billion tweeters, near about 1 to 17 petabyte, and YouTube data near about 1 to 2 exabyte. At the same time, when we are talking about genomic data, every year these data come 1 zeta basis per year. And this need 2 to 40 exabyte storage for storing this database. And this, especially when you are going for analysis of this data is very heterogeneous data, and you need high level of skills for analysis of this data. With this context, my today's focus is how we can understand the ecosystems of microbes and how they communicate with each other, and through that communication, what they are releasing for betterment of human being. And that topic is microbiome. And here, that microbiome is nothing but the encompass of bacteria, archaea, viruses, phage, microbial eukaryotics, some worms, fungus. That whole microbial ecosystem, when you are going to sequence it and when you are going to compare it, that whole system is known as microbiome. And when you are going to compare this system, or when you are going to work in microbiome, you don't need to do whole genome sequencing. You can only focus on there is one fragment every living organism has, which is present in microbial ecosystem, and that fragment is 16S ribosomal RNA gene. This gene is very important because this present in non-living organism. It has only single copy, highly conserved, and highly variable regions are present there. And at present, I have told you every year 1 zettabyte bases are people storing in database. That means there is a huge reference database is present. Because this is the condition for analysis. You need to have a reference database. So that reference databases are present in microbiome in a huge quantity. It's very easy to speak on microbiome or metagenomics, but it is very hard to analyze that data. And that is depend on, again, what is your target? In what way you are going to what you are targeting or what you are expecting from your project, that is important in microbiome. If you are looking microbiome project, there is a particular flow. That flow is you need to work on sample collection. That sample should be very clear, qualitative, and after getting sample, then you can use different DNA isolation kit or DNA extraction kit. In present in market, you can use Kaizen kit or in vitro gene kit. There are number of kits came in market. And third one, I am talking about workflow of this. Third one, you can amplify 16S gene which is present in microbiome. And then fourth will be the analysis. For analysis, you can do different. You can design and develop different pipelines. See, pipeline is nothing but because when you are doing after amplification, you are doing sequencing of that 16S RNA, and when you are doing sequencing, you will get a huge amount of data. After getting data, that data should be very qualitative. There should not be noise present in that data. Now, see, for amplification, your PCR strategy should be proper. Like there should be proper melting temperature for amplification. DNA sequencing, you should be focused on DNA sequencing length. Primer should be specified, and you above all three parameters, you should always compare with previous study. So you will get an optimum noise and you will get a good quality data after sequencing. See, when we are talking about 16S RNA, that RNA has a various region. From V1 to V9 regions are there. Usually people speak or people work on region V3 to region V6. These four regions are very important for analysis of data, and for this again the most important is you should read more and more previous paper. Through that theoretical experience, you judge your data and then you focus on particular region. So you will get a very clear your results. See, when we are doing sequencing, there are again number of platforms are there, like Illumina sequencer is there. Previously, Iron Torrent was there. Now Iron Proton is there. PacBio is there. Nano sequencer is there. Number of sequences came, but again, see these sequencers are not core technologies. I can say they produce only sequences, but what type of sequences you need, that is in your hand. After getting sequences, the most important and critical job is to analyze it. Generally, whatever data I got in my laboratory from my people, we analyze QIIME module. There, when we are using this module, we do first quality check, sample diversity on the basis of taxonomy aware, similarity among samples, then association with metadata. As I have told you, this data is coming in a flood, zettabyte data is in the form of zettabyte. So there are limitations for us. So with the help of high throughput computational technology, nowadays came in market, that is machine learning classification. You can use machine learning technology and algorithms, and after doing above all things, you can predict functionality of your data. See, there are two platforms we can use, that is QIIME and Mothur. QIIME platform you can use with Python, And Mothur is also, it's depend upon your familiarity of sequences. Personally, I am very much familiar with R language and then Python. See, I have told you how you are going to analyze. Initially, you need to check quality of the data. You need to check the score of that data. The sequences should be minimal length of consecutive high quality bases, maximum number of consecutive low quality bases, maximal number of ambiguous bases, and minimum Fred quality score. There are a number of softwares present in market, open source as well as chargeable, like Quota Depth, Trimomatic, and Sickle. These are the prime quality filtered softwares are present. And there are some chimera checking softwares also. Through these softwares, or we can say, go on online platform, check 2 to 3 times the data, get quality data, and proceed for see, when we are talking about 16S RNA, I have told you this is a phylogenetic marker. And when phylogenetic marker come, or when phylogenetic word come, then there are 3 types of diversity come in your data sample. That is alpha diversity, beta diversity, and gamma diversity. You analyze your data through these 3 domains or 3 types. That is, to describe the diversity of sample, you need to know what you are counting. Individual sequences, most precise but vulnerable to sequencing error effect. Or you can cluster the sequence. And here, people are using alpha diversity for taxonomic group formation. Another one is another one parameter at the time of this data analysis. You can use operational taxonomic unit clustering also. Through that clustering, you can calculate distance between sequences also. Next one, see, if you want to analyze this data a very simple way, then BLAST software is there from last 22 years. NCBI is promoting this software. And PAPLacer, this is another phylogenetic placement you can use for analysis of quality sequence data or sequence parameters. Later on, you can also use machine learning classification package for analysis of these data. Second one is, first one, you are going to analyze sequences on the basis of alpha diversity. Second one, you can analyze your database or data on the basis of beta diversity. Here, you can perform pairwise comparison between all sample to build dissimilarity matrix. This is one of the parameter you can use for beta diversity analysis of the data.
Sir, più na, che Morris pani clean and Java, Kaya for 50. Correct. ऐकलं
डायरेक्ट कनेक्ट करो दिसत नाही.
करतो.
Sorry, everyone. I guess Dr. Sanjay Harke is facing a network issue. He will be joining in a minute.
Hello.
Yes,
Hello.
sir, you're audible.
Nandini, are you getting my voice?
Yes, sir, you're audible
Okay.
now.
Okay. Sorry for interruption. See,
No issue.
after analyzing data with reference database, actually there is a skill of experienced person to judge your targeted or your required information are you getting or not from the data. That is very important. But day by day, machine learning algorithms are coming and they are giving, I can say, more accurate prediction of your targeted, of your target. Through machine learning, you can identify aspect of community structure that are predictive of sample attribute. There you will get an advantage of machine learning, that non-linear combination of variables, data transformation, and it can accommodate many different representation of the data. That is very important. or I can say these are the advantages of machine learning algorithm data analysis of microbiome. At the same time, this machine learning will provide little bit more complex, and it is again more time consuming. So if these two hurdles, or if these two problems solve, you can predict very nicely your data. See, after analyzing, what you are doing is very simple tree-based classification you do, and lastly you can analyze, or you can predict it. What I assume from this, 16S is a very effective proxy for microbial diversity. As on date, scientists have uploaded near about 10 lakh sample data, microbiome sample data from worldwide. That is again 16S. And these data, when you are going to observe, or when you are going to read papers, this is very effective method for understanding microbial diversity. This 16S data will create equal with results that are comparable. This 16S operational taxonomic unit describe ecological cohesive also. Cohesive. And ultimately, as I have data for tree of life or phylogenetics, but what exact we are expecting from this huge data, there are some perspective points are there. I feel we need to analyze data with heterogeneous data also. Like metagenomics, metatranscriptomics, metaproteomics, and metametabolomics. These three, I can say these four different data, when you are comparing with each other, I feel you will get a more accurate your targeted prediction, or you will go very close to your desired results. And at last, you are through such type of databases, or such type of research, you can get real-time microbiota data, and according to that, again I have told you, it's depend upon your view, but what we are expecting, see, after this people are going also for pangenomics, that is through that pangenomics, multi-laboratory evolution data. There are some laboratories are presently working in world. Through that, they are going to analyze some bioactive molecules. They are expecting bioactive molecule for pharmaceutical industry, food industry, agro-based industries, and all. So it means that before starting such type of big projects, I will not say for investment, But for big database projects, we need to set our only first target, and then and there you start to work on such projects. With this word, I will stop my lecture, and once again, I will thank you for whole committee to allow me or give me opportunity to present my this thought in front of you. Thank you. Hello.
Thank you, sir. Thank you for the interesting and informative talk. We request the audience to post questions in the chat box. Once again, if there are any queries, you can please pose your questions in the chat box.
Nandini, I feel this is little bit database oriented. So I don't know how much they understand this my presentation, but this is very important for people who are working or who are very newcomer in laboratory research. Thank you.
Okay, sir. Thank you. Thank you so much, sir. Over
Thank
to you,
you.
Ms. Jovita.
Thank you, Ms. Anjita. Albert Einstein once said, "If we knew what it was we are doing, it would not be called research, would it?" It's an occasion like this we get opportunity to test our knowledge and understanding. As an explanation of the quote, I'm honored to welcome our guest of honor, Dr. Wanga Howlin. He is an associate professor at the University Science Malaysia. His research key interest lies on plant-based nanodrugs and nanoformulation for drug delivery. He has international and national grants on plant metabolomics and drug delivery, and has over 85 paper publications. And he has 26 project proceedings and has been an author for 5 book chapters. He's been honored and recognized by Non-Aligned Movement Senior Fellowship India, Australia Award Fellowship, and he's been a visiting professor and scientist in China, Australia, Bhutan, and USA. We take the privilege to welcome you, sir.
Hello. Can you hear me?
Yes, sir, you're audible.
Okay, that's great. Thank you very much, the Ms. Chairperson, Ms. Jovita. Yeah, I'm very glad to be here again. I remember that last year I presented a keynote speech on the metabolomics, and today we are going to have another great conference again. And I believe that it is the joint conference with the 9 public mental health neurosciences, as well as the 3rd International Conference on Data Sciences and Bioinformatics. So it is a great pleasure to know that the Sumana Association, together with the Padma Shri Group of Institutions, the Vasis Genomics Research Lab Private Limited, and also Sanatana Yoga Vinana Kendra in Bengaluru, India, are hosting these 2 conferences. And I believe that this is a wonderful event that allows the young researchers as well as the experts to present their work, to receive feedbacks, and probably to enhance their quality. And I also understand that the call for papers are favorably received, and this event probably allows the academic members as well as the industry professionals to share suggestions on how to improve the effectiveness of their research or maybe in their work. So I believe that the keynote from Dr. Sanjay just now was very interesting and also was very enlightening, where the rise of new technologies has led to the increasing interest in various topics such as the data science, the machine learning, as well as the metabolomics. And I believe that the goal of these 2 conferences today and also tomorrow would like will bring the various components of computer science, biology, and technology to address the challenges, probably in terms of the big data biology. And I also believe that it also aims to provide probably a forum for exchanging experiences and also finding some of the solutions to many challenges that the researchers may encounter when it comes to analyzing and managing big data, as what mentioned by Dr. Sanjay just now. Well, thank you very much. I believe that the conference will provide an opportunity for instructors, students, as well as maybe industry experts to discuss the latest developments in metabolomics, in integrated spirituality, genomics, proteomics, so on and so forth. And again, I would like to express my gratitude to Mrs. Padmasree Murthy, the conference patron, and Dr. R. Somashekar, the events chairman, for their effort in making these two conferences successful. And not forgetting my sincere thanks to conveners, Dr. Pranan Bakchi, and also Dr. Kitty. So I'm sure that the delegates will leave with happy memories. Now, having said that, the title of my keynote today is, hang on, let me share my slide. Do you mind to, oh, okay. So screen number 2. Can you see my slide now?
Yes, sir, it's visible.
Okay, that's great. I'm going to put it in the presentation mode. It could take probably a little bit of time. Can you see that it is in the presentation mode?
Yes, sir.
Okay, that's great. Okay, so the title of my talk today is The Frontiers of Nanoinformatics in Nanomedicine. So as we can see here, we started the whole thing with informatics from the computer, from the maybe starting from the biomedicine, then it moves towards the medicine, and now it moving towards the nanomedicines. And probably we started from the bioinformatics, then now going into the nanoinformatics. So, yep, my name is Van Gao. I'm from the Advanced Medical and Dental Institute, University Science, Malaysia. Very happy to see you all again. Okay, let's start off with our beginning for the biomedical informatics. As you can see here, the use of the computers in biomedicine, which I think probably began in 1950s, with the early application in hospitals, right? And also some of the pioneering scientific computer programs for the modeling of the diagnostic processes. So somewhat later on, the computational biology actually emerged to tackle some of the problems. What type of problems? It could be like the scientific inquiry at the molecular level. Okay, so we have the component sizes and also the technological relationships. So we can see here the field of medical informatics, it actually gradually developed. Then it comes to the areas such as the computerized medical records. As you can see that in the previous, we always do whatever medical records in the hard copy way. But then when it comes to the computerized era, so all the medical records are in a digital mode. And moving forward into the 4th industrial revolution, we have the artificial intelligence systems for the medical diagnosis, as well as in terms of planning, in terms of laboratory, in terms of radiology, and also the hospital information systems. So what you can see here is like the whole thing is actually moving forward very fast. So in computational biology, as you can see here, the databases and some of the macromolecular sequences the structures as well as the functions were actually developed, and it is validated in some of the experimental data. And what's more, later on, the Human Genome Project actually produced some of the massive bioinformatic data sets, and the systems as well as some of the software tools that developed in the project were essentially come to the completion of the project ahead of schedule. So it's actually accelerate the whole project by the Human Genome Project. So if you can see that despite the very broad scope of application in biomedical informatics, we could see that it could be ranged from molecular medicine to the public health. Now it's in the field of nanomedicine, which has actually received a very vast extension, a very vast attention as in nowadays. So we could see that a lot of information communication sciences in terms of nanomedicines are blooming nowadays. So what is the technology and informatics? As you can see here, there are many different challenges in nanotechnology for nanomedicine, such as the need of new informatic area. So we have in terms of biomedical context, now we have the nanoinformatics, which actually refer to the nanoinformatics of the techniques, the analyzing, and also I would say the processing information about the structure and also the physical chemical characteristics of the nanoparticles and also the nanomaterials. How they behave, their interaction with the environments, their interaction with the application, their interaction with the whatsoever. All right. So when we see about the term of nano informatics, the term is actually they started in the Europe probably in the 2007. As we can see here, the nanoparticles has the span dimension between 1 to 100 nanometers. So below 1 nanometer, their behavior can be probably explained by the atomic or the molecular and maybe the ionic interaction and some other forces. But what happens when it is above 100 nanometers? Well, I would say that their properties will probably be similar to the bulk properties of a certain material. So as you can see here, 100 nanometer may also exacerbate and enhanced bioavailability and transport in probably in the biological organisms, some of the tissues, cells, organelles, membranes, and so on and so forth. So from here, we can see that the advances in nanotechnology can be used in the drug carriers, as well as the drug's reactivity, the strength, and probably the delivery can also allow the drugs to a targeted tissue. So when it comes to a targeted tissue, it will release a drug at a certain control rate and probably a drug detoxification maybe in the early stage of cancer or carcinogenic process. So when we talk about the nanoinformatics, it is about the scientists that predict the structural relationships, the methods and some of the protocols that involve the specific structures to get the optimal result. So in this case, we are here to identify some of the application using high performance materials. As you can see here, the scientific approach actually prescribes the structural precision as the key for some technological practices and also some of the cost-effective solution at this scale. If you can see that the figure number 1, there's an example of a certain curve of the discovery where the science is actually at the peak height as the expectation where the precision actually drives up the properties. However, the hype actually went down through the disillusionment and it goes slightly up curves to the map of economic stages during the development and it reaches the plateau of the productivity, which is the cost at the engineering. Having said that, we have conventional nanoscience So we have the conventional nanoscience and we have the nanoinformatics. So when we are comparing between these two, we could say that the conventional nanoscience is actually the bottom-up approach. All right, it's a bottom-up approach, which is actually based on the rules of the science itself. So we come out from the term of nanoinformatics is actually more towards the top-down, which based on the experiences, which is actually we call it as the inductive. And because of this, each approach have a different logic in terms of the conventional nanoscience, maybe based on the paradigm, where the nanoinformatics probably based on the repeat paradigm or estimate paradigm. So when we see the figure here, is the conventional nanoscience is basically is sort of knowledge or a system. Okay, and it is involved in some of the experiments or certain part of the simulations. Whereas the nanoinformatics, as we can see here, it is applies the algorithm or some of the model as mentioned by Dr. Sanjay just now, and it also deliver a better or a different understanding for a system or a problem here. So we can see from the figure here, it is actually illustrated with the approach of a mirror of each other, which can actually form a productic of the feedback loop, as we can see the probe and also the algorithm where the conventional science extrapolates as well as interpolates, as we can see from the figure. Having said that, nanoinformatics can be used in a lot of predicted properties. And number one could be the cellular uptake. Now, we will always say that cellular uptake could be conducted in the in vitro experiment. However, if you see in terms of nanomedicine, some of the significant efforts in this area could be able to understand and also improve the cellular uptake as well as the targeting itself. So what we can see here, this is primarily driven by the desire to use the nanoparticles to treat the cancer and also to deliver biologically active compounds to the cancerous cells. So some of the predictive models, maybe QSAR in this area could be very beneficial because we see that the development cost of some of the novel nanoparticles could be quite high, and also we could say that the desirable molecules before the synthesis will also dedicate to the limited sources to the performing experimental work and also to most of the promising candidates when the drugs has been targeted into the cell. We could see some of the case studies. Number one would be the case study of modeling of cellular effects induced by diverse manufactured nanoparticle, or we call it as the MNPs. It is critical for nanotechnology, where we have the experimental studies. We also have the experimental toxicology, which if we do it in the lab is actually very time consuming and it is very costly. It is often quite impractical, and why not we just do it in the efficient computational approaches by some of the predicting biological effects of the manufactured nanoparticles. So from here, some of the chemometrics methods can be used, for example, like quantitative nanostructure activity relationship, which is the QNAR modeling. We can some of the generate the model using the machine learning approaches, as what mentioned by the previous speaker, Dr. Sanjay just now, which some of the approaches such as the support vector machine, which is the SVM, and also we can use some of the k-nearest neighbors or KNN based regression, some probably of the axonal prediction power, and yeah. So one of the approach will be the can be employed, could be the predicting the biological activity profiles for some novel nanomaterials. And next would be prioritizing the design and also manufacturing of nanoparticles towards maybe a better and also a safer products. If we see the study design here, the quantitative nanostructure activity relationship, it is actually established based on the QSAR modeling workflows, where similar to QSAR modeling strategies, where the overall objective is actually to set a descriptor, which it is used to characterize the manufactured nanoparticles based on their cell viability and also the cellular uptake. So we can see that such model can also be newly designed based on their biological effects, as what you can see from the heat map here. So we can see that these are the case study where we can describe two relatively large series of manufactured nanoparticles that has been tested for their effects. Effects of what? Effects could be in effects in the different metal cores and also surface modification. As we can see, that will be tested in several different cell-based assays, where we could see some of the core, some will have the same core but different surface modifier. So we can see that in terms of molecular weight, in terms of compositions, in terms of the parameters as well as the physiochemical properties. So these are all grouped into the fingerprints of the nanoparticle from the left side, okay, the nanomaterial. And on the right side, after the quantitative nanostructure activity relationship, we use some of the machine learning methods and also model validation. Then we are able to actually group it using the Z-scores, and we can actually group it in a heat map following the different toning colors, and some of it are negatively and positively correlated to the activities. Okay, from here, the investigation continue on the dose dependent of biological effects induced by the manufactured nanoparticles. We can see that this is a heat map activity representing the biological activity induced by the manufactured nanoparticles, where at the 4 different concentration, we could see that is the Z-score for about 51 nanoparticles that have been tested, and we could see that the higher dosage will actually have the stronger nanoparticle effects. And we can see that in the hierarchy clustering analysis, we could see that based on the 51 nanoparticles that have been used in the biological activity, so some of it form 3 classes of the distinct classes based on their biological activity, and also some of the blue and also green colors, where the green or yellow colors indicates the small or low or medium similarity between the particles. However, the blue will show the high similarity between the particles. So from the heat map, based on a certain algorithms, you could actually see that there is a high similarity between the nanoparticles based on different kind of samples, as well as the low medium similarity between the nanoparticles. So we could see the in terms of from the graph of Z-scores, we could see that the 3 dot samples, as we can see here, basically the nanoparticles are quite equal to each other, although they are in different concentrations. So it shows the most dramatic dose dependent from their biological effects, particularly in the high manufactured nanoparticle concentrations. So we could see that the machine learning are able to classify the nanoparticles based on in the field of nanoinformatics. Okay, the next one, we could see the heat map that is representing the cellular uptake of different nanoparticle preparations. So the column from the right to the left, as you can see from 1, 2, 3, 4, 5 here, is a small number. It's actually showing the pancreatic cancer cells. The number 2 will be the macrophage cell line. The 3 will be the resting primarily human macrophages. And the 4 will be the activated primary human macrophages. And number 5 will be human umbilical vein endothelial cells. So we could see that each column here represents the mean value of 6 different experiments, where the red is actually refers to the lowest accumulation and the green refers to the highest accumulation. So when we see the nanoparticle, basically it hits the identifier of the large cream. From the figure here, we could see that the quantitative fluorescent activated cell sorting, basically it analyzes the 3 separate experiments, and with the certain microscopy of 680 nanometer channel, we could see that some of the cells actually merge into some of the contrast images. In terms of cellular update for targeting experiment of different nanoparticles, we could see that the preparation into the macrophages actually could be separated. In the comparison in the D, it shows a different affinity. And we could see that the FAC analysis of starting material for the nanoparticles, and also with the combined effects after the peptide attachment, where the modification of the endothelial cells. And finally, we could see that the microscopy shows the endothelial cells with the fluorescence and the channel that merge into a contrast image. So from here, when we do comparison into the treatment of the nanoparticles together with the heat map using a certain types of algorithm and also machine learning, we could know that this could be easily analyzed for a big data of nanoparticles. Next, what should we do in the preparation for nano-QSAR modeling? It could be in the first part of data collection and also data processing. Now, as Dr. Sanjay mentioned just now, there are a lot of databases that we can use for nano-QSAR modeling and also quality assessment. So the first part of the implementation could be the nano-QSAR model that involves the data collection and also probably from the descriptors and also the endpoints and some of the data processing. So you could see that some of the resources can be obtained from the literatures, from the databases, from the experiments. So it is easier basically to obtain the data for the model if the sources are actually available. So I'll tell you a little bit about the challenges of nanoinformatics later on. So from here, we could see that if, let's say, we have a poor quality data input for the model results, it will actually produce an unreliable data output. That's why in terms when you're doing nanoinformatics, the researcher has to be have to investigate in such a very suitable criteria, as well as the evaluation of the quality of the obtained nanoparticle data, which is focusing on possible application of the computational evaluation based on the algorithm. So as you can see here from the nanoparticle information, that will be the theoretical descriptor based on the data calculated in algorithm. And also we have the experimental descriptor from the experimental measurement. So from the theoretical, we go into experimental to prove that the algorithm actually work. And from the experimental conditions, we do some of the mediums and also into biological response alteration. Then we go to the endpoints. We see the bioaccumulation factor. We see the cell viability. We could also see the the cellular uptake and also long-term as well as the short-term toxicity, skin irritation corrosion for nanoparticles that has been formulated into nanoemulsion into the skin products. And we go from data processing and the pre-processing techniques. We could do the standardization and also the normalization. We could do the feature reduction and also some of the selection and data splitting. Then there will be a lot of multivariate data analysis that happened in the machine learning and also big data analysis. Moving on. Yep. So basically, if let's say you have the nanoparticles that maybe analyzed using the SEM, the scanning electron microscopy, and also the TEM, the transmission electron spectroscopy, and also the AFM, the atomic force microscopy images can also convert it into numerical values. And from here, we can also convert it into the multivariate data analysis, and we can analyze it using the algorithm that has been established. So the next one will be based on the electronic health record. I'm pretty sure that the technology of maximum technology of the health or biohealth nowadays are moving into a very advanced state where the best challenge of nanoinformatics is to link the related data of nanomedicines to the electronic health record of the patient itself. So development of a novel diagnostic as well as some of the new nanomaterials could enhance the I would say development of the personalized medicine based on the advancement of the omics that the whole world is developing now. We could see that the management of the nano-related information actually requires the creation of probably a big potential tool for decision making, along with the development of a new model of electronic health records. So we could see that in order to accomplish the new extension for the current standards, some of the tools must be developed in order to incorporate some of the terminologies and procedures of the nano-related information. I know that some of the nanoparticles are actually quite toxic, and they actually exert the toxicity at the cellular level. So there are certain of the precautions and some of the policies as well as the acts can be used to determine the possibility of nanoparticles for therapeutic purposes. So this will actually provide the level of hazards for patient safety due to secondary effects of nanoparticles when they are being analyzed. So in this case, nanoparticles or nanoinformatics can contribute significantly to extract some of the automatically to organize some of the specific information on nanotoxicity that is available in some of the scientific papers. So you can see that some of the extracted information for various purposes we could do to deliver such issue. So in terms of challenges, one is the data management. The two is a search on nano ontologies, and it could be some of the traditional electronic health record that will include the nano-related information. Okay, having said that, nanoinformatics will give some of the impacts to a few areas and also to a few fields in the world today. There will be in the industry where we could see that certain new industry areas of development, and also new informatics techniques that may allow some new companies and academic settings to work on nanotechnology. Some nanomedical research that usually requires some of the resources in order to overcome the traditional research in the area. In terms of economic, as you all know, economy is of financial importance. It gives a new job opportunities for the data scientists. And also in terms of international collaboration, I believe that this is still ongoing. We have collaborative research and facilitating and exchange international collaborations. National policy, yes, I think every country will have their national policies on nanoparticles or nanotechnology, and maybe in this case, the nanoinformatics, where it gives a new opportunity for new research and also for people or researchers that are working in these areas. Informatics, definitely, nanoinformatics, it involves the biomedical and nano services if you have the company or universities that are doing the services. And in terms of society, a very big issue will be the nanotoxicity, where we see that nanotoxicity either in the animal, in the human, or towards the environment. So this will also affect the ethic, the culture, the media, the whatever religious aspects, and so on and so forth. So there are a lot of points to be taken into consideration when you implement the whole nanoinformatics or the nanotechnology in the country. In terms of educational, it also gives some impacts where new academic programs will be developed, where you could see that a lot of nanomedicines that have been blooming nowadays, and also scientific in terms of impact for the laboratories, where the omics are developing so vast now, and we could see that there's some certain new synergy established between the biomedical informatics and nanomedicines. Then we could see that the involvement in biology and also the chemistry with the engineering could be maybe with the other fields, which will lead to a new sub-discipline. We call it as the nanoinformatics. So this is how important is nanoinformatics in the nanomedicine itself. So these are the challenges for nanoinformatics in nanomedicine, where we can see that the bioinformatics actually evolved to solve some of the problems that related to data management in topics such as gel electrophoresis, maybe amino acid sequencing, gene mapping techniques, or maybe in some plants in metabolomics. So we still need the nanoinformatics to address the two main issues. Number one is the data results from nano-level experimentation, which actually lead to large and constantly changing sets of the continual growth of a certain scientific knowledge. For example, like nanotoxicity, we do not know the toxicity of the nanoparticle itself, so we need maybe more of a data or experimentation to enhance the scientific knowledge of the nanotoxicity, and also the need for the control of the system. So here will be some further of the elements where the researchers need to probably manipulate some of the research instrument or the refinement of some of the experiments to make it the experiments as standardized as possible, to follow the guideline of the nanotechnology or the so-called nano guideline that has been developed according to respective country. And probably the development could be ever increasing application of nanotechnology and nanomedicine may have uncovered maybe some of the different problems that benefit from or require nanoinformatic techniques. So in this case, that nanomedicine, I will say that the main issue could be the nanotoxicity. So the nanotoxicity is the main issue that the most of researchers and also the doctors, the clinical scientists that have faced. So the nanotoxicity, we could use the nanoinformatics to come up with the risk of some of the toxin compounds. Then we can move further into what can be done from the nanoparticles when it is from the bench into the bed, where it can actually benefit the patients in the future. So as a conclusion, we could say that the nanomedicine actually raises numerous challenges and requires significant investment in informatics to accelerate the current research and developments. As we all know that the nanomedicine is not cheap, the research in nanomedicine, but in terms of nanoinformatics to help into nanomedicine, it could actually speed up the process and also to maybe reduce some of the cost when we get the result from the descriptor, from the algorithm. Then we will move towards the experiment. Then it could maybe save some of the costs. So such informatics, some of the requirements actually related to biomedical informaticians also carry out in some of the research project, the post-genomics, and also some of the transform biomedical research. In terms of the nanotechnology, some of the additional requirements are needed to accommodate the use of computational model, as also mentioned by Dr. Sanjay just now. So we use certain simulations with the algorithm, with the sharing of some of the codes, and we try to assimilate or we try to integrate the models from the different disciplines and from the different areas of research. So that means that the different areas could be from the chemistry, from the technology, from the biology, and also from the engineering, and also from the computational science, mathematical science, again, integrate together and put it into the informatics that we need. And having said that, the nanoinformatics also still needs a common agreement on certain field, on certain research topics, on certain ethical requirements, which is the most important when we talk about society, which also could lead to a more specific agenda for the research and environment, as well as the development of the R&D. So here we actually emphasize on quintuple helix model, where it involves the government, the universities, the industries, companies, the societies, communities, the environment, as well as the, what is the sixth one? Environment, as well as the shareholder, okay? The usage, which is the under the form of the society itself, the end user of the nanoparticles or the nanotechnology. So last but not least, because of the more philosophical point of view, we could see that nanotoxicity and nanomedicines are inherently two complementary elements. Complementary elements in terms of what? In terms of one discipline under the nanotechnology for the sustainability and also for a better life in the future for the next generation. So in other words, we love, we care, and we share for the future. So here I would like to send my appreciation and also special thanks to the collaborators, as stated here, the University of North Texas, University of Nottingham, University of Queensland, Taylor's University, as well as Singsale Medical University, and also some of the local universities in Malaysia. Thank you very much for the collaboration. Without all of you, the whatever today would not be successful. And also I would like to thank my members, my team members, which are my students and also my postdoc under the Plant Metabolomics and Nano Drug Delivery Research Group. Thank you very much to all of them. And if any one of you have not been to Malaysia, I will probably introduce a little bit of my institute. It is actually, if you see that this is the world map, Malaysia is actually here, and India is actually here. Okay, Malaysia is actually here. We are actually quite near. Okay, then in Malaysia itself, we are divided into two, which is the Peninsula Malaysia and also the East Coast Peninsula. And then my institute is actually, hang on, okay, it's actually based in the northern region of Malaysia, which is at the state of Penang. As you can see here, so this is actually the state of Penang. So in University of Science Malaysia, we have we have 5 campuses, which the main campus is in the mainland. Then we have another 3 campuses. The engineering campus is in the south, and the Institute Medical and Dental Institute, which is my institute, is actually the campus at the north of Penang. Then we have another campus in the east coast of Malaysia in the Kelantan state. And we have another campus, which is a medical campus in India, which is in Belgaum. So, yep, my campus, Advanced Medical and Dental Institute, is actually based on the mainland side of Penang state, which is at the northern region. So this is a front gate or front entrance into the institute. It is called the Advanced Medical and Dental Institute, University of Science Malaysia Penang, and this is a website. You can actually key in and have a look at the website. So this is about me. My interest is mostly on metabolomics-based quality control of herbal medicines, and also drug discovery from the natural products from preclinical studies, and nanoformulation for targeted drug delivery system. And also I'm a member of the societies that stated here. If you have any collaboration, if you like to work together, collaborate with me, you are free to contact me at this email or this number. Okay. With that, thank you very much for your attention, and I'm happy to answer any of the questions if you have. Yeah, thank you very much.
Thank you, sir, for giving your valuable insights on this topic. Sir, as our audience have some questions to ask, can we take them one by one?
All right, sure.
Yes.
All right, let us see the first question from Dr. Sumana. Are there any manufacturers who can design nanoparticles as per our study requirement? Can we custom model nanoparticles? All right, as you can see that nanoparticles, it is pretty much depending on what type of nanoparticles that you want it to be. You can actually customize the nanoparticles based on the application itself. For example, if let's say you want to do the nano emulsion for skin topical application, you can actually form the the nano emulsion, where you can actually make the particle into the nano size, and based on the one phase or two phases, then you put in your active ingredients and make it into nano emulsion. And yes, you can customize it on your own in the laboratory lab, in the laboratory scale. Then you can translate the scale into the industry scale. You can actually assign the company or the whatever factory and ask them to do it in the industry scale, and you can basically sell if the nanoparticle itself or the nano emulsion itself is actually a therapeutic value for a certain diseases or a certain cure. Yes, you can. All right. So to the second question, do you see nanoscience as next generation medicine? In fact, nanoscience, or I would say the nanomedicine, is actually moving very fast. We even have the vaccine that is in the nano size. The vaccine, if I'm not mistaken, is the Pfizer that is in the nano size, where they do the nanolipids. So the nanolipids that we can actually form the formulation, and we can put the whatever mRNA inside and do it a formulation. So that will be in the generation of nanomedicine, and we can even do a personalized nanomedicine in the future. Yes. So yep, we can actually see nanoscience is the next of a generation of medicine. But that also depending on the personalized medicine is depending on whether depending on the patients, whether some of the drugs that we put in will formulate into the nanoparticles, whether it is suitable for individual patient or not. That we'll see in the next future for personalized nanomedicine. And doctors prescribing nanomedicine. Okay, doctors prescribing nanomedicine is pretty much depending on, I would say, it is more for the cosmetic product that is available here. Some of the nano white that you use for your face, I would say cosmetic mostly do on the the nano cosmeceutical. In terms of doctor that prescribing medicine, I would say that will be the vaccine that we have.
Maintain silence.
Is it audible?
Yep.
Ma'am, yeah.
Yep.
So you can go on with the questions.
Okay, I have finished answering the questions.
I request the audience to post your questions in the chat box if there are any. I once again request the audience to post your questions and queries in the chat box. Okay.
Yeah. Any more questions? Hope you can
Okay.
understand the talk just now.
Yeah. Thank you, sir, for sharing your deep knowledge on nanoinformatics with us.
Thank you very much.
Thank you,
My
sir.
pleasure.
Over to you, Ms. Ajita.
Thank you, Ms. Jovita. Experience is the hardest kind of teacher. It gives you test first and lesson afterwards. As the explanation of the quote, I'm honored to welcome Dr. Anuradha M. She is a principal at Padma Shri Institute of Management and Science and a director of PIMS Research Center and Biotech Finishing School. She has secured university first rank and received a special prize from the Chancellor of Andhra Pradesh in the year 1989. She was recognized as a research guide by Department of Biology in Acharya Nagarjuna University, Guntur, Andhra Pradesh in the year 2006. She has won third prize for poster presentation in Bangalore, India Bio in the year 2015. She has over 100 publications, 6 patents, and over 10 research projects. We welcome you, ma'am.
Thank you for your information. Can you enable me to share my screen?
Yes, ma'am. You can share right now.
Is the screen visible? Hope it is visible to you.
Yes, ma'am. Yes, ma'am.
Yeah. Very good morning, everyone. Today, I'm going to deliver a talk on a very interesting topic, that is nutraceutical nootropics. The overview of this topic is, let us first see what exactly is mental health and what are nutraceuticals and what are nootropics and what the various types of sources and their effect on your mental health and summary of the same. All of you know that mental health is emotional, psychological and social well-being. It's very important and it determines how we think, we feel and act. Definitely, we need to be healthy to handle stress, to relate to others, and to have both emotional and psychological well-being. It is a very important thing at every stage of life. When you are a child and when you are adolescent and when you grow up as an adult, mental health is very, very important. Like any other organ, brain also will have a lot of diseases, disorders, and also degenerations. It is the utmost important chemical reaction happening place in your body. As depression increases, it increases the risk for diabetes, heart disease, even cancer many times. So you need to maintain mental well-being to be physically well, emotionally well, and psychologically well. As the mental illness increase, the risk of many diseases also increase. At the same time, it is vice versa. Other diseases like chronic diseases like diabetes, cancer, and other debilitating diseases will increase the mental illness. So it is a two-way process. You know, mental illness and chronic diseases are directly related to each other. When you have long working hours, you have work pressure, and you have economical and financial pressure, and you are under emotional stress. And most important thing, you are not having proper nutrition. This will definitely cause mental illness. Due to this, you need to really maintain your healthy lifestyle. In a healthy lifestyle, most important things, two things are very important. One is exercise and the other one is diet. Nutrition and exercise plays important role to be mentally well. How common are these mental illnesses? Most of us may not be knowing that more than 50% of us will be diagnosed with mental illness at some or the other point of our lifetime. Very alarming, isn't it? When you come to America, 1 in 5 Americans will experience mental illness every year. Why I'm providing statistics of America? Because they are the ones who regularly, thoroughly get checked up, and they go to physicians and psychiatrists, and they will try to get rid of this, and statistics are very clear there. And 1 in 5 children at some point of their life have debilitating mental illness, either during their childhood or during their adolescent or pre-adolescent or post-adolescent period. They will have different kinds of societal and educational, parental, peer, and other kind of pressures which will really cause mental illness. Apart from this, 1 in 25 Americans lives with serious mental illnesses such as schizophrenia, bipolar disorder, and sometimes major depression syndromes. Then what are the causes for this mental illness? There is no single cause for this mental illness. Many a times, early adverse life experiences cause mental illness. Trauma or history of abuses like child abuse or sexual abuses, or sometimes even in office, there might be some kind of abuses which will happen maybe from different segments of life. This trauma definitely causes mental illness. Initially, it may cause some stress, and later it will get into depression, and finally may lead to serious mental illnesses. This results in chronic medical conditions, and sometimes schizophrenia, sometimes dementia, sometimes Alzheimer's, various conditions because of this prolonged chronic diseases, bad lifestyle, and they lead to chemical imbalances in the brain. Use of alcohol or drugs are also results in mental illness. When you have feelings of loneliness or isolation, definitely you are prone to mental illness. We have to be aware of all these things to not to succumb to mental illness. And also, I'm going to talk about what are the various other things which you can manage to avoid this lifestyle related or nutrition related mental illness. My title, I have mentioned nutraceutical nootropics. I'm sure that many of you might be listening about this word nutraceutical and also nootropics for the first time. What are these nootropics? Nootropics are drugs, supplements, nutraceuticals, and functional foods that improve mental health. Nootropics offer a lot of benefits for cognitive aptitude and brain health. In whatever you eat, there are many nootropics which will help in boosting your memory and to balancing your mental health. These nootropics are smart drugs and which enhance your memory, neuron function, cognitive ability, and attention spans. Nootropics as a drug, sometimes used for the treatment of Alzheimer's disease, Parkinson's disease, Huntington's disease, dementia, and cognitive symptoms of schizophrenia. Then let us just oversee what are the various types of nootropics. In this present talk, I'm going to focus more on nutraceutical nootropics, but there are other nootropics. Most of them are synthetic drugs, and what exactly they do. I have given the group of compounds and also what exactly they do. Ampakines, they improve attention, learning, memory, and vigilance. Cholinergic nootropics, they help in neurotransmission of acetylcholine associated with cortical excitement, attentional tasks, alertness, reward processing. Eugeroids, prescribed for narcolepsy, excessive daytime sleepiness, and chronic fatigue. These days, most of us are night owls. We work up to 2 o'clock or 3 o'clock or sometimes 4 o'clock in the night, and we will get used to this daytime sleepiness, which is very dangerous for students like many of us. And also when you have to go and work in the office, if you wake up excessively in the night and later it becomes a habit and your circadian rhythm will get disturbed, and finally excessive daytime sleepiness will become a disease for you, which will result in chronic fatigue. In such scenario, they use this kind of nootropics. to treat, they use this kind of nootropics. Apart from this, you have dopaminergic nootropics, which influence levels of neurotransmissions, include dopamines and reuptake of inhibitors. GABAergic nootropics, they influence the neurotransmission of GABA, decreases stress, and also energizes central nervous system. Some more important nootropics are hormones. They improve cognition. It is very, very important. Like you need to have hand-brain coordination, and your cognitive abilities should be very good and in a balanced manner. Otherwise, you become mentally ill. These hormones elicit a response from certain neurotransmitters and alter brain activity, increase arousal and brain activation, promote sense of calmness and concentration. They play a very important role, especially when women are either during adolescent phase, when they are entering into puberty, men are boys and girls, when they become mature, reproductive maturity, what I'm talking about. And sometimes women during post and premenopausal days, they will have this hormonal functioning, hormonal imbalances, which will cause lot of stress, and people will have problems with their behavior, and they will be sometimes angry, and they sometimes will have mood swings, we call it as. So hormones play a very, very important role. And metabolic enhancers, they increase metabolic processes, cellular membrane functioning, give brain cells more energy, and improve cognition, and they eliminate toxins, which are very, very important. Finally, you need to understand one thing. Any chemical imbalance in brain or any part of the body is mainly because of this accumulation of toxins, because of accumulation of reactive oxygen species. These need to be neutralized and they need to be eliminated. And these are called as neuroprotective agents, and they are all nootropics. I request all of you to mute your speakers. Please mute your speakers. And the other important nootropics which I am going to focus today are nutrients. They improve focus, they prevent cognitive decline and promote neuroprotection. I'm going to give you few examples because these nutrients are again nutraceutical nootropics. What I'm going to talk today is about nutraceutical nootropics, and they are important in maintain mental health and cognitive functions. Psychostimulants stimulate the production of dopamine in the brain. It is very, very important factor, chemical balancing, which is required for your psychological balance, and activate the sympathetic nervous system, which leads to increased physical and mental arousal. Neuroprotective agents. I have given about these neuroprotective agents in my earlier slide. They promote healthy brain function by repairing damage. Continuously, there will be some damage because of the toxins, because of the exposure to pollution, because of the stress factors, because of your bodily reactions. This damage is a continuous process. It is a dynamic process which will be happening. Damage occurs and repair happens in your body. These nootropics help in repairing, and they promote nerve growth and maintain neuronal health. The antioxidant properties mainly help optimal cognition and prevent a cognitive decline. As I told earlier, it is very clear that the reactive oxygen species and also toxins will cause damage. It may cause damage to neurons, it may cause damage to certain brain cells. To avoid this damage, you need to use certain antioxidants which are natural in its nature, which are there in your food. What are the other nootropics? Riacetams, compounds having the ability to significantly improve cognition. These are all prescription drugs sometimes, and sometimes you get over the counter also. But as a drug, They are not available as over-the-counter medications you need to get prescribed by a psychiatrist. When I talk about nutraceutical nootropics, most of the things are available as over-the-counter drugs, and even you can do it, practice it, using it at home. Serotonins. Serotonergic supplements like 5-HTP, enzymes, and various antidepressants, they also act as neuroprotective agents. Certain serotonergic substances boost serotonin to improve cognition and brain function. If there is excessive production of serotonin, certain nootropics reduce it. So you need to have a balance of serotonin production. All these dopamines, serotonins, these need to be produced in a balanced manner in your brain. Herbal supplements, most important topic of the day. This is the one which I'm going to give you in detail. Many of these supplements are taken for their antioxidant properties and are thought to help maintaining cognition and also prevent cognitive decline. Then what are these nootropics are used for? They are used for mental diseases, mental illness, chronic fatigue syndromes, neurodegenerative disorders, sleep wakefulness disorders, treatment-resistant depression, performance enhancement, academic advantage, occupational performance, concentration, and motivation. Till now, I was talking about what all nootropics. Let us know what are these nutraceuticals. This term is being defined by Stephen DeFellis in 1989, which is derived from two words, that is nutrition and pharmaceutical. The name itself says that nutraceuticals, ceutics means treatment. Ceutics means which is used for treatments. Nutraceutics means it is food or part of food or a nutrient which provides health benefits, which include prevention and treatment of disease. This nutraceuticals they may come from plant source, animal source, microbial source. You might be wondering what is this microbial source? That is probiotics. Now, gut, brain are linked. These two are linked. Many a times when you feel stressed before your examinations, when you go and sit in a stressful condition, you feel butterflies in your stomach. That is why, that is for the reason that you have gut-brain link. Gut is called as a second brain. So gut health is very, very important to have the good mental health. That is why I'm telling that nutraceuticals may be from minerals, micronutrients, plant source, animal source, microbial source, algal source also. There are various nutraceuticals. It is a wonderful world which really allow you to maintain a very good health and which will not make you feel sick. It is prevention is better than cure always. That is why you are what you eat. So have mindful eating to have a healthy mind. Let us talk about certain nutraceuticals. Very few examples I have taken. Generally, proteins. When you talk about proteins, protein, all of us know that it is muscle mass builders and it is very, very important for your body. What are the do's and don'ts of this protein? How much you need to take? Many of us are confused, whether animal protein is good or plant protein is good and what exactly type of protein you need to take. And sometimes most of us will have problems when you ingest lots of protein. However, proteins are essential for the production of vital neurotransmitters like dopamine, noradrenaline, histamine, and glycine. Tryptophan, which is an amino acid, a precursor for serotonin, which has effects on mood and memory. Many proteins have this tryptophan. Proteins are very important. How much you need to eat? You need to eat proteins a fistful, or in other words, your weight in grams. If you are 50 kg weight, if you weigh 50 kgs, then you need to consume 50 grams per day protein. That is the essential amino acids should be there in your protein, and also many proteins are many of us will not take protein in a proper way. For example, if you talk about proteins from soybeans or proteins from pulses, we generally have problems with digestion and also problems with gastric excessive gas production and all these things. This is because of anti-nutrients and phytic acids present in these seeds, pulses. What exactly you need to do? You need to soak them, sprout them, and then consume. Then you can get rid of this anti-nutrients or phytic acid, which will cause problems for your digestive system. And also, one more important thing which you need to remember is protein should never be fried. Deep fried protein will denature, and finally it will cause again problems. That is why when you eat a cooked, steamed, sprouted protein, you will not have any problem. But when you take deep fried vadas or bhajis, you will have a problem. So you need to really consume protein in a proper way. Having protein is one important thing, but at the same time, protein should not be deep fried. This is a very important thing which I want to tell you. And protein, you can consume either plant or animal protein. It depends upon your lifestyle, but try to have minimum of a full of nuts or soaked nuts or sprouted things or any other protein, which is very important, which has an effect on your neurotransmission and mood and memory. Nutrient, nutraceutical, nootropics. This is one more important thing which I want to give you, because most of us will not be bothered to take balanced nutrients. Generally, either you take in excess or you take less. Either you take in excess or you take in less. Again, I request someone has unmuted, please mute your speakers. Please mute your speakers. It is causing a lot of disturbance here. Iron, zinc, choline, selenium, iodine, magnesium, vitamins B, A, and C. And it is energy metabolism. It really acts on energy metabolism, neurological enzymatic reactions, brain lipid, neurotransmission. They help aid in brain lipid neurotransmission.
Before the disturbance, ma'am. Dr. Sunita Vivek. Ma'am, could you please mute yourself? Thank you, ma'am. Ma'am, you please continue.
Yeah. Iodine. It is an enzymatic form. Iodine is being utilized in enzymatic formation of thyroxine, and deficiency lead to mental retardation at any age, and it is important during infancy. Salt and seafood are the best sources of dietary iodine. Iron is such an important thing, especially children at the age of 2 to 4 years will be definitely invariably iron deficient if mother is iron deficient. And if they are having genetically iron absorption is less, or whatever children eat or drink at that age may not be with sufficient amount of iron, they really have a lot of cognitive maturation delay, and they'll always be tired, fatigued, irritable. So you need to really balance child's nutrition with a lot of iron. Vitamins, you all know, folates and folic acids are even important in the first 1,000 days of the child before birth, even when the child is in mother's womb itself. Otherwise, they'll have spina bifida, which is a serious illness of nervous system. So So it causes folic acid deficiency causes a brain atrophy. Atrophy means it degenerates and it causes necrosis, or it causes sometimes lesions, or many other problems will occur when folic acid. And see the link between folic acid and iron. If folic acid deficiency is there in your food, iron absorption will not be there. Hence, you need to really balance folates and folic acid you need to have. It is present in lot in green leafy vegetables, which many of us don't consume. They should be part of your daily diet, which really has got a lot of folates and helps reducing in brain atrophy and also having positive effects on your mood. Vitamin B12, essential for all neural functions. Generally, when you observe these vegetarians, they will have the problem with vitamin B12 deficiency, because vitamin B12 is present in lots in non-vegetarian food. Vegans only, except for dairy products, vitamin B12 is very, very less in all other things. But still, I suggest you to have a lot of curds and other dairy products for having good vitamin B12. Even in green leafy vegetables, to some extent, you can find vitamin B12. The vitamin B12, really, it is such a dangerous vitamin. Its deficiency will cause permanent damage to neurons. It is irreversible. Keep this thing in your mind and don't have this vitamin B12 deficiency. As you age, vitamin B12 deficiency is predominant, because absorption will go down and it will become more and more. So be careful, especially vegans need to be very careful. You will be tired and you'll have muscle aches, muscle pains, and you'll always irritating. And this is due to vitamin B12 deficiency. When somebody claims after 45 or 50 years, if they continuously say that body pains or body aches and muscle pains and irritable moods, then better get checked up for vitamin B12. Lean beef, salmon, and eggs are good sources. Apart from this, even dairy products also has got a lot of vitamin B12. Sunshine vitamin D, which is very, very important. And it is also helps in calcium absorption. If you don't have good amounts of vitamin D in your body, it will definitely affect your mental health. Neurosteroid and its receptors are widespread in your brain. And this, it is a neurosteroid, and it binds to neuroreceptors, and which are widespread in your brain. They play a regulatory behavior. They play important role in your regulatory behavior. Deficiency cause migraines, depression. If you are constantly getting headaches, midnight headaches, which are called as alarm headaches, every day at one particular point you get headache. This may be due to vitamin D deficiency, and you may be having again pain in your legs, hands, or any parts, where there is a lot of bone mass is there because of vitamin D deficiency. Your calcium content would have gone down, and you have psychosis. As you are having depression, migraine, you will have sleeplessness, which finally lead to psychosis and epilepsy and anxiety. Vitamin D therapy is used for mood elevation. Generally, people who are deficient with vitamin D, you need to use vitamin D therapy at least for a few days till you get to normal doses, and don't forget to sit in sun at least for 10 to 20 minutes, either in the morning or in the evening. Vitamin D rich diet reduces multiple sclerosis. These are all nutraceuticals, nutrient nutraceuticals, which has got nootropic effects. Essential fatty acids. Generally, whenever we feel that we are putting on weight, we will cut down oil. See, to some extent, we need essential fatty acids, which are present in nuts and oily foods, and essential fatty acids will increase myelination and provide components of myelin sheath, which is an important structural part of nervous system, and they regulate membrane fluidity, and also the deficiency lead to altered blood-brain barrier functioning. It is very important. Not all the things can go, not all the nutrients can go into your brain. You need essential fatty acids to carry to your brain. If you want to have healthy brain, have essential fatty acids, which are present in many foods. You can have even in fish. You have many foods which have got essential fatty acids. Look into the chart where there are omega-3 fatty acids are present, and then have try to have these foods. Neurotransmitters, they are neurotransmitters and also part of very other important peptides. Now let us talk about certain nutraceutical herbs which are neuroprotective in its nature. Best herbs to boost your memory and brain power. I have given the list of herbs here. Apart from that, there are many other herbs, and this list I have selected both from food, and some of the things are we don't use it in food. For example, ginkgo biloba, we don't use it in our food. And Terminalia cebula, we don't use it in our food. Glycyrrhiza glabra, we don't use it in our food. But Allium sativum, which is garlic, we use it in our food. Curcuma longa, we use it in our food. So other things, what we lost the habit of using it in our foods, were used in Indian traditional systems of medicine, and in Chinese traditional system of medicine, in Tibetan traditional systems of medicine from long ago. Many of the times, 120 traditional herbs are used, which can really help to get rid of central nervous system disorders, is in practice in Asian countries from times immemorial. Then how to go about this? If you have little knowledge about this, you can incorporate into your food. You can continue using it. You can try to find out alternatives if you are not getting it in India or in your places, wherever in your respective places. Let us see what are the best herbs to boost your memory and brain power. And these are the herbs which you need to consume regularly, either in part of your food or in your soup or in your regular herbal teas. First thing is turmeric. It is a super spice. And turmeric was, I can tell you, the price of curcumin has gone 300 times, 3,000 times during post and pre-pandemic period. You know, during pandemic, COVID-19, turmeric is in high utility and it is being used left, right, center, which was a regular ingredient in Indian foods. Then it is used for chronic epilepsy as a preventive measure because it is an antioxidant. As I already told you, turmeric can get rid of this reactive oxygen species, and it is used in chronic epilepsy. And it helps in attenuating glial activation and suppression of the activation of cytokines and chemokines. See, I want to tell you, whenever I give this example of turmeric, I want to tell you how turmeric is being processed, and whatever you get in the market, turmeric, how different it is. I'm not talking about curcumin, where you get a pure form of turmeric. That is, curcumin is one of the components in thousands of compounds present in turmeric. It has been extracted. It has been extracted and it has been sold in the form of capsules. Unfortunately, this curcumin is not water soluble. It is fat soluble. That is why in our ancient cooking recipes, we use curcumin. We put this curcumin turmeric powder in hot oil. When you prepare lemon rice or you prepare anything, any tadka, you put turmeric in hot oil because curcumin is oil soluble. Another bitter fact about turmeric is whatever turmeric you get in the market is being processed turmeric, and this during the processing, they literally cook this turmeric rhizomes, and in this you lose very, very important proteins, therapeutic proteins like turmeric. They're all water soluble. Water soluble components are lost when you do the cooking process of turmeric. Then how to do this? Certain parts of Karnataka and certain parts of India, they use turmeric leaves and fresh turmeric rhizomes in their food, in their preparations. That's how you have to look at it. It is a holistic herb and it is a super spice. Then I don't suggest that you stop using turmeric powder, which is available in the market. You can use it, but put it into the oil, hot oil. At the same time, try to have this fresh turmeric pickle or fresh turmeric leaves for your some of the food preparations at least once in a while, if not every day, which can boost your brain health and also your immunity, which is an immune booster. Coming to Centella asiatica Brahmi, whenever I talk about, this is also called as Gotukola and Mandukaparni, and it is used in traditional system of medicine. When it comes to Brahmi, there are certain confusions among people. They think that Brahmi is Bacopa monnieri and sometimes Brahmi is Centella asiatica. One is Neer Brahmi, that is Bacopa monnieri is Neer Brahmi. This is the actual Brahmi. Whenever I talk about this wonder herb, I always try to explain what is this doctrine of signatures. The doctrine of signatures means God has assigned on every herb and sent it for our utility. See, this shape of the brain, shape of Brahmi leaf is like brain. If you can associate the shape or color or any other portion of the thing to the one of the body parts and say that it is good for that particular body part health, that particular theory is called as doctrine of signatures. Centella asiatica leaf, that is Brahmi leaf, looks like brain, that is good for brain health. Another one which has got a lot of essential fatty acids which exactly look like a brain is walnut. If you see walnut, you can feel that it looks like brain. And there are many other examples like that. Even ginkgo biloba leaf looks like brain, and these are all good for your brain health. This is a memory enhancer. In certain parts of South India, they give this leaf extract to children for improving memory, and it revitalizes brain and nervous system. It acts like an antidepressant, and it improves cognitive functions. And they are having triterpenes and saponins, which are part of this Centella asiatica. How to consume this? You can have Centella asiatica chutneys, or you can have dried leaf of Centella asiatica and prepare a tea out of it, or you can use it in soups, or you can even use it as a dressing in your bread or anything like topping. You can use it. Then it is very good for memory, especially for children. Along with curry leaf powder, you can have this at home and you can use it regularly. A spoonful of it in your curry can take care of asiaticocytes and triterpenes present in Brahmi. Then this is one of the wonder herb, that is ginkgo biloba, which is used in Chinese, traditional Chinese medicine for memory. This has got memory enhancing action. Even the leaf of ginkgo biloba is similarly, looks like brain. It has got it increases the supply of oxygen. When we were during the COVID, many of us suffered with low oxygen levels. There are certain herbs which increase the supply of oxygen in the blood, and because they eliminate free radicals, which will avoid the actual oxygen. They compete with these free radicals, compete with the oxygen supply. That is why you need to use certain antioxidants to have good oxygen supply in your body, and also to get rid of free radicals. These are called as antioxidants. They improve cognitive function, and it increases healthy blood flow, and they have antioxidant production, and they really improve memory. That's what Chinese medicine, and also in certain part of traditional systems of medicine, ginkgo biloba is used as a memory enhancer. It has got excellent phytoconstituents like terpenoids, bilobites, ginkgolobites, flavonoids, steroids, that is cytosterol and stigmosterol, which are very, very important, and organic acids, and also many other phytoconstituents which will help in neural health. Penaxensin. See, I have chosen herbs which are used in Chinese medicine, Korean medicine, Tibetan medicine, and Indian traditional system of medicine like Ayurveda, and also in homeopathic medicine. This is called as Korean ginseng. If you can see the root of the plant, it looks like a man with a head and 2 hands and 2 legs. That indicated that this is good for as a revitalizing herb. This is a wonderful root which is used from thousands of years as an anti-aging herb. The tablet Revital contains this ingredient of panaxensin. These are ginsenosides, and they even grow these roots in reactors. However, I can talk about Indian ginseng here, that is Vithania somnifera, ashwagandha, which is one of the important root which has really become very, very expensive, and it was not available during COVID pandemic because they improve your immunity. And this is an adaptogenic herb with anti-stress properties, anti-trauma properties, anti-anxiety, and reduces fatigue because it modulates immune functions. They provide protection against neurodegeneration by multiple mechanisms, and the main phytoconstituents present in panaxensin are saponins and ginsenosides. Coming to Bacopa monnieri, it is again, as I told you, this is also called as Brahmi, and it is also one of the neuroprotective and memory enhancing herb, and it is used in Ayurvedic system of medicine for centuries. It is called as a brain tonic, and it is used to reduce epileptic disorders and also to reduce anxiety and to enhance memory. The major constituents responsible for Bacopa's cognitive effects are bacosides A and B. The bacoside A and B aid in, again, help in repair of damaged neurons. How it happens? It enhances kinase activity, neuronal synthesis, and restoration of synaptic activity, and ultimately it enhances nerve impulse transmission. And this is again one of the wonderful herb which is available in India. In many other parts of the world, you can use this herb in your soups or in your food, or you can directly get Bacopa extracts as capsules or tablets. Coming to a summary of this nutraceutical nootropics. Nutraceutical nootropics are very, very important, which you have to incorporate into your food to avoid memory loss and age-related mental disorders. If anyone says that, if anyone behaves little abnormally, you say that they have become little mentally weak because of their age. That is a common factor. What we call it as age is one of the common factors to get reduced memory, or also certain other important, very serious, debilitating mental disorders are Alzheimer's and Parkinson's. It's Alzheimer's may cause, I think recently I read somewhere, humans are the only animals where with age, degeneration of brain happens, brain shrinks, it seems. That indicates that you need to really take care. Take care of the most important organ of your body by having nutrition and a nutraceutical in your food. You need to follow holistic approaches, and nootropics are providing excellent results when you have consumed along with food. Mindful eating is very, very important. Along with mindful eating and have a good healthy lifestyle, practice yoga and meditation, give proper rest, and try to focus, try to concentrate, and play Sudoku, and try to memorize the things. If you have an elderly person at home, if you don't talk to them, they just get withdrawn. That is because of the loss of memory. So you need to really trigger it, use the brain, and try to give the fuel which is required for the brain. Help people work longer and accomplish more. They are experienced and very rare commodities, where these people are required for the society. With their experiences, we can learn a lot. So take care of them and try to keep up their memory till the end of their life. Nootropics help people with Alzheimer's and various cognitive disorders. Hence, have nootropics and nutraceuticals in your food. I'm not talking about smart drugs which are prescribed by doctors. I'm emphasizing on nutraceuticals which you can incorporate into your food, which can become your healthy brain soldier. It can protect your brain. With this, I really thank all the organizers, especially Padma Shri Murthy and also Prinon Bakshi, who gives me opportunity every year to share my experiences. Even then, I learn a lot while preparing these slides and other things. It is a really good experience to share my experience with all of you. Thank you, one and all.
Thank you for the interesting talk, ma'am. We request the audience to post their question in the chat box. Ma'am, there are a few questions. Can we take one by one?
Yeah.
Okay. Ma'am, there is a question from Dr. Rashmi. The clinical trials of all nutraceutical are done to check the cytotoxic effect, or is there any room which has to be filled? Which type of the cell lines are more preferred?
Can you repeat the question?
Yes, ma'am.
Yeah.
The clinical trials of all nutraceuticals are done to check the cytotoxic effect, or is there any room which has to be filled?
Yeah.
Which type of cell lines are you preferred?
Yeah. See, there are 3-4 parts in this question. First thing is clinical trials for nutraceuticals. Generally, nutraceuticals itself is food or part of food. For food, nobody does any clinical trials, but it is changing its shape. People are getting confused with nutraceuticals and standardized herbal extracts and also drugs. See, when it is used as a nutraceutical, that is, it is part of the food. No clinical trials are required for this. Because over a period of time, who will do clinical trial for a tomato? What is the nutraceutical present in tomato? Lycopene. Who will do clinical trial for curcumin? It is not required. But when you isolate it, when you purify it, when you convert it to water-soluble form, and you claim that, for example, I say that curcumin can reduce cancer. If I want to claim this, definitely you need to have clinical. For that, you are asking me the cell lines. When it comes to cell lines, it is not clinical trial first part. It is a preclinical trial. Preclinical testing, efficacy testing, we call it as. When you are doing this cell line, it depends upon for what kind of thing you are testing that nutraceutical. If you are testing the nutraceutical for neural cells, you need to, for brain, you need to use neural cells. There are many cell lines available in the thing, and you may have to go through the catalog of MTCC, or all these people are there, American type collections, that is 80 CC. You can go through that and get your cell lines. Yes. Any
Thank
other?
you, ma'am.
Yeah.
There is another question from Anusha. Taking a regular intake of Brahmi leaves, like 1 or 2 daily, does it cause any problem, ma'am?
No, because it is used as a green leafy vegetable in certain parts. Okay, when, as I told you, when you are using it, then it is a green leafy vegetable. It is being used from ages, nothing will happen. It is being used as a chutney for making pickles and chutneys at certain parts of these things. And again, I'm telling you, any herb, when you are trying to use it, we don't know where it is being grown. If it is grown in industrial effluents, it will automatically will have high amounts of lead and pesticides and all these things. These are more dangerous than the nutraceuticals what we are talking about. You can consume it, no problem.
Okay, ma'am, thank you. There is another question from Jayanti Shukla. What do you think is the best approach for integrating traditional medicine with modern medicine and science? What would be effective method?
Yeah, see, for this, one should be very matured enough. And it is like both are, instead of feeling that they are complementary to each other. But many times people feel that they are antagonistic to each other. They are antagonistic to each other. It is not so. They are complementary to each other. And sometimes this, as I'm talking about traditional systems of medicine, is focused, a real traditional system of medicine I'm talking about. There are many quacks and they have not done practices, and you have to really follow the Pakka traditional system of medicine. It always targets for the cure, that is root cause. But most of the modern systems, I'm not telling all, some of the modern systems of medicine, apart from surgeries and antibiotics, it is just treating the symptoms. If you have fever, you take an anti-inflammatory or an antipyretic drug, which will reduce your fever. But why you got the fever? People get fever for various reasons. It may be viral infection, it may be stomach infection, it may be even cancer. It may be for some other reason. If you have fever, you take one antipyretic drug and it reduces fever, but it cannot cure the root cause. So generally, traditional system of medicine, they follow, especially Ayurvedic system of medicine, they always think that all your treatments are there from your gut. That is why as soon as you get into any Ayurvedic system of medicine, they first clean your gut. They give you a lot of laxatives and they give enema and they clean the gut, your stomach, and then they proceed with, it is holistically they will do it. They will give you a little bit of yoga, meditation, and a lot of massages, and also removing toxins. It try to manage with, but sometimes, many times, what we do is we go to these traditional healers only after finding no other way. And generally, we go when it is at the terminal illness stage. At that stage, it is very tough for anybody, either for modern or for traditional system of medicine. That is why you are try to balance your health by using nutraceutical concepts every day. It is not that when I get sick, then I will try to use something. Instead of that, don't get sick at all. That is the one practice which all of us need to really do it cautiously, mindfully.
Thank you, ma'am. There is another question. There is another question from Nandini Rao. Are there any specific herbs for the symptom of sickle cell anemia?
I'm not very sure, ma'am, because I need to really check. If you can just post it into my WhatsApp number, I will try to give you some information.
Ma'am, I have one question.
Yeah.
Ma'am, I had this question that as you have promoted on consuming turmeric, but in the market, the turmeric we get are like really like it has many kinds of additives which are not like good. For example, lead. So pure turmeric is like not such available in market. So ma'am, how do you suggest we consume this turmeric?
It is available. It is available. There are certain organic turmeric which is available. And also this lead, when you talk about this lead, nobody will add lead to turmeric, but lead is invariably present because we have a lot of roadside cultivation, lot of vehicle effluents will have lead. That is why you will have this lead problem in all the vegetables and fruits, whatever we are having. But what I suggest is, if you are very keen about using a pure turmeric, you can go ahead buying organic turmeric, which they grow it in a very, very pure manner. They don't use any chemicals, any pesticides, and their soil is free of lead. And in such a scenario, there are many good companies which are giving this organic turmeric. You can buy it.
Thank you, ma'am.
Ma'am, there is another question.
Mm.
Do you think traditional okay
Yeah.
do you think traditional medicine and modern medicine method can be used to be treat certain disease at the same time?
No. See, the concept is both, and certain disease at the same time. See, some of the diseases, especially when it comes to skin diseases, no, many times the psoriasis and all, people believe in traditional system because it takes long time in modern system. And also again I told you, these are all autoimmune diseases. For autoimmune diseases, it is very difficult to find. You can manage certain diseases, can be managed, and treatment you cannot assure. Under the same system of, for the same disease, both the systems are having, it is very vague. And I feel we need to really think about which disease we are talking about, and how, as I told you, it is holistic. You need to have either you use modern system or traditional system. I want to tell you one thing: you need to have a healthy lifestyle. You need to have a holistic approach. It starts from early in the morning when you get up and ends when you sleep. Even it will not end when you sleep also. You need to have a perfect sleep, even for that matter, for many diseases. It may be a very common disease like a cold. For that also, you need to have a good, healthy lifestyle and hygiene, cleanliness, and also perfect lifestyle.
Thank you, ma'am. Behalf of our audience, it was a very informative and interesting presentation, ma'am. Thanks a lot. Over to you, Ms. Jovita.
Thank you, Ms. Ajita. For our next keynote speaker, I once again welcome Mrs. Padmashri Murthy, who is the president of Sarva Sumana Association Bengaluru, to deliver the keynote speech.
Good morning, everyone.
Yes,
Am
yes.
I audible?
Yes, ma'am.
Yeah. So just now, all of us heard Dr. Anuradha's speech. It is really a preamble to whatever I'm going to say. So I'd like to share the PPT also now. Yeah. So I will start with a story. Am I audible?
Yes, ma'am, you're audible.
Thank you, Jovita. Thank you. So let me start with a story. An old man was doing his daily walk along the beach one morning when he spotted a young boy bending down by the water, removing something up from the sand and throwing into the sea. The beach was normally empty at this time of day, and the old man was spotted to watch for a while. He noticed that the boy kept on shuffling a little further down the beach, then repeating this same action again and again, stopping somewhere, removing something, throwing, and moving. So the old man asks the boy, "What are you doing there, boy?" Then the old man walks closer to the boy. Then the boy says, "I'm saving these starfish that are homeless. Those are helpless," replies the boy. And he continues, if they stay on the beach, they will dry out and die. So I am putting them back into the ocean. so they can live. The old man was silent for a second. Then he says, young man, on this stretch of beach alone, there must be more than 100 stranded starfish, helpless, homeless starfish. And if you go further, there must be at least 1,000 more. So this goes on for miles and miles and miles. There must be millions of helpless starfish. I don't want to say this, but I don't think this will make any difference. So the boy replied while continuing his work, and he was throwing the starfish into the sea, and he said, well, I just made a difference for that one. And he continued his work. So I would like to ask you people to share your opinion on this story in the chat box. Meanwhile, I'll be continuing with my talk. Let us discuss about this later also. So what is storytelling and why is it important in counseling? I'm in this field since 2011, almost 11 to 12 years of counseling experience is there with me. I use stories mainly for students and also teenagers when they come to me for counseling. Storytelling plays a vital role in the process of assessment and intervention during the counseling. Children and teenagers can identify themselves with the characters of the stories what they listen to, through which they can overcome the difficulty of self-disclosure. Many a times I keep asking them the questions. So why are you here? What made you to come to me? But they hesitate sometimes. So when I tell a story, that would help them to like tell their problems. Stories can be narrated through pictures, pictures of animals, forest, king, queen, dinosaur, mountain, ocean, or mermaid, or unicorn. And also through art. We can use puppets, origami, drawing, painting, even dance and music. Through dance and through music, through songs also we can narrate stories. And verbally, like without using anything, any visual media, we can just keep on telling the stories so that the listener will imagine. If I tell her that there were a lot of birds on a tree, then the listener will imagine lot of birds on the tree. So I will not specifically show any picture of a parrot or a crow, so that the listener, it will remain in the imagination of the listener, whatever the bird he or she can imagine, that is left to them. That would help them to imagine many things, and stories can be told in all these ways also. So what is that these stories would like communicate? What happens if I listen? What is that I'm going to learn? Stories would help us to understand the culture. Culture means the food we eat and their dresses, what they are wearing. Different countries will be having their own dress codes, and they're very comfortable based on the atmosphere, based on the weather, and based on their comfort level. They will wear different dresses, and the festivals they perform, and the rituals they follow, games they play, and values, ethics. All these things comes under um, culture. And also the music they listen, the music they play, and the dance, and the education system, all these we can understand through a story. Stories of different countries globally. A lot of stories are told to children and even the middle-aged or all age group of people will like to listen to stories. And then what we learn from the storytelling is next is the sequence. There is a small shloka, Purvam Ramam, Tapovanani Gamanam, Vaidehi Haranam, Jatayu Maranam, Sugriva Sambhashanam, Vali Nigrahanam, Samudra Taranam, Lankapuri Dahanam, Paschat Ravana, Kumbhakarana Matanam, Etadhi Ramayana. So in these 4 lines, whole Ramayana is narrated. Like Purvam Ramam. Earlier, there was a king whose name was Rama. Tapovanani Gamanam, he went to forest. His wife was kidnapped. Vaidehi Haranam, Jatayu Maranam. Then Jatayu was killed. Sugriva Sambhashanam, he met Sugriva, Vali Nigrahanam, and then he killed Vali. Lankapuri Dahanam, whole Lanka was burnt. And then Ravana Kumbhakarna Pathanam, both were killed. Both were killed by Rama. Etadhi Ramayanam. This is the story of whole Ramayana. So if this is told to a student or to a child, then he will definitely understand the sequence. What happens next? What is told now? Then what comes? Then what comes? What is further? All these things students will be learning. This problem also I would see among students or the children of nowadays when they see the cartoons on the television or on mobile, the fast moving pictures, when they see all these things, sometimes it's very difficult for them to follow the sequence. And when they narrate something at home or while answering a question, the gap of following the sequence I would see. So that gap will be filled by this storytelling skill. And then gain knowledge. Through stories, we can gain knowledge, like even science fictions are there for technology, about a war or a vehicle, cooking. All these knowledge can be told in the form of stories. Ethics and values, like honesty, responsibility. When we tell a story, a child will understand what he or she learned from that story. At the end of the story, if we ask them, what did you learn? So they keep telling the kindness and self-discipline, giving respect to elders, compassion towards all animals, birds, plants, all these things, kindness. So all these values they are going to learn from stories. Influence in work and relationship. Even in workplace, when we interact with our colleagues, when we conduct meeting, while taking decisions or leading a team, these stories would help us. Relationship. When it comes to how to behave with a father, with our mother, brother, a wife, husband, in-laws. So if you listen to the story of Mahabharata, we will definitely understand how to behave with whom, how to put them in different containers based on the relationship, what we carry with them. And also, it helps to improve our communication and observe behavioral change also. Like after listening to a story, we develop a lot of insights within us. We change ourselves. I have seen children changing themselves after listening to this story, and they will connect with the character of that story, and then they will carry with that, saying that I'm going to continue with this. I will not tell a lie, and I'll be kind from now onwards. I'll respect elders. They will say all these things. And improvement in communication. Through storytelling, we learn a lot of words. Our vocabulary will be improved. and we know how to talk to the people. And it's not like only talking. Communication means whatever I want to convey to the other person, that also we will learn through storytelling. So in Indian scriptures, these are all the stories we can see, not only in India, as I told, across world. Lot of stories are there. In here, I have mentioned few. You can also put it in the chat box if you know other than this. Panchatantra stories, all of us have heard animals, birds, plants, everybody will be speaking, and they all show different feelings like desires, and then love, compassion, kindness, and Jataka stories, Aesop stories, fairy tales. All of us have grown up by listening to the story of Cinderella, Snow White, Sleeping Beauty, all these things, and epics, Ramayana and Mahabharata. Famous scholars have told like nobody is there in India who have not heard Ramayana or Mahabharata. Everywhere, wherever we go, we connect some stone, some river connected to Ramayana or Mahabharata. Sita was here, Rama just threw the arrow and water came out. And this is the stone lifted by Bhima. So we connect with all these things from the stories of Ramayana and Mahabharata. And many stories like Mantesami Kavya and Malay Mahadeshwara Kavya, Mailaral Mahalingeshwara Kavya, all these famous stories are there in Kannada. Through stories, we learn Kanta Samhita, Mitra Samhita, and Prabhu Samhita. These three mainly I also use during counseling. Kanta Samhita means like it's like a wife telling to her husband. There is a story in Ramayana when Rama and Sita were there in the forest. Sita asks very politely with Rama that Rama, you are wearing the dress of uh, like sannyasi, but you are wearing arrow and the bow. What does it show? Like all the uh, people who are doing penance here, all the ascetic people, they will be afraid of you, and even animals think that you have come here to kill them. But why are you wearing all these things? She definitely will not say directly that you should not bring all these things, or why are you wearing this dress? But very politely, she communicates whatever she wanted to tell him. Like that, the stories will communicate in the form of Kanta Samhita and Mitra Samhita. Mitra Samhita is like a friend. Stories act as a friend. It will tell us uh, sometimes with love, sometimes with authenticity, and sometimes with anger or with assertiveness. A friend will tell us what to do, what not to do. Stories will do the same thing. And Prabhu Samhita, sometimes it orders us. Stories will order us to do something and not to do something. So all these three factors we are going to learn from the stories of Indian scriptures also. And to that fact, we have the stories like Iliad, Odyssey of Greek, epics of other countries also, and folk tales. If you see the folk tales of the world, wherever you go, whichever country you go, you will see hundreds and thousands of folk tales which are going to communicate a lot of values to us. So now, let me just brief you about mental health. Just now, Dr. Anuradha also told us about this. Mental health, according to WHO, good mental health is related to mental health and psychological well-being. That is what WHO says. Mental health includes our emotional, psychological, and social well-being. It affects how we think, feel, and act. It also helps to determine how we handle stress, relate to the choices we make. Anuradha ma'am also gave the list of all the disorders. If you click on Google, like mental disorders, you will see some 2 to 3 sheets or more than that, on the screen of the Google. These are all some of the disorders I have just mentioned here. Anxiety disorder, stress and trauma related disorder, eating disorder, OCD, obsessive compulsive disorder, speech disorder, and dyslexia. Ma'am has told all these things in detail, so I'm not going to go in detail with that. So just I would like to share how this, I'm jumping from mental health to this, what is Panchatantra stories, how is it going to help us in mental health? I would like to narrate that. Before that, I would like to tell you something. It is to all the audience who are there with us today. All of you would have heard a lot of stories from childhood. Remember a story. Remember any story, a favorite story of yours, and remember all the characters of that story. Any story, could be any story. Remember all the characters of that story, and then connect with any one of the characters of that story. And then you can share which is your favorite story and for which character you have connected to. In the chat box, you can post, or if you want to unmute yourself and tell me, one or two can definitely share your favorite story here. I think a coordinator would help this. Jovita?
Yes, ma'am.
Yeah, you can just unmute anyone of, or you can also participate.
Could you please post your answers in the chat box, or else you can mute yourself. Guys, I once again request you to post your answers on the chat box.
Shall I repeat the question? If I thought of doing a few more activities, if participants are also interested and they are like listening to talk, then
yes,
ma'am. Not, yeah, I would continue. If not, okay. So the Panchatantra stories, I would like to just introduce how it was came into existence. There was a king called Sudarshan. The king had 3 sons named Bahushakti, Ugrashakti, and Shakti. Yeah, somebody has posted slow and steady wins the race. So I was asking, like, imagine a story and tell me with which character you have connected yourself with. So in this story, slow and steady wins the race, with which character you are connecting yourself with?
Probably the tortoise and the rabbit.
Yeah, so with both the characters?
Tortoise, I guess only one.
Tortoise, wonderful. 5?
Slow and steady wins the race.
Okay, wonderful. So you can just go through this, I mean, whatever you have told just now. What makes you think like that? Why you are connecting with tortoise? And then what made you to think about slow and steady wins the race? What has happened in your life? And with which instance you are connecting yourself with yourself? So you can just think about all these things. If it is a workshop, we would have discussed in detail, but you can think about it. We can connect in future regarding this. Okay, Jovita?
Yes, ma'am. Ma'am, it's Dr. Nandini's answer, if I'm not wrong.
Oh, wonderful, Dr. Nandini. Thank you so much. So it was for her. So I was talking about how these Panchatantra stories uh, like came into existence. As I told, there was a king and he had three children. Though the king himself was a scholar and also he was a powerful ruler, his sons were not of any use. They were very dull-headed, and the king was very desperate. So the king approached his ministers, and he wanted to bring his children, some discipline into them. So one of his ministers, his name was Sumati, and he says, if we want to teach them anything, or if we want to educate them by telling them about the scriptures or texts, it would be very difficult and it takes a lot of years. Some years they have to learn and practice all those things. It's not going to happen with them. So he suggested a name called Vishnu Sharma. Sumati says if Vishnu Sharma can accept these children to educate, then definitely something will happen. So Vishnu Sharma was invited to the court. The king offers him 100 acres of land if he could teach his princess. But Vishnu Sharma refused to accept the award. But he says that he is not going to sell his knowledge for the money. But he accepts the task and he took the princess along with him. Only by telling stories, he taught politics, leadership, discipline, everything to the children. Whatever the Panchatantra stories you are listening nowadays, all those things were told by Vishnu Sharma to these three students. So he also understood that the Panchatantra means 5 treatises that he used. Like first one is Mitra Bheda, that is the loss of friends, and Mitra Samprapti, the winning of friends, and third one is good versus evil or light versus darkness, and also the Labd, that is a loss of gains, and Aparikshita Karakam, that is imprudence. All these things he taught. Not only these 5, but many lessons he teaches through stories to all the princes. Now, not only telling the stories, during counseling, telling stories is one part. Then the second part is listening to the stories. After telling the stories, I would lend my ears to the client or to the person with whom I'm talking. They will start telling their stories. Some stories are joyous and some are tragic. Stories, whatever they are telling to me, that will tell me how they have adjusted to the different conditions of life and how they are living with memories of abuse or assault, and also how they have sustained during the loss of loved ones, and also coping up mechanism. Everyone has a story to tell, and everyone deserves a chance to tell his or her story. So this is what I told just now. So Dr. Nandini replied back to that. So what happens if I tell stories to students? I have told all these stories to them to motivate them to study, to do hard work, sculpture in the stone, and many would have heard this story also. Like a sculptor, he chisels and he chisels a stone which is lying at the bank of a river. It becomes a idol of Ganapati, and all the other stones which were around that Ganapati stone, they were feeling like, okay, thank God I was not the one chosen by the sculptor. Whenever he was hitting the hammer on the idol of Ganapati to give a shape, that stone was crying and sighing, and it was undergoing lot of pain. But at the end, it was the idol of Ganapati, and all the people started worshipping that idol, and they were pouring tender coconut, milk, and honey, all those things, and they were lighting the lamp. At the end, when it came to offer the tender coconut or the coconut or fruits, they were hitting the coconut on the stones which were lying around and offering the actual fruit to the Lord Ganesha. So all the other stones which were thinking like we were left, thank God the sculptor was not chosen us. They were like hit by all the devotees throughout their life. So when I tell the story, student will understand, okay, this is the time I have to do hard work. So I tell the story at length and not to procrastinate, as just now Dr. Nandini told, tortoise and rabbit story and importance of friendship, ants and the elephant, all of us have told. Elephant always believes that I'm the strongest animal in the forest and nobody can beat me, but ants, when they were just getting into the ears of elephant, he suffers a lot and he surrenders. So when the snakes or the birds came to ants and took the help of ants to teach a lesson to the elephant, this teaches the lesson of friendship. If somebody is not mingling in the classroom with other friends, and not to hesitate to seek help. Eagle story is there, planning, king in the forest. This story is also like uh, every 2 years there will be a city, and once in 2 years a king will be selected by an elephant. After 2 years, the king would be sent to the forest, which is on the other bank of the river, where all sorts of animals, like wild animals, would be there. Nobody knows what happens once he is sent to that forest. So meanwhile, one king who was very wise, he makes all the plans in those 2 years. He builds a new city on the other bank of the river. By the time comes, he goes to the forest, there would be another city by itself. So how planning works. So after telling the story, a student would definitely try to plan for the forthcoming examination or the final board examination. So these stories would help them. And then this is also one of the activities. Jovita, if any 2 to 3 or 4 volunteers come forward to choose one of the sentences over here and think for a while, and then they can share whatever comes to their mind by connecting with the instances of their life.
Okay, ma'am.
Yeah.
Are there any volunteers for this activity?
They can choose any one out of these 4.
And yeah.
Yeah. Last time when I told a lie, the best gift I received till now, the risk I took changed my life. If I was raised differently by my parents.
Ma'am, Dr. Nandini Rao, she like to
Yeah,
try.
she can unmute herself, or she can post in the chat box also.
Okay.
Sure, I would like to talk here.
Yeah, ma'am, please.
So the third one, the risk I took changed my life, is that, I was working in Manipal, KMC Manipal, and I was a faculty over there. And then I got a postdoc fellowship in the US. And to go there, I had to resign. But I took that risk. I resigned, went there, and I got a great experience in the research labs. And then because of that risk, now I'm a professor in a medical school in American University of Antigua. So that risk really changed my life. And if I hadn't taken that risk, I would still be a faculty, But I wouldn't have had these kind of experiences outside India. So that is my story there. And I really
Wonderful.
love your concept of telling the stories. In fact, even I'm thinking of, I'm a biochemist. So, you know, biochemistry has many pathways, cycles, etc. And the medical students do think, why should we learn these pathways? And it's hard, it's more of memorization. So I want to implement this kind of concept where a pathway can be told as a story. And now I'm getting new ideas where I can incorporate some of our own Indian stories in these pathways. And thank you very much, Dr. Murthy, for that.
Thank you very much, ma'am. So just one more question, Dr. Nandini. At that time, when you took the risk, what was your like feeling at that time? What were you thinking? As you told, after all these years, now you are a researcher. But when you took that decision, what was going on in your mind?
It was a mixed feeling. You know,
Exactly.
I was scared. I was also excited. So it was
And-
really a mixed feeling. And so when I say scared, because you know, I was thinking, wow, I have a great job here. And do I really need to leave such a secure job and go?
True.
So,
Exactly. That's what I wanted to understand. Yes, whenever we take a decision, we don't know what is going to happen in future. At that time, that mixed feeling would be there, and we will be scared of future. Normally, we hesitate to come out of the comfort zone.
yes.
So yeah, I really feel very happy for you. You took that risk. Congratulations. All the very best, Dr.
Thank you.
Thank
Thank
you.
you, Dr. Murthy. Thank you.
Anybody else? Uh, any of the students? Okay, so I will come to the end of this activity then. So I would like to conclude here. Stories will teach us lessons, and they share messages and motivate others. Some stories are happy, while some stories are very sad. While counseling teenagers, adolescents, when teaching happens about the human behavior, family, all these things, a lot of challenges will come. I use therapy also, but sometimes, not sometimes, many a times, I have found that these stories would play a very important role, and students and even parents, they feel connected, and they have resolved many of their issues. So thank you very much to everyone.
Thank you, ma'am.
Yeah.
If there are any questions from the audience, you can put them on the chat box. Thank you, ma'am, for an interactive session and also being an inspiration to us. Over to you, Ms. Ajita.
Thank you, Ms. Jovita. As the Kenneth R. Miller once said, our own genome carries the story of evolution. Written in DNA, the language of molecular genetics and narrative is unmistakable. As a representation of this quote, I will take this privilege to welcome Dr. Krithi S. Jwalar, She is a passionate homeopathy healthcare strategist, a poet, a writer, a storyteller, a counselor, and a psychotherapist who believes in bringing about the holistic approach to health. She has completed her BHMS in 2009 from Rajiv Gandhi University and has completed MD in acupuncture in the year 2014. She is the director of Vices Genomic Lab Private Limited since 2021 and is looking forward to building up research in the field of homeopathy and genomics in the future medicine. Her vision and purpose is to provide homeopathic medicine as a prophylactic in pregnant women, similar to the field of genetic counseling, which ensures a discontinuity in transference of genetic hereditary illness from mother to child and reduce vaccination in infants and small children. We welcome you, ma'am.
Hi, Ajita. Can you hear me? Am I audible?
Yes, ma'am, you are audible.
Okay. Screen is okay, fine?
Yes, ma'am.
Okay. Right. Thank you so much for this kind introduction. My name is Dr. Kirti Javadkar. Okay, I think you got it a little wrong. All right. My today's topic is, can I share the screen, Ajita?
Yes, ma'am, you can.
Okay. Voice is also clear, right?
Yes, ma'am, yes, ma'am.
Okay. Can you see my screen?
It's visible, ma'am.
Okay. How do I make this full screen? So I have to go to the presentation view, right?
Yes, ma'am.
Okay. I can't find it. Just a minute. Okay. I'm not getting the full screen thing. I'm not a very tech savvy person. Sorry to take your time. Can anybody help me with how to do the screen completely, the whole laptop? How do I do it? I'm not getting the screen thing. How to go into the presentation mode?
Dr. Chavalkar, if
Yes.
you look at the menu at the top,
Yeah.
After slide, there is a slideshow option. At the
Okay.
top, in a file edit.
Master
Yeah.
file. Am
At the
I
top.
going the right place?
I'm
Yeah,
on top, on complete top,
No,
ma'am.
complete
File,
top,
edit.
ma'am.
Yeah.
At the top,
Ma'am,
where
to
you
the
have
left.
file edit view,
Yes,
next,
ma'am.
Yeah.
next slide,
Yes,
you
ma'am.
have slideshow.
Yeah, this one.
Next
On the
slide.
slideshow, ma'am.
Okay, I think I have this one here. To move this. Oh, okay, yeah, now yeah, I got it. Yeah, okay. Thank you so much. Okay, this is F5. All right. Yeah. So just a minute. Okay. Yes, thank you for the introduction and sorry for wasting your time. This is Dr. Kirti Javadkar. I finished my homeopathy and I've been practicing homeopathy for almost 15 years now. So how do I go to the next slide? Just enter. Just a minute. I'm not understanding how do I go to the next
Ma'am, your
slide.
keypad.
Yes, ma'am. You can
Using your
use your
keypad.
key, arrow key.
Arrow key, right? It's not
Yes, ma'am.
working. Acha, okay.
Yes, ma'am. It's now. Yes,
Right. Okay, I got it. Thank you so much. Okay. So what we need to understand here is we're trying to mix two wonderful system of sciences. One is genomics and one is homeopathy. So while we are coming to an understanding of the topic, we just have to go by basics. Okay. While like how I started, like understanding genohomeopathy, like it's famously called. While genomics includes scientific study of complex diseases and the change in genetic patterns due to environmental changes, influences, genohomeopathy includes observatory changes seen in genetic patterns using homeopathic drugs on individuals. So here what we are doing while the genetic study is going on, when the genomic study is going on, we are giving homeopathy to this individual and seeing what are the kind of genetic patterns which are being changed. Fine. So how is the data collected for the genohomeopathy? By drug provings. We simply do drug provings by DBDT method, which is direct blind drug trials on healthy men, women, and children, and people above 60 years of age. So what we do, we take down these symptoms, which is physical, mental, emotional, and changes which are very minute, as small as being sensitive to environment or any kind of sensations from where the pain is going, which side is going, and how it is traveling, the kind of trauma the person is going through. All these information is taken in the DBDT trials. Now, and the drug provings, our data is collected when we are having to treat a person during illnesses. So the same thing, mental and physical signs and symptoms are recorded during that time. And when you give the medicine, this data is collected. And sometimes during accidental provings. Accidental provings is when certain changes are seen. When the medicine is given, when we don't actually understand what is the medicinal quality, but you get to see some changes. It is like an accidental proving, okay? Or accidental recording, if I should say. So these are the ways how the data is collected. And also through previous cases or previous histories. Like, you have a data of a lot of patients. So by data analysis also, we get this data collected. Fine. Next screen, just a minute. Yeah. So the genomic, is my screen completely visible? Oh, yeah. Yeah, the genomic perspective during drug proving. So what are the perspectives we have to keep in mind when we are doing a drug proving from a genomic aspect? So samples are collected in the homeogenomic perspective, I mean. These samples are collected mostly saliva, and in very rare cases, blood sample is taken. And at every stage of illness, either noticed during drug proving or illness is seen. So any, just like now you've given the medicine to the person and then you're taking the samples, then again some new symptoms come up, again samples are taken. So that's how samples are collected. So DNA assays suggest changes upon behavior of human organisms under ultra-dilution that works at a quantum level is noted. So when we are seeing these samples during the drug proving or the genomic, during the drug proving, a lot of genetic changes are seen, and the medicine which is given is beyond the quantum level. We'll come to understanding that. Different gene expression profile of cell systems treated with same drug with different dilutions suggest the existence of hormetic mechanisms. Genetic expression profile treated with copper, apis mellifica, gelfinium, these are all homeopathic medicines, are characterized by same common denominator of the concentration dependent inversion of genetic expression that justify the simile concept of homeopathy. So simile concept and all that also we'll understand in the further discussions. Unprecedented therapeutic application is to optimize the use of nanogene based medicines with a low dose potency or pharmacological based on a reliable experimental platform. So what we're trying to do here is we are studying the genes, the genomic patterns of a particular set of genes under the influence of homeopathy. And this is going on during the time of drug proving. Fine, so it's a very complex kind of a thing. So for that, we need to understand what is the science and philosophy of homeopathy and why mixing these two fields is going to be beneficial to the society. So understanding the starting of homeopathy, homeopathy was established in the year 1796 by Christian Friedrich Samuel Hahnemann as an alternative system of medicine when he was proving cinchona bark on himself. So what happened is he was suffering from cholera for a very long time. And then he came across this book called Medicina Alternatia. And he found that the cure for cholera is by taking a small, thick, concentrated juice of cinchona bark. So he took that juice, and then he realized that most of his symptoms were getting more. Like he started with more chills and more, diarrhea and very erratic temperature rise and all that. So when this was happening, he started to read that book completely, and then he understood that principle of homeopathy cannot be, giving more medicine to a person and making the person suffer a little more. So he went to come to the principle. Then he brought up these principles, saying that similia similibus curantur, or something like cures like. Like cures like is a principle which we use to a person who is suffering from any illness, and we find a medicine which is very similar to that illness, and we give it to them. Okay. So, and also we have a lot of philosophy and everything in homeopathy, and concising it is really a big challenge. So just grasp as much as you can, and then we can discuss it at the end of the session. Homeopathic philosophy is also based on the principle that cure should take place rapid, gentle, similar, with harmless, with the least harmless way, and with a minimal dosage. So after this Syncona experiment, he realized that this much of medicine is not required. I just have to minimize the dosage. So then came the law of potentization. Law of potentization is a law where you take one part of the medicine and dilute it to 10 parts of water or alcohol, or otherwise we call it as vehicles which are neutral in their nature. Okay. So we take one part and then 10 parts, and then from that one part is taken and again diluted in another 100 parts, and the same 100th part, one part is taken and diluted with 1000, and so go on further, and further goes on the dilutions. So every time a medicine was diluted, what Hahnemann observed was it started reducing the side effects and it started making him feel better. So he started, then the dilutions kept increasing, it went up to 50 millismol scale. So 50 million thousand times, it means that one drop of medicine is diluted 50 million times. Yeah, so that's the kind of dilutions which we follow in homeopathy. So these medicinal substances had gone to a level where they surpassed the material dose, and only the medicinal action effect of the medicine was there. This is called as drug dynamization. Okay. Homeopathy also believes in a holistic approach of treating an individual with a psychosomatic or somatopsychic occurrences, which means that a person does not suffer only with one part of their body. If they suffer on the whole. Now, if I have a headache, I do feel the pain. I do have discomfort in my body. I do have body pain. I have muscle cramps and things like that. I get irritated with sound, noise, or light. So these are the things which he thought was important to include in his recording or during his drug provings. Okay. Yeah. Yeah. So that was what came to an understanding of. So I was in the fourth slide, right? Yeah. Understanding of where am I? Yeah. So yeah, drug dynamization. So homeopathy also believes in the holistic approach, like I said, and there are about 8,661 homeopathic medicines that have been proved and are in the use, out of which 6,107 medicines have completed their symptomatic representations on the physical level, and about 50 to 60 remedies are showing its actions on the mental, cellular, somatic, and intellectual levels, which we famously call them as polychrist remedies. So the next slide comes into understanding the philosophy and why this science is so huge and why it can mix with a genomic study. So we also have something called theory of miasms, where there are 3 miasms. Miasms are polluting agents or agents which bring about disease in your body. So, sorry. Yeah, disease in your body, and there are 3 types of miasms, which is mainly psora, syphilis, and psychosis, and one other one is a tubercular miasm. So why are they called as miasms, and why are diseases or illnesses formed into 3 different types? Because it's because of its manifestations, its pathophysiological changes, and the kind of mental disturbance a person goes through, the physical appearance of the disease, the discharges, and the kind of morbidity which it brings, the kind of damage it brings to an individual. Okay, so that is the psora, syphilis, and psychosis. Psoriasis is very, like for example, psora is very basic. Psychosis becomes skin rashes, very hard eruptions, like very pus formation, very offensive smelling kind of illnesses, and illnesses which are deep rooted in your organs and things like that. And syphilis is about damaging, where the apoptosis of cells takes place, where cells keep destroying each other, like in cancer. You first have growth and then you have destruction. So you have growth and destruction both happening at the same time. So that's how these 3 miasms were formed. And then we also include something called temperaments. These are also inherited internal constitutions or a state of internal brief manifestations on the physical mental levels. Now there are different kinds of temperaments. Why I'm saying all this is because we have a lot of structured understanding of illness which is happening in homeopathy. So we have something called as phlegmatic temperament, which really brings about a lot of phlegm. You have these individuals having a lot of cold and cough all the time. That these are the phlegmatic people. The melancholic temperaments, people who just blow out of their mind for every simple problem. Sanguine temperaments are people who are very broody and who go into a dull kind of dull and sunken kind of reaction. Nervous and choleric temperament. You don't need to know all of them. It's basically to understand the science of homeopathy and how we can join it with genomics. And the principle of diathesis. Diathesis is also a predisposition to certain illnesses, mostly on physical aspects due to inherited tendencies. Now, you can find a lot of people saying that, yes, my mother had piles, even I'm having piles. I'm having, my mother had this, my father had heart illnesses. So even I'm having heart illnesses. So it's something like that, BP, diabetes and things like that. So these are termed as diathesis. These play an important role in the type of disease manifestations one goes through. So there are hemorrhagic, lymphatic, and tubercular diathesis. And there's also a theory of individualization where a medicine is prescribed on the totality of symptoms from head to toe recorded on a similar medicine which is prescribed, believing that only a similar, more superior form of energy in the form of medicine will be able to bring out cure. So when we are doing this drug proving, I had mentioned that we take down all the mental symptoms, all the physical symptoms. Why are we doing that? We put a person into observation for 1 to 2 months. We take down all these symptoms from the patient and we keep it aside, saying that this medicine brings these kind of symptoms. Now, when I get a patient with a similar kind of symptoms, we use that same medicine and we give it to the individual. But the only difference is that we modulate the dosage, or we modulate the potency, or we modulate the force of the medicine which has to be given. It's believed that only a similar superior power can overcome the disease situation. Fine, we'll come to understanding that also. Why I'm telling you all this. Okay. So homeopathic pharmacy and pharmacodynamics. Since we're dealing with genetics, we have to come through pharmacy and pharmacodynamics also. So what do we do in homeopathic pharmacy is, first is a selection of the right medicine source. So we have medicines where we prepare from metals, from non-metals, from acids, from salts, from bases, from plants, roots, fruits. You just name it. You have it. You have even medicines from sun and soil and even the sea. Okay, seawater. So yeah, so these are the kind of medicines these are the sources of medicines which we get, and methods of extracting the medicinal properties and preparation of these medicines are also studied. So how we have to bring out only the medicinal property of these medicines. We don't want the other, hazardous aspects of the medicine or the poisonous aspects of medicine. And then there's a potentization, which I mentioned earlier, and pharmaconomy, or the methods of application of medicine. Now we have different methods of how we apply it. I can either smell it, apply it on my skin, or use it as an external application, take it in orally, like oral medicines, how they are usually taken. So these are the various ways of taking the medicine. Dosage, preparation, and repetition. This is what we learn in pharmacy. So when you're doing the drug proving, we understand that, at what point the person stops giving symptoms and at what time we should give the dosage. So this makes us understand that minimum dosage given to a person of a different diathesis, temperament, and miasm, which is working in them, will make sure that we're giving just enough medicine to bring about cure in them. Okay. And the types of homeopathic medicines we can give. So we give it in the form of pills, we give it in the form of external applications, we give it in the form of mother tinctures, and we also give it in the form of tablets. Okay, simple tablets kind of. And recording the molecular and structure. And this also enables us to record the molecular, structural, systemic, and mental changes in a person when we're doing the pharmacy or the pharmacological studies of homeopathic medicines. Now coming to pharmacodynamics, why am I putting it as under pharmacodynamics is because our medicines are diluted to 1:50 million times that it even crosses the Avogadro-Loschmidt limit. Okay, so the drug receptor interaction is noted. So this, when a medicine is diluted to a nano level or even lower to that, to a quantum level, if I should say, what is the kind of reaction we see, and what is the dynamic differences or changes we see at the genetic level? So these are the genetic, genetic kind of changes we see. The drug receptor interaction is noted. Mechanism of signal transmission and amplification also was noted when we gave these highly diluted medicines to individuals. Very low dilution shows changes in biological system to bring about exogenous and endogenous signals by creating sensitivity by bioelectromagnetic information signaling and regarding bifurcation points of systemic network of the genetic patterns. So nanomedicine creating a greater plausibility with homeopathy. When we're seeing these changes, when the medicine substance itself is not seen, but still you're seeing these genetic changes, it means there's something else that is to be worked out, and that homeopathy is showing evidence in its functioning, right? So homeopathic cure. And while we're seeing the genetic changes, we're also seeing a betterment or cure which is taking place in the person. The person is not just feeling, his symptoms are not just going off, but he's feeling better mentally. His skin is better, his sleep is better, his, food is better, his tiredness, lethargy, everything is better. So why are we, and we need to observe these things as homeopaths, because there is something called homeopathic cure or the law of cure, which is by Herring's law of cure, which is different for acute cases and chronic cases. So we're following this to understand the physical, somatic, cellular changes in every individual. Emphasis on cure plays most important role in homeopathy, as mere relief of symptoms and signs may not indicate the removal of disease in itself. So some people say, I was better taking this medicine, but I came back with the same problem after 2 months, after 1 month. So that means that medicine has not acted upon in a level to have removed to have removed its effects from the body on the whole. So that's why we have a lot of laws and cures where we take care of all these things. Fine. So that's how the homeopathic cure takes place. Now, this is the part which I want to emphasize on: genomic study in homeopathy. When we took a lot of medicines and we started giving it to people, and we took their saliva samples and we kept reading it for over like almost 5,000 to from ranging from 5,000 to 50,000 people or volunteers, we saw their genetic patterns coming across these changes with homeopathic medicines. A combination of genetics with homeopathy suggests that every individual has a gene which is particular to himself or herself, where 99% is the same and only 0.1% is variable, which is responsible for hereditary problems. Through genetic testing and samples collected, can predict the health of an individual by 10 to 15 years by revealing the pre-genetic modifications, genetic blueprint, rearrange many genes, downregulate the expression of almost 50 different genes. So these are the kind of observations we saw in these individuals over a period of time when they were taking homeopathic medicines. In the field of cancer, which has been a very long study which has been going on, in the field of cancer treatment, the role of P53 gene in blood, which restricts the growth of cancer. Homeogenetic studies focus on identification of remedies that support these expressions in TP53. Medicines like Scrofularia nodosa, Phytolacca decandra, Pondorango, Thuja rota, carcinosin, and Lycopodium have shown required contributions during this study. So, like you all know, P53 gene is there, which restricts the growth of tumor cells, and that is reduced in the blood when you're attacked by a tumor, a cancer kind of a situation. So these medicines have shown to have increased the support of P53 in the genetic level, so as to help in resolving the cancer tendencies. Right? Homeogenomics evidence and the future of medicine. Why homeogenomics is the future of medicine? We have seen DNA demethylation. Yeah, we have seen DNA demethylation and hypermethylation was seen with a medicine called Marsdenia condoranga in the treatment of tumors. Transcriptome analysis with highly diluted medicines of gelsemium, which is also another medicine, Gelsemium apis mellifica, was seen in prostate cancer. Homeopathic drugs given in tincture form. Tincture forms are the crudest forms where you don't dilute it so much to even the quantum or the nano level. Show downregulation of genes in many diseases and more personalized medicine based on molecular genomics and homeogenetic analysis data makes it as the future of medicine. Now, these are just few medicines which we have handpicked and we have given it to a particular condition. But when the studies are even more deeper, you can go into various aspects, not just a tumor growth or cancer or anything. We have medicines which can even bring about changes in structural changes in heart by controlling the cholesterol levels and showing the opening of the blockages, minute blockages in the heart, structural changes in the kidney, where we have a lot of renal failure or chronic renal failure conditions, and the whole functioning of the kidney has improved. We have medicines which can remove the kidney stones. They're just mere medicines. We don't ask them to go to any surgical procedures. They're just mere medicines. When given, they bring about these changes. We have wonderful medicines for skin illnesses, from any kind of skin conditions, which may be hereditary. Hereditary skin conditions are a little challenging, but otherwise you have wonderful medicines even to bring out the any skin pigmentations and things like that. So that's a very limited study which has been happening in homeogenomic field, and to talk about this topic in itself is very challenging. But whatever information we have got mostly has been focused on cancer and immunity based studies. But the other studies are yet going on, so we might just see a lot more research papers on that. So scope of research and collaboration and conclusion about the topic. Having understood a brief insight about the research and study based on genomics or homeogenomics, the suggestive future and approach to any illness has to be refined so as to amalgamate the science of genomics on an holistic basis to bring about a wholesome approach towards an individual. Now, if you want homeopathy and genomics to come together, it should be a wholesome approach. It can't be just simple genes and simple medicines. No, because we are dealing more with a holistic approach. We are dealing with more mental changes and a lot of somatic changes, cellular changes, mental changes. So we have to include all these things. Environmental influences, of course. So these are the things which has to be taken care on a very broader perspective. And in the field of genetic counseling, like in my introduction, yes, it has a great scope in the field of genetic counseling, where we can, to a certain extent, stop hereditary illnesses coming from parents to offspring. So that's a very challenging kind of a field. And also it is useful in diagnosis, prenatal tests, like I said, you have the double marker tests and things like that normally, which is done. So you can give the mother homeopathic medicines and see if these double marker tests turn negative in mothers who have shown a history of miscarriages or a history of deformities in children when they're pregnant, infantile deformities and things like that. So so you can use homeogenomics in prenatal tests. Hereditary dispositions, like I said, epigenetic factors will help us assess the risk involved and how homeopathy can play an important role. So it's a very vast subject, a very vast topic, which has not been explored actually, but to talk on it is, I'm trying to make it as simple as I can, okay? Right, yeah, and the future of homeogenomics. What can we look forward in homeogenomics? Like a few examples I gave, removing the tendencies from mother to child, removing the tendencies in renal cases, removing the tendencies in BP, removing the tendencies in cardiac issues, cardiac illnesses, removing the tendencies with digestive and liver related illnesses. So mostly they are lifestyle related. But when we come to a genetic diathesis, like I said, when we're coming to a diathesis, we can stop the diathesis at a certain level. So homeopathic medicines are used during pregnancy to ensure that the hereditary diseases and tendencies and a chain is disrupted, discontinued from parents to child, and early intervention to ensure the health of the child and future health, resulting in less health hazards and more qualitative and productive future. So this is about homeogenomics on the whole, and also in the field of cancer, which is very, very rapidly picking up. In cancer, what we're doing is we're giving chemotherapy, we're giving homeopathic medicines at the same time. And we're seeing that people who are going with chemotherapy or even drug, even medicines when they're taking them, they are not showing that many bad effects, side effects of chemotherapy or radiotherapy for that matter, when they are on homeopathic medicines. So there is something which is giving them a back support during these treatments. And their recovery rate and the time of recovery to normal health is also much faster when compared to people who are only taking chemotherapy and radiotherapy. So yes, this is the future of homeogenomics, and it would be wonderful if the health department and health sections get to do more research and funding for these topics, so that we can be a little more useful to the humanity and do our bit in that. So with this, I'd like to finish my presentation. I don't know how much I was able to reach out to you people, but yeah, I think I can take a few doubts if there are any.
Thank you, ma'am. Thank you for the interesting talk.
Thank you.
Ma'am, there are a few questions from the audience. Can I ask one by one?
Yeah, you can.
Okay, ma'am.
I'll try my best to answer, okay, because I'm not from a genomic background, but I'll try my best, yeah.
Yes, ma'am. There is a question from Dr. Pinon Bakshi that is nanomedicine and homeopathic medicine can be related?
Yes, nanomedicine is based on the understanding that very minimal medicine is required to bring about cure, right? That's what they do in nanomedicine. Very small doses are injected into you, and that works as a medicine inside you. So we don't inject homeopathic medicines, but yes, we give them in an oral way. And this can work as a nanomedicine because we are surpassing the material dose. So I can state it as nanomedicine because we are surpassing the material dose of the medicine. And now that we have seen the genetic changes also after giving homeopathic medicines, I can say that yes, it is a nanomedicine. Yeah, it can be.
Does the dilution are same, ma'am? For the nanomedicine as well as homeopathic medicine?
Yes. So what happens in homeopathy is we take one part and we keep diluting it, right? So when we come to 300 dilutions, okay, which we call it as 30C. So beyond 30C, you can't find a material dose in the medicine. So from 30C to 200, you'll find no medicinal substance in the homeopathic medicine. So when it has crossed that threshold, you can put it into a nanomedicine, and it's basically an energy medicine or a quantum medicine, I can say. So nanomedicine, I'm not too very sure, how many dosages and what is the repetition of dosages they give in nanomedicine. But in homeopathy, we look at the signs and symptoms, and like I said, the mental improvement, the physical improvement, the molecular, cellular, structural changes, everything improves. We obviously need the laboratory tests also to support our claims, to support our understanding if the person is going on the right path of health or not. So yes, we can. It's beyond 200 potency or 200C that medicine loses its material substance. Yes. And repetition is based on the patient's soul. Yeah.
Thank you so much, ma'am. And another question from Dr. Nandini Rao.
Yes.
How do you check drug receptor interaction?
Okay, now this is a genomic question. It has just happened. Okay, I'm just telling you what research has happened behind that. how do you know if it is see, it's a very detailed kind of a genomic thing, drug receptor interaction. I have to go deeply into that, okay, because it just states that genetic patterns are changing to support each other and bring about health. I'm not too sure about that, Dr. Nandini, because I'm not basically from the genomic field. What I'm trying to do is I'm just learning everything from scratch. So drug receptor interaction, I have to understand that. Maybe it's something to do with you know, cell membrane or DNA or something like that. I'll get back to you on that for sure. You can DM me. Yeah, thank you.
Thank you, ma'am.
Can
And
I just
yes,
add
yes.
something on this?
Yeah, please, Dr. Pinanya.
Yeah, in homeopathy, even in Ayurveda, I have never come across this term like drug receptor interaction. Whereas in our biotechnology or bioinformatics, we rapidly use this term. So, if at all we can see this particular terminology even in Ayurveda and homeopathy, it will be wonderful. But so far, I have mainly in bioinformatics, I have seen this.
Okay.
This term,
Yeah.
drug receptor interaction.
Yeah, because when I was going through the studies also, I never came across this, and I'm a little put into a thought process. You know, when you ask me that question, maybe something can be done. We just have to figure it out. Yeah. Yeah.
Thank you, ma'am. Thank you, sir.
Yeah,
There is
thank
another
you.
question, ma'am.
Yeah.
In P53 study, which cells were used?
Okay. I have very little knowledge about what cells were used, but what happened? See, I really don't know about the genomic level. Okay, but if you want, I'll just give you the articles and you can explain it to me. That will be much better, because when I was reading this, I'll only read it from the homeopathic perspective. I'll not read it from the genomic perspective. So P53 cells, what I understood so far is these cells seem to reduce in cancer conditions, right? And when we gave homeopathic medicines, the TP53 started working, and that's how the cancer growth was controlled. So that's what I could understand, because I don't know what kind of I don't know if I've answered your question, but I need to go more deeper to understand what kind of cells were used in that. I'm not too sure about it.
Yes, ma'am. Thank you so much.
Yeah.
Audience, do you have any questions? Please do post in the chat box. Yes, ma'am. Thank you so much, ma'am. There is no question no more.
All right. I don't know
Thank
if
you,
I've
ma'am.
cleared your doubts or put you into more doubt. Okay. But yeah, like I said, this topic has not been thoroughly put up in any source. If I have to, confess. So writing about it was quite a challenge. But for me to understand that homeopathy is working on a genetic level itself is an overwhelming fact. So if I go more deeper into studies of genomics, then maybe I'll be able to answer you better the next time. Yeah. I hope I've done justice to the topic. And yeah, thanks a lot. Thank you for giving me this opportunity to talk about this unusual topic. And yeah, all the best. Thank you.
Thank you, ma'am.
Can I stop sharing?
Yes, ma'am. Yes, ma'am.
Yeah.
Ma'am, Dr. Nandini Rao has asked that could you please share the article with her?
Okay. The article on which one?
On this topic, ma'am.
Okay, yeah, I'll do that.
P53, about
Yeah.
P53.
Madam, just I think I'll just leave my contact details and my LinkedIn account to you guys. So you can just get in touch with me. It will be more helpful to me also. I'll send you my email ID. Okay. And Yeah, if you DM me, I'll be able to give you my LinkedIn account as well. I'm not able to. Yeah. Thanks. Thanks
Yes,
a lot,
ma'am.
Dr. Nandini. You put me into a new thought process. Yeah. Thank you so much.
Thank you, ma'am.
Thank you. Yeah.
In this busy world, all we need is purest love and even sometimes a lunch break. Catch you all after a great nutritious lunch. Kindly assemble back at sharp 1:45 p.m. Thank you.
Thanks a lot, Jovita. Thank you, everybody.
Thank you, ma'am.
Shall we leave the meeting?
Yes, ma'am.
Yeah. Thank you.
Good afternoon, all. Welcome back, everyone. As the section is about oral presentation, we would like to instruct every presenters to be ready with their presentation. The instructions are: the speakers have timing of 6 plus 2 plus 2 minutes for the presentation, 6 plus 2 minutes for presentation, and 2 minutes for discussion. As the indication of 6 minutes has passed, there will be a buzzer sound. Completion of 8 minutes will be also followed by the buzzer. However, due to paucity of time, we would request the speakers to limit their respective presentation in 8 minutes. Over to you, Ms. Anusha.
Thank you, Ms. Ajita. We request the oral presenters to switch on their video while presenting. Hope the instructions are clear. We shall start the oral presentation of the day. And our first oral presenter is Egna Narayanan Venkataraman and P. Kalai Selvi. Ma'am, you can start presenting.
But I cannot share my video. Everyone, my video is shared?
Yes,
video
ma'am.
is visible?
Yes, ma'am, your screen is visible. You can start, ma'am.
Okay. Good afternoon to everyone presented here. I am T. Kalichalvi, full-time research scholar, Department of Mathematics, Kalangsalam Academy of Research and Education. My supervisor, Dr. Egna Narayanan, sir, same department. Our paper topic is dominating sets in protein-protein interaction network. What do you mean by dominating sets and protein-protein interaction network? My voice is audible?
Yes, ma'am.
Protein-protein interaction networks are visual for explaining the structural function architectures of the cells. To have your comparisons of mechanism finding deriving proteins that are virtual for the control of the site never is a pertinent task in system of biology. Lately, it has been studied to find the smallest set of derived nodes to control the whole network. How the dominating sets are used in PPI to apply and on network, finding the smallest are undirected. Here we used the graph that are undirected. Nacher and et al. Viewed this problem for the point of finding the smallest dominating sets. In this paper, we explain the above proof how various centrality measures such as degree centrality, centrality, such as degree centrality between the dominating self help us understand PPI network. Denote a PPI. PPI means protein-protein interaction network. Interaction network by a graph G equal to B of B comma E, where B is the set of n vertices and E is the set of edges defined by adjacent matrix. We have a two protein between interaction between two proteins. We give a value 1 and otherwise we give a value 0. Also, we deem the G has no selfless loop as protein I. Selfless loop means a point join a point, a line join a point to itself. We call a set P subset of V of proteins a dominating set. Each of protein V belongs to V is either a member of P or joined by means an edge to a member of D. Degree centrality means DC means DC equal to degree of V. For degree means how many lines incident, that means how many proteins incident that vertices. Closeness centrality CC means CCV of V equal to summation V belongs to V of G degree of V. And subgraph centrality also. Whenever you feel like writing. Hello, ma'am.
I request Shri Rajeshwari to switch off. Can you mute yourself?
Okay.
Shri Rajeshwari Devi.
Okay. Wait.
Please continue, ma'am.
Oh, okay, thank you. The subgraph centrality characterizes the role of every vertex in the subgraph in a network. Subgraph centrality lesser size or given more weight than the ones with larger size. This attribute is APT for describing network modifiers. It can be obtained mathematically from the spectrum of the adjacent matrix of the network. This measure is more sharper to characterize the vertices in comparison to measure like degree between a centrality, etc. Strong correlations are there between various measures of centralities. In a graph critical, in this graph, we give a graph with between the centrality and the value of subgraph centralities. Suppose let G be a graph, simple graph with P vertices. It is known that the subgraph spectrum is the collection of those eigenvalues corresponding to the graph G adjacent matrix A. Suppose that A UR of I is the number of closed walks of length or in I starts and ends, which means it also called the local spectral moments, can be computed as the ith diagonal entry of the rth power of A. That is, UR of I equal to A power KII. These closed walks are linked to the subgraph of the network under consideration as subgraph centrality measure. This is that is the sum of closed walks of distance length with start and end with vertex I is the subgraph centrality. Protein interaction. The critical set is defined as the interaction among the created dominating sets as least size. Then it was further generalized to create A dominating sets of least size with large difference among all of them, where K is the number of dominating sets of least size. We give a graph G, we find out a number of dominating sets of least size. All the vertices in PPI network can be described as the critical set exactly defined at the Cray critical set. Further, a new set of protein appear in K-1 dominating sets of least size was collected, and this set was identified as the K-1 critical. We give a brief example. For example, consider the graph in the following figure. Here we have 14 vertices. Vertices number 1, 2, 3, 4, 5, 6, and 7, 8, 9. Note that there are only two vertices of degree 6, namely 1 and 2. 1 and 10, sorry. Here 1. 1, 2, 3, 4, 5, 6. And this also 10. P equal to 1. It's or P equal to 10 cannot be a dominating set. So because these vertices are not in adjacent in that 1. So it cannot find, it is not a dominating set. So a dominating set P of G should contain at least 2 elements. Suppose we form P received by adding both 1 and 10. And P reverse sets, then also P reverse is not a dominating set since 3 and 8 are not dominated. So we have paused the 3 sets and we have the combinating. In figure 1 shows a network instance G with where the verticals have different degree with different dominating sets. The dominating sets are here. Interestingly, the network has no critical vertex. 1, 3, and 8 are the intermediate vertex.
Excuse me, ma'am.
Yes, ma'am.
Ma'am, your time has been finished. Can you wind it up?
Okay, ma'am.
Yes.
Next, we give another example. This vertex, graph has critical vertex also, and intermediate vertex. All the vertices here. Next, conclusion. We give a brief about the importance of BPI network, and it is we have highlighted the pertinence of computing a dominating of least size and explained various types of proteins classification like critical, intermediate, and redundant in such dominating sets to facilitate certain biological process involving interaction of proteins. We also briefed about certain centrality measures such as between centrality, subgraph centrality are computed for the same network graph. We also pointed a mathematical truth related to subgraph centrality. That is also truth for all. These are my few references. And thank you.
Thank you, ma'am.
Thank you, ma'am.
Over to the judges.
The judges for the oral presentation is Dr. Ravinder Henry and Dr. Rajesh T.S. So, over to you, judges.
Yeah, thank you. The Kalachilvi ma'am, can you
yes,
hear me? Yeah.
Yes, ma'am.
Can you tell about that outcomes of your study?
How the dominating set is used in protein-protein interaction network, and we have to give how the minimum proteins used to in our biological technique.
Okay. Fine, we have so many bioinformatics tools also for this protein-protein interactions.
Yes,
like
ma'am.
Any other outcomes?
No, ma'am. We have worked on it.
Okay. Okay, fine. Thank you.
Thank you, ma'am. Any other questions?
Yeah. Are you a mathematician or a biologist?
No, sir. I am a mathematical student.
Okay.
Okay. Yes,
so, what kind of determinants? What are the other centrality measures that may look in the future?
Between a centrality, subgraph centrality, we have searched new other centrality, or we formed new centrality, half certified.
Are you going to work on these things, or you already compared?
Yes, sir. Working on it.
Okay, thank you.
Thank you, ma'am.
Thank you, ma'am. Thank you, ma'am.
I would like to inform the oral presenters that the judges for the oral presentation is Dr. Ravinder Henry and Dr. G. Dhanalakshmi, ma'am. We shall move on to the next oral presentation by K.S. Sri Rajeshwari Devi. Ma'am, you can start your presentation.
Hello. Hello.
Yes, ma'am. Your screen is
visible. Okay. I'm Sri Rajeshwari Devi, counseling psychologist, life skill coach, and grapho-analytical therapist. I'm an author also. Good afternoon, everyone. Today I'm going to present the topic about the relationship between the self-affirmation and life skills for better mental health and well-being for youth. The word affirmation comes from the Latin word affirmare, which means to make steady, confirm, or strengthen. Affirmations help you to alter your subconscious thoughts while repeating encouraging phrases, often making it more likely to believe them. In turn, you will start acting in the ways to make your affirmations become reality. According to World Health Organization, there are mainly 10 types of life skills, which are very important for everyone. Life skills are the skills required to deal positively with any challenge to get the desired. With positive life skills, you will get the ability to adapt to all kinds of circumstances and succeed in every aspect of life. There is a connection between neuroplasticity and affirmations. Neuroplasticity is the brain's ability to change, reorganize, or grow neural networks throughout the life, from birth to the end of life. The whole point of affirmations is to create positive change, to reduce suffering, and help you accomplish your goals and live a happier life. Affirmations influence thinking patterns, behavioral habits, creativity, health, and moods. Self-affirmations lower stress and rumination, which are major and anxiety. What?
Sorry, ma'am.
Okay.
Miss Kalaiselvi, please mute yourself. Yes, ma'am, please do continue.
It helps to develop positive life, leads to well-being and success in life. There are mainly types of skills: personal skills, thinking skills, and interpersonal skills. Self-affirmation will boost self-esteem. Affirmation like acts of kindness and love towards yourself create inner confidence that is beneficial for your entire life. Affirmation boosts self-confidence, self-esteem, and self-worth, which in turn reduces negative thoughts, which in turn reduces stress and anxiety. It boosts interpersonal skills also. Affirmation helps to perform better at work since it increases good communication skills, which in turn increases interpersonal relationship. As a counseling psychologist, I've introduced positive self-affirmations for many people as a part of therapy. It has helped them to enhance their self-esteem, self-confidence, decision-making, creative thinking, effective communication, coping skill, critical thinking, empathy, which are all good life skills. If one learns good life skills, one can lead a life with good emotional well-being and better mental health. Practicing self-affirmations are beneficial to all age groups, no matter what stage of life. Future research can add significantly by using mixed and qualitative methods and assessing their effectiveness in diverse age and cultural group. Life is full of challenges. Life skills are the skills required to deal positively with any challenge to get the desired results. It increases feelings of hopefulness. Affirmations are used to treat people with low self-esteem, depression, and other mental conditions. Rewire your brain for positive thinking and start living your best life with affirmations. So thank you all for your time. Have a good day.
Thank you, ma'am. Judges, over to you.
Yes, ma'am. In terms of, are you doing any MRI studies regarding the affirmations?
Actually, 10 years back, I met an accident. Okay, then I'm a big fan of Louis A. Then I started doing experiment with myself. I did affirmations. I was not able to move my neck also at that time, and I was not able to talk also. At that time, I did affirmations daily. I used to get up early in the morning, and I used to stand in front of the mirror, and I was saying like that, I can do it. I will definitely do it. Like that, after 6 months of physiotherapy, actually I felt good. All these affirmations worked for me. So I started doing research on that, and it helped my counselees also, like that.
Have you done any scientific studies using MRI or any other brain imaging systems?
No.
Okay. Okay.
We
Thank
have
you.
opportunity. Thank you.
Okay. Thank you.
Rajeshwari ma'am, I have one question.
Okay, tell me.
Neuroplasticity you tell about, no? There is any scientific proof for that?
Yes, that is like, see, when we are born, we will start to talk or see things. I mean, visualization will all start with this neuroplasticity only. We start the brain, in the brain, neurons will start rewiring and making connections. They with the synapses only, we all start seeing like that. This is a small example which I can share. By affirmation, what happens is our false belief will go. We will have more confidence, and brain, whatever we think, it thinks that it is true, and it starts giving us more confidence, and it will help for life skills. Like that, there is a connection, I think. Hello. Is there any question?
Ma'am, thank you.
Thank you.
There's one more presentation by K.S. Sri Rajeshwari Devi. Over to you, ma'am.
Yeah. Am I audible?
Yes, ma'am.
Yes. Yeah, I am Sri Rajeshwari Devi, and I am a counseling psychologist and a life skill coach. Today I'm going to talk about parenting stress and resilience in parents of children with the two most common neurodevelopmental disorders, namely autism spectrum disorders, ASD, and attention deficit hyperactive disorder. The objective of the study is to evaluate parental stress in the ASD and ADHD group, impact on family health, challenges faced by the parents, some of the feelings that the families may go through, and complexity of the stress. Some of the feelings families go through are mixed feelings such as denial, anger, anxiety, confusion, rejection, bitterness, self-pity, shock, sorrow, deep pain, and most important one is why me questions. Some mothers struggle with guilt and self-blame for something wrong they might have done during pregnancy or delivery. Most of the parents are worried about the future of the child's education, ability to become independent, marry, have children, and lead a normal life. Some of the parents have marital stress, financial stress, lack of proper social life, loss of relationship sometimes, unable to plan for the future, and no time for recreation. Balancing work and family life is very much difficult for them. They have to put extra effort by giving them time together, organizing the play area, and providing reinforcement and therapies apart from schooling. Most of the time, either elder or younger siblings of ASD or ADHD children feel unloved or ignored by all the attention and care that goes for the special child. That frustration or sibling rivalry might turn to attention-seeking behaviors. The sample size which I took is 20 parents with ASD children and 5 parents with ADHD children. And parents of ASD and ADHD willing to participate in the study were selected. A special school for special needs in Bangalore was chosen for this research. I made a Google form and sent it to all parents. Interviewed with each parent. Parents were given parental stress and coping inventory. And the result which I got is the 60% of parents had moderate stress, 27% of parents have mild stress, and only 6% have a high stress level. Parents who developed resiliency were able to cope up with the situation. It's important to further explore this area to determine additional coping strategies that could assist parents in overcoming stress when raising a child with ASD or ADHD. What would I recommend is to increase public awareness about parenting stress. Parents of ASD and ADHD should receive help through family center supportive services that offer counsel to decrease stress by using appropriate coping strategies and other resources. Mindfulness-based interventions may help reduce parental stress in parents who have children with ASD or ADHD. Thank you. Hello.
Thank you, ma'am. Over to you, judges.
Yes, ma'am. We have studied only one school. We explored any other qualitative analysis?
Yes, I was not able to explore other schools because I have limited time.
Okay. Okay. So it's a very small data set. So we need a large data set to come to a proper.
Okay, there are so many research going on. As a counseling psychologist, I thought it will be helpful for me if I do a little bit of research like this, so that it will help for me to counsel so many children with this disability.
Okay. Okay, fine. Thank you.
Thank you. Is there any question?
Dhanilakshmi ma'am, you have any questions?
No, I can go ahead. Actually, one suggestion, Rajeshwari ma'am.
Okay.
Actually, you can compare with some qualitative analysis of biochemical or hormonal assays also. It is good for your studies, actually. It is very brilliant for you.
Yes.
Yeah.
I'd like to study further.
Thank you.
Thank you, ma'am. Our next presenter is Kushagra Agarwal. So you can start presentation.
Yes, just a second. I don't have access to sharing. Can you? Is my screen visible?
Yes, sir.
Your screen is visible, sir.
Okay. Thank you. So, a very good afternoon. My name is Kushagra Agarwal, and the paper I'm presenting is called Comparative Analysis of SARS-CoV-2 Variants Across 3 Waves in India. This work was done at the Center for Computation, Natural Sciences and Bioinformatics in IIIT Hyderabad under my professor, Neeta Parekh. So, as we know, nearly 437 million COVID-19 cases had been reported worldwide till early this year. More than 43 million of these were from India alone, making it the second worst hit country after USA. The objective of this study is to understand at the genetic level the role of acquired mutations in the circulating SARS-CoV-2 virus and their possible impact on the spread and virulence. So we collected data of Indian SARS-CoV-2 isolates over the 2 years of the pandemic at 4 strategic time points. First, dataset 1 included 685 samples collected in the early phase of COVID-19 from 27th January 2020 to 27th May 2020. Next, Dataset 2 included samples after the first wave of pandemic until 11th January 2021, containing 4,708 samples. Dataset 3 with 45,171 samples was still after the second wave, and finally, dataset 4 containing 1,01,527 samples was still after the third wave in India. Among the various analyses, we first performed a phylogenetic analysis to identify mutations and clusters. Next, we performed principal component analysis to cluster sequences leading to the identification of state-specific subclades. Finally, we performed a pangolin lineage analysis followed by understanding the mutational impact on protein function using SIFT and Provine. For the phylogenetic analysis, we used the bioinformatic engine Augur and the visualization tool or SPICE from Nexttrain. To do this, we created a snake-make workflow using Nexttrain, which included a multiple sequence alignment using MAFT, maximum likelihood tree estimation using IQtree, and time resolution using TimeTree. Finally, clades, mutations, and ancestral traits were inferred, and a NAVIC format tree was generated for visualization. In the figure on the right, you can see the time-resolved radial phylogenetic tree of Indian SARS-CoV-2 isolates. So we identified a total of 1,279 variations in dataset 1 and 7,126 variations in dataset 2 when compared with the Wuhan 1 isolate in China as a reference. Amongst them, the most frequent was the clade 20A mutation A23403G, which is also called the D614G mutation, which is present in the receptor binding domain of the spike protein. In the figures on the right, you can see the D614G mutation on the phylogenetic tree and its prevalence across the different states in India. Some of the other frequently prevalent mutations in dataset 1 and 2 are shown in the plots above. Our analysis revealed that few states exhibited high severity of COVID-19 cases. For example, Gujarat with 5.12% and Maharashtra with 4.19% reported higher death rates compared to the country average of just 2.67%. To understand the significantly large difference in mortality rates in the first year of the pandemic at the genetic level, a detailed analysis of the mutational profile of sequences was carried out, and we hypothesized that the presence of region-specific subclades might be one of the causes. We performed a principal component analysis on the mutational profile of genome sequences, resulting in a separate pink cluster corresponding to Gujarat-specific subclade IgG20A and an orange cluster specific to Maharashtra. Many other state-specific clusters were also observed. As mentioned previously, Gujarat had a significantly large difference in the percentage of deaths, and to understand this difference at the genetic level, we carried out a detailed analysis of the mutations in the sequences from Gujarat and compared it with those from the rest of the country. This revealed a novel subclade, IgG20A, specific to isolates from Gujarat. The subclade was observed in more than 50% samples from Gujarat, but was present in less than 5% samples from the rest of the country. We also found novel subclades corresponding to Maharashtra, Telangana, and Andhra Pradesh. You can find the details of all of these in our paper. Lineages that pose an increased risk to global public health have been termed variants of interest or variants of concern by the World Health Organization. We wanted to analyze the frequency of these lineages in the Indian context. The figures show the prevalence of different lineages across India, Europe, and USA. When Europe was experiencing huge numbers of COVID-19 during the summer of 2020, as you can see from the plot, India luckily missed the first wave. Timely screening of international travelers, quarantine, and contact tracing can be attributed for this. Since the second wave was very severe with very high infectivity and fatality, analysis of new emerging variants between the first and second waves is important in the Indian context. The most important was the Delta variant, which was first detected in India in late 2020 and swept rapidly through the country, accounting for 99% of the cases. It is thought to be twice as contagious, with viral loads up to 1,000 times more. Another prevalent variant during the second wave in India was the Kappa variant, which is also called the double mutant. This is a variant of interest by WHO and accounted for 35% of all sequenced cases in India. With the end of the second wave in India, the number of deaths reduced drastically. This can be accredited to the large-scale vaccination campaign that took place post the second wave and prepared India for a third wave. The third wave was characterized by mild infections, few hospitalizations, and mostly home treatments. On comparison of isolates in datasets 3 and 4, we found differences between the circulating variants during the second and third waves. We found a new variant, BA2, which is a subset of the Omicron variant, which is a variant of concern, and was found to grow in the country with up to 11% prevalence. The Omicron has increased transmissibility and reduced severity. Finally, in this paper, from the genetic analysis of mutation spectra of Indian isolates, we discussed the insights gained in its transmission, geographical distribution, containment, and impact. Thank you.
Thank you, sir. Judges?
Yeah, very good. Can you conclude which one it is very higher version of that is higher mutatant variant? Any genomic sequence study you did?
Yes, so to analyze the mutations, we did the study in two parts. First is we looked at individual mutations in the genomes, and the first part of my presentation was focused on that. So using individual mutations, we are finding novel subclades in the Indian context.
I think you.
Next. Hello. Am I audible? Hello.
Yes, sir, you're audible.
Okay, so next we did this on a genome scale. So there are multiple mutations that co-occur together, which are called lineages. So then we looked at the whole mutation spectrum using this simultaneous mutations that occur together, which we are calling lineages, and that is the pangolin lineages which I introduced. So we looked at it from both aspects of the genomic sequence.
Okay, fine. What's your reference sequence?
The reference sequence was the Wuhan sequence, the first sequence that was sequenced in Wuhan.
Oh.
That was considered as a reference for our phylogenetic tree and for further mutational analysis.
Okay, good. Thank you.
Thank you.
It's a good work. It's a good work. So how did you reduce the number of variables in the PCA for the data set 3N?
How did you reduce the? Can you please repeat?
Variable factors in the PCA analysis.
For the PCA analysis. Okay.
Yes.
So
I talked about. Yes, PCA. Yeah.
Yes. So for the PCA, what we did is we considered every sequence, like every sequence from the Indian isolate, as a vector of, let's say we had 7,000 mutations in dataset 2. So every sequence was a 7,000 vector where each position referred if the mutation that we discovered was present in that sequence or not. And that 7,000 vector was used for the PCA analysis. We did not do it on the exact entire genome, which is of length 30,000. We did it instead on the 7,000 for better representation in the latent space.
Okay, fine. Did you find any marked differences between the versions in the Maharashtra and the Gujarat you talked about?
Yes. So in Gujarat, we found a subclade ICG20A, which had specifically 4 mutations apart from what are present in clade 20A. Whereas in Maharashtra, even though it is close by, we found a completely different subclade, which we called IMH2 in our paper. And it has 2 mutations, namely A313T and C5700A, which are not present in any other mutation in the entire world, not just in India. And these 2 novel subclades were completely apart on the phylogenetic tree as well.
Are you going to further work on this and determine the factors, or you are just going to drop the
Yes.
What is it?
Yes. So now we are looking to use graph theoretical approaches on the protein structure to find the impact of the mutations. So we'll look at the original protein structure. We'll look at centrality measures on the original protein structure, and then we'll introduce this mutation and try to find how these centrality measures have changed. This will enable us to understand the impact of the protein mutation on the protein function as well.
Are you going to trace down the migration history to do that, or how is it? How the
Can you
people
please repeat?
The migration history of the population, or just the current population? We'll go back and verify the data.
We'll not take a new data set. We'll work on the data set that we had, and we'll work on it on the individual protein scale, not for the entire population.
Okay, good work. Best wishes. Thank you.
Thank you. Thank you.
Hello, I have a question.
Yes.
Hello. Yeah, I wanted
Yes, sir.
to know the source of your data set, which I could not.
Yes.
I don't know if I have missed it.
Yes. So the data set was from GISID, which is the Global Initiative for Influenza Data Sharing. So we downloaded all our data from that portal.
Did you consider that migrant? Because I don't think in the database the migrant population was there. Was it included?
Which population? Can you please repeat?
Migrant population.
Yeah, the migrant population was not there, which is why what we did is that's why we had those 4 strategic time points. So whenever we talked about state specific subplates, we only looked at the part till the lockdown was enforced in the country. We specifically did not extrapolate that results into the 3rd and 4th data sets, because that would be incorrect because of the whole migrant population movement that took place. So whenever we talked about state specific subplates, it's what it was for data set 1 and 2.
Okay. Thank you so much. Nice study.
Thank you.
Thank you, sir. Do we have any further questions? Shall we proceed?
Yeah, please go ahead.
Okay, ma'am. Thank you. Our next oral presenter is Agna MP.
Am I audible?
Yes, ma'am. You're audible and your screen is shared.
Okay, thank you.
Can you please put it onto slideshow mode?
Yeah, it's in slideshow mode.
Your
Is it
time
okay now?
starts now.
Yeah, thank you. Good afternoon, one and all present over here. So my name is Agna Impreet. I'm from Christo Jayanti College, Bangalore. And my topic is perception of learned helplessness in relationships among female emerging adults. So I have done this under Assistant Professor Shamli Chatterjee from the Department of Psychology from Christo Jayanti College. So learned helplessness. It is a state that occurs after a person has experienced a stressful situation repeatedly. They come to believe that they are unable to control or change the situation, so they do not try even when opportunities for changes become available. Adult relationships. So in this research, it is mainly focused on romantic relationships. So here, adult relationship is a close connection between two people, especially the one involving romantic and sexual feelings. And emerging adults is the term used to describe a period of developmental span from about 18 to 29. So there are mainly 2 concepts in this research.
Ma'am, sorry to interrupt.
Yeah.
Can you please put it onto slideshow mode?
Presentation. Can you please make it to presentation mode in the PPT? Presentation mode. So that it is visible on the full screen. Yeah.
Sir, I don't know where exactly that is.
At the bottom, can you see a symbol? It will be there.
You can
Yes,
press
sir.
on F5.
F5 also you can press.
F5 you can press.
The third icon, bottom.
Is it okay now?
Ma'am, near the volume, there is a third icon. Please click on it. There's 48%, right? Before that, there's one icon. Please click on it.
This one?
The
Ma'am,
next
it's
one.
on the bottom of
Yeah.
right side, ma'am. Yeah,
Yeah,
that
that
is
one.
yes, ma'am.
Now? Okay,
Yes.
thank you so much. Thank you so much.
Perfect. Yeah.
These are the main 2 concepts of my research: female emerging adults and the perception of learned helplessness. So as I said, female emerging adults means the people who are between the age of 18 to 29, and the perception of learned helplessness, as I said before, when the individual continually faces a negative situation, they stop trying to change the circumstances, even when they have the ability to do so. So nowadays, females are going through many stressful situations and are tolerating certain abuses. Thus, it is important to have a better insight about learned helplessness in relationship, especially for women. So this study can be used by psychiatrists and social workers, and also psychologists who are dealing with relationship problems of emerging female adults. This will also help to have a newer insight into the concept of learned helplessness in relationship. I have referred many studies for this research, but among them, these were the most important studies. The very first one was about Martin Seligman's learned helplessness study. So Martin Seligman was the person who introduced the term learned helplessness in 1967. So he had done certain experiments in dogs, and this paper was about the experiment in dogs, and he found out that the conclusion of that paper was that the emotional stress experienced by the dogs are the same as depression in man. And the limitation or the research gap of the study is that the study was only about learned helplessness. It does not have any explanation about the effects of learned helplessness in human beings. The next study was about the romantic love, commitment, and marriage in the emerging adulthood in Indian context, and this study was by Jigisha Gala and Shagufa Kapadia. And in the findings revealed that even when the emerging adults are open to experimentation before marriage, the ultimate goal of romantic relationship is to establish a long-term relationship culminating into marriage. And the study focused only on relationships of emerging adulthood, but not on learned helplessness. So this is my research question: how female emerging adults perceive learned helplessness in relationship. And the objective is to explore learned helplessness in relationships of female emerging adults. And I used a pure qualitative research study, and the participants were female emerging adults, and the data was collected through purposive sampling and snowball sampling. And the participants were 10 female emerging adults from Bengaluru urban, and the data was collected through socio-demographic form and telephonic interview. And the data analysis were done through thematic analysis. These are the final themes I derived from my research. The very first one is societal and parental controls can contribute learned helplessness. That includes the subthemes of parents to take decisions in relationships and parents' attitude towards the relationships and the negative influence and attitude of the society as well. The next one is battered women and learned helplessness. Here, parents and societal training to women to be submissive and men to be dominant, and societal concepts about women, how a woman should be. And then physical and cognitive changes. So when a woman is in learned helplessness, it is easy to manipulate. So there are participants in my research who reported that they are emotionally compelled to send nudes and restriction to wear certain dresses and also eat certain kind of food as well.
Just to interrupt you again, your presentation is not yet in the slide share mode. Please click that one more time.
Is it okay now, sir?
It is not in the slide share. I don't see it in the slide share mode.
So, if can you see the slide? Is it okay now, sir?
The slide is visible, but it is.
Yes, it is now.
I think you have to physically you can move the slides then, because we don't see it. You can move the slides physically for the others.
Is it okay now? Can you use?
No. And you can stop your sharing option. Then again, you can start freshly.
Sorry.
Ma'am, can you stop sharing and share it again?
Okay.
Yes, ma'am. Thank you.
It's perfect
Is
now.
it okay
You can
now?
continue.
Fine.
Thank you so much. So for a person who is in learned helplessness, they can also face another mental health issues along with learned helplessness. So that include depression, anxiety, and helplessness. And role of social media also has a very important role in helplessness. So portraying women in a negative way after breakup, and also the negative portrayal of women through Facebook, Instagram, and other social media platforms, etc. So although these are some of the limitations of my study, although the researcher has aimed to collect the data only from females, the data will give only the details of females, and only learned helplessness of females between the age of 18 to 25 is provided, and a semi-structured interview was restricted to English-speaking people only, and it lacked a quantitative investigation on the topic, and the sample size was restricted to 10. There are many studies done on perception of learned helplessness in relationships, but no particular study has been done specifically on female. Therefore, this study adds something new on to the existing literature. That's the significance of my study. And these are some of the very few references I used in my research. Thank you so much.
Thank you, ma'am. Over to judges.
What was the qualitative questionnaire contain? What are the things you looked at? Specific aspects, parameters?
So, sir, it contained questions about the relationship status, and also what are the problems, especially after breakup, what are the problems they have faced in the relationship, and likewise, sir.
But your data set is very, very small. So how to come to a conclusion? So it's only 10, right?
Yes, sir, I am focusing to increase my population, sir. Like the sample, sir. This is a minor research conducted, so I'm working upon it, sir.
For any quantitative study, we cannot come to a proper conclusion, right? So what are you planning to do in terms of quantitative studies?
Sir, I will read about it, sir. I'm not able to answer that.
It's nothing great. You have to just quantify whatever contents you get to the questionnaire. That's all. Yeah? It's a simple thing. Yeah. Anyway, good.
Yeah.
Yeah.
Thank you, sir. Thank you so much. Any other questions?
Dhanlakshmi ma'am, do we have any questions?
No, ma'am. We can go with the another question.
Okay, ma'am. Thank you, Agna.
Yeah.
Thank you.
The next presenter is Ms. Pooja Renuka Das.
Hello. Am I audible?
Yes. Yes.
Yeah. Okay. Let me share my screen. Is it visible?
It is visible.
Okay. Is it in the slide share mode?
1 second.
Yes, ma'am.
Okay. So I have.
Your time starts now, ma'am.
Yeah. So the topic of my paper is, I'm a graphologist. I'm Pooja Renuka Das. I'm a graphologist, handwriting analyst for the past 6 years. And I also come with a richer experience of IT industry, wherein I have worked with on different types of projects, both national, international, and a variety of people. And considering that as a profession and the topic that I actually I'm focusing for the past 6 years, which is handwriting analysis or graphology, I have this is what I have come up with in my paper. So it is graphology, an inclination to profession. So I intend to cover these things. A little bit about graphology, how graphology is interlinked with personality, a little bit about handwriting traits, and why is profession important, career for students. So basically career and profession for different age groups, and then how graphology can be used as a selection tool. So what is graphology? Graphology is actually a study of handwriting to reveal character and personality of an individual. It basically comes from the Greek word. If we break it down, it is grapho. Grapho means writing, and logi means study. So when we are actually writing, the hand is holding the pen, but the brain is directing the movement of the hand, and it results into a series of actions which include a number of things like the speed of writing, the pressure of writing, direction of writing, rhythm in which the content is written. And to achieve all this, the brain, muscles, nerves, they all coordinate with each other. So this is on the physical side, but they move in response to the conscious, the subconscious, and the emotional stimuli. And that's how the writing appears on paper. And that's the exact reason why each of us develops our own unique individual script, even though as children, we're taught to form letters in a prescribed format. So how is graphology linked with personality? Graphology can actually identify about 300 aspects of personality, and it can do so by examining the zones, the loops, the letter spacings or the word spacing, line spacing, slants, height of the letters or the size of the letter, whether it has ending strokes, starting strokes, etc. So every symbol in a given handwriting sample is actually interpreted as a personality trait, and it can reveal a number of things. So on a physical and material drive, we can say it actually tells us about the individual's energy level and vitality levels. For emotional characteristics, it can portray the feelings or the moodiness or how the emotions trigger dangerous reactions. For an intellect or a thinking style, we can say that we can come to know the methods employed in problem solving, or whether the writer is paying attention to details or no. With respect to personality traits, we can say we can find out about the confidence or lack of it, or self-esteem, adaptability that the person has in different situations. Social behavior, it can be like ability to handle clients, associates, people around us, and sensitivity to their emotions and to your own emotions. And vocational implications, like ability to tackle a project or a work project, or his job efficiency, his performance, his organization skills, etc. And how do we do that? So there are certain handwriting traits. There are many traits. I'm concising it to very few over here. And all these actually work in conjunction with each other. They have to be combined. Different permutations and combinations have to be applied. And based on that, the personality of the person can be derived. Some of the traits are like a slant, connections, thinking patterns. Slant actually depicts about the emotional makeup of an individual. If a person is having right slant, it means he's warm-hearted, he's adventurous, he's social, he's courageous and spontaneous too. A left slant indicates someone who is loyal, but he's secretive also, and he's reflective. And most of his decisions are based on his past experiences. Whereas an upright slant indicates that the person is more practical. He is in the present, he's level-headed, and he is more inclined towards common sense and a logical approach. Now, coming to connections, there are again many different types of connections. So by connections, I mean how the letters are connected to each other. If they are connected at the baseline, then it is called as a garland connection. And if it is connected from top, it is called as arcade. There are other types of connections also, which is angles or angular, and wavy or thread-like connection, which again has its own implications and fallouts on the personality. We can also come to know about the thinking pattern of a person, whether he's a methodical or a comprehensive, investigative, analytical. Now, each of these thinking patterns have their own meanings. For example, methodical person, he would prefer to collect all the facts before making any decision. It is a slow thinking pattern, but he's more creative, and he gives time to whatever thoughts that or whatever comes in front of him, and based on that he makes logical decisions. So he collects all facts and then makes up a decision, which ends up in having a decisive kind of an approach. Whereas a comprehensive thinker is like a lightning fast mind. He's very curious and intelligent, but that means he has impatient mindset also. They're actually very active, but then he ends up thinking too fast, and at times he makes impulsive commitments also. An investigative thinker is a person who learns very quickly. He is into more of questioning mode. And analytical is a person having a strong reasoning. There are some more traits: size, spacing, speed, and the list actually goes on. I would prefer to move on to the other part of the paper, which is, why is a profession important? A profession actually forms a central part of a person's adaptation to life. So a career makes up a really big part of an adult. We spend a significant amount of time on at work, and so it's very important for us to make sure that that time isn't killing our soul. It provides us, a profession actually provides us with a sense of direction and purpose in life. It also gives us long-term financial security and a sense of achievement. Hence, it becomes very important aspect of life. How about career selection for students, profession, when they end up selecting a particular profession? Yes, so that is also important. It actually is very important for students, because that's the beginning of their career or their professional life. So deciding on a career path is actually a very complex process. And to come up with the choice, they have to actually introspect themselves on many areas, like what are their current values, interests, skills, preferences. And the ability and the potential also of the student should also not be ignored. They actually face quite a lot of challenges, in terms of like finding a perfect fit based on their ability and potential, understanding their own aspirations. They also fear failure. They quite a lot of time they have lack of course and career awareness, and peer pressure also. So it becomes self-assessment becomes very important for students, and they can assess themselves on these parameters.
Hello, ma'am.
Yeah.
Can you please wind it up in a minute? Your
Sure.
time is up.
Sure,
Yeah.
sure. So self-assessment based on their strengths and weaknesses and what their dreams and goals are becomes important for them. And personality at workplace, of course, I spoke a few slides ago about that also. And organizations, they also need to find the right candidate in their organization, so that they can save time and resources and money, of course, and achieve business growth through good customer service. So graphology, how does it fit into all this? It can form a very good tool as a selection tool. It's an objective way of analyzing a person. So it is not a self-report, or it does not even have invasive questions as we find it out in the psychometric test. So it actually, the traits can be selected unique to a job position. So while analyzing a handwriting, we can use graphology to identify the right traits for that particular position that the person has applied, or for that for which the organization is looking at. And usually successful employees, they typically have personality commonalities. So we can identify those common traits also, which eventually can lead to their success level. So handwriting analysis also evaluates the total personality and not a part of it. So it gives a very introspective kind of and a wholesome kind of a report. It is also very, not as expensive as the other tools that are currently available, and it's cost effective, time effective, staff. So little of staff time is required, and it also gives the interviewer an immediate insight into the personality. So basically, there are on so many aspects. Graphology can act as a selection tool. You typically, usually for different positions, organizations hire graphologists, like if they want an accountant. So based on the characteristics, the personality characteristics, the handwriting analyst can find out whether he's a right fit for a job. So with that, we can conclude that understanding and analyzing self is an important aspect of career, and graphology can be a very powerful method to identify and understand, even for a student or for a working professional, his own personality traits. It can also help organizations to know the other person's or the candidate's strengths and weaknesses. And thus it can be used in transforming and enhancing a personality or an individual's personality to be successful. So that's all that I have. Thank you.
Yeah, Pooja ma'am, I have a question.
Yeah.
Actually, it is an interesting topic, but based upon what parameter you are finding out about that patient's or the person characteristic feature? Any tools or any qualitative analysis you are using for that?
Tools. So basically this is, I mean, we do it manually. Handwriting analysis is done manually. And for career selections, there are a number of tools like the MBTI, the Big Five model. So big organizations usually hire it, hire other companies to do this psychometric testing for them.
Yeah, that's why I'm asking.
Mm-hmm.
Because only for that purpose of handwriting, we are observing from 10 people or 20 people, we can't conclude, right?
Yes, but then that has been matched with the results that were achieved through these tools also. So these tools basically, now if you consider MBTI, it has it mostly considers only the first 16 personality types, and based on that it suggests. But then it will not give the temperament of the person. So it only gives, MBTI will give you only the right fit, whether the person is has attention to details, good concentration level on very few aspects, whereas handwriting analysis can also talk about the temperaments, whether the person lives more into in his past, or is he more practical oriented, or is he opportunist. So all these, all the positive, so it's a combination of many traits that come into handwriting analysis, whereas the psychometric, they focus on very few things.
Okay. Okay, thank you.
I have one.
Sure.
It's a very interesting topic, ma'am. But there is a group in Toronto University, in the Department of Medical Physics. They have published a paper which correlates between magnetic fMRI studies with handwriting. So I think if you can take towards that, that helps in designing new kinds of tablets, because most of them are moving towards.
Yeah. So yeah, we're looking forward
using tablets, typing. So if you can turn your studies more towards fMRI, what is the relationship between the different cognitive patterns within the brain, and how the complex function of writing comes up? I think you can go through a couple of papers that will be very, very useful. It's a big group. They are working with many tablet producing industries. You can just Google search. It is a very easy paper, well-known paper published in 2018, which is the first kind of analysis on fMRI analysis on handwriting. Just Google it up, you will get it.
Sure.
Thank you.
Sure. Thank you so much for this insight.
Thank you, ma'am.
Yeah, thank you so much.
We shall move on to the next presenter, that is SS Power.
Can I start?
Yes, you can start.
Okay. Good afternoon, all the dignitaries and participants. I'm Shweta Sanjay Pawar from Dr. P.D. Panjabrao Deshmukh Krishi Vidyapit, Akola. I'm second year student. Today I'm here to present my research topic, that is targeting GM-BADS to gene-specific mutation and designing of allele-specific marker for aroma. In the post-COVID era, we got the importance of protein-rich diet. And soybean is the rich source of protein, which is almost 40%. But we can't eat a soybean directly as a food due to its binny flavor and some anti-nutritional factors. Actually, the soybean is categorized into vegetable and grain type. Vegetable type soybean possess some characteristics such as bold seeds, sweet in taste, and and having some distinct aroma, which fetch higher price and demand than those of normal normal soybean genotypes. For actually for aroma in actually the aroma in vegetable soybean is due to the volatile compound, that is 2-acetyl-1-pyrrolene, which is controlled by the single recessive gene, that is BADH2. As we know that aroma are the key drivers of our flavor experience. As much as 80% of what we call taste is actually the aroma. Here we can see that it is reported that for aroma, BADH2 gene is responsible. Here we can say that when BADH2 is functional, there is GABAL is converted to GABA, that is gamma aminobutyric acid, and there is no accumulation of volatile compound, that is 2-AP. But when it becomes non-functional due to some mutation in that BADH2 gene, there is conversion of GABAL into delta pyrroline, and there is accumulation of 2-AP in aromatic soybean. Here, in this figure, the structure of GM-BADH2 gene is given here, showing the 15 exons and the 14 intronic regions. And here we can see that the SNP changes for the development of aroma in different crops is given here. Here we can see that SNP, that is the T-whisper gene in exon 10 of soybean, is responsible for aroma in soybean. Now we can see the importance and need of study. As I explained earlier, vegetable soybean is highly nutritious and possesses distinct characteristics, which sweet taste, extra large pod seeds, and distinct seeds fragrance. Until now, there is no report of any Indian soybean aromatic genotype. And if this genotype will be developed, it will be very helpful for the farmers, as they can fetch the more earnings. Also, it will be very helpful for the development. It will be very helpful for the livelihood improvement in order to get the enhanced nutrition such as vitamins and proteins. Also, to identify the allele-specific mutation in BADH2 gene sequence, designing of gene and exon-specific primer is the first emphasis for the marker development for further marker-assisted selection study. Here, investigation aims to find molecular relation and to develop the marker that can be exploited for the further breeding program for fragrance. By considering the importance and need of this study, these are the objectives. In that, first is the primer designing for BADH2 gene targeting CDS and non-exonic regions, and second is the validation of allele-specific primers in aromatic genotypes. For that, I have collected this the material which is required for my research work from AVRDC Hyderabad and Dr. PDKV Ekola and ISR Indore. In these, I have collected some vegetable type genotypes and some grain type genotypes. This is the method. In first activity, primer designing for BADH2 gene targeting CDS and non-exonic regions is carried out. In second activity, the PCR profiling of genotypes using design primers is carried out. And in the third activity, identification of primers are identified which discriminates between the aromatic and non-aromatic soybean genotypes. The primers specific to the whole BADH2 gene, by considering the all exonic and intronic regions, have been designed with the help of Primer 3+ software. The primers are designed by using this criteria. After we will see the results. We have screened all the selected aromatic and non-aromatic soybean genotypes with the help of designed allele specific or we can say the exon and intron specific primer. In that, the GM-BADH2 G2 primer showed the polymorphism between aromatic and non-aromatic soybean genotypes. It shows the single polymorphic amplicon, which is of 200 base pair in aromatic genotype AGS464. After that, we did the sequencing of that aromatic genotype, that amplicon in AGS464, and also we have did the sequencing of two non-aromatic genotypes, that is Swarnasoya and NRC147. After sequencing, we have did the sequence alignment, BADH2 gene sequence alignment with the grain type genotype, that is AMS MB518. In that, we can see here that here we can find the changes in triplet codon, that is AAA here we can see the changes in triplet codon AA, which leads to formation of amino acid lysine. Also, in gene sequence alignment of aromatic genotype AGS464, we have found the SNP, and we have found this changes in gene sequence from nucleotide sequence from AA to CAA, which leads to amino acid change from lysine to glutamine. After we have did the protein-protein interaction with the help of string software, the string protein interaction studies found in the string protein interaction studies found that the target protein interacts with the 4 regulatory proteins. After that, the P5CS, that is proline 5 synthetase, plays a key role in the proline biosynthesis, and that leads to the osmoregulation in plants. After, to find the exact location of that mutation in the sequence data, we have found that exact location of mutation in the sequence data with the help of Munster software. Here we can see that in table that this table shows the credentials of the exon edit as per the requirement of STR draw. And the script to get the result as a structure image in Munster software with the finding mutational regions in exon in the 10th position of BADH2 gene. Here we can see at the exon 10 we found different mutation in the AGS464, that is aromatic genotype. We found the mutation at the 61 base pair and also at the 125 base pair. And also in non-aromatic genotype, we found the mutation at 213 base pair, 246 base pair, and also at 263 base pair. After, by considering this mutation which we found in our sequencing data, we have designed the primer which are specific to that SNP. After we have designed these primer pairs, the analysis of PCR amplification with 10th exon specific markers showed the polymorphism between the aromatic and non-aromatic soybean genotypes. Here we can see that PCR amplification profile of that 663 plus 884A primer shows the polymorphism between the aromatic and non-aromatic soybean genotypes. After we have also done the phenotyping of that selected aromatic and non-aromatic genotypes to check the presence of two volatile compound, that is 2AP. And in that we have did the GC analysis. In that we found these are the chromatograms of that gas chromatography analysis. In that, we found total 91 compounds along with 2AP. These are the
hello, ma'am.
common compounds. Hello.
Can you wind it up in a minute?
Okay, ma'am. These are some common compounds which I have been listed here. The 2-acetyl-1-pyrroline, which is a volatile compound, was found in aromatic genotype. But it is did not detected in non-aromatic genotype, that is Swarna-Vasundhara and AMS-10039. It is found to be highest in aromatic genotype AGS465. From this study, we conclude that the SNAP-based marker showed the polymorphism between the aromatic genotype AGS464 and non-aromatic soybean genotypes, that is MS-10039 and 89, 127 and 147. This study revealed that the mutation in the exonic region of exon 10 of BADH2 gene. The mutation AAA nucleotide base pair changes found in non-fragrant genotype, that is Swarna soya, while CAA base pair changes was found in AGS464, which lead to amino acid changes from lysine to glutamic acid, and the confirmation of mutation being carried out using the gene and exon specific primers. The implications of this study are the designed allele specific primer, that is 663 plus 884A, can be further exploited in marker assisted selection for the introgression of fragrant trait for the introduction of fragrant trait in a grain type soybean genotypes. And similar to exon 10, the finding in SNP is possible in different exons that will be responsible for aroma in soybean. Thank you.
Thank you, ma'am. Over to you, judges.
A very good presentation, Pooja. Can you tell about this, how to find exons? Hello.
Ma'am, can you please repeat the question? Ma'am, your voice is not clear. Can you please repeat the question?
Yeah, how to find out the exons?
What, ma'am? How to find out?
Exons.
Exons. Ma'am, we can find, first we have to retrieve the gene sequence from NCBI. And after that, by using the bioedit tool, we can find the exons and intronic regions.
Okay. Only that only you are utilizing for identification?
What, ma'am? Ma'am, can you please repeat the question? I'm not getting your question because your voice is not clear. Hello?
Clear.
Ma'am, can you please repeat the question? Because of some network problem, I'm not getting your question.
Are you using any other identification technique?
Yeah, you're using. Yeah, yeah.
Hello.
Hello. Yes, sir. Please.
Yes, I'm audible, right?
Yes, sir.
Yeah, this is Sailesh here. So my question is, so I'm audible, right?
Yes, sir.
Okay. So my question is, so you have selected the GCMS, right?
Yes, sir. GC. I only selected GC.
Yes. So my question towards you is, so is there any other identification method instead of GC? Because you have identified certain compounds, right?
Yes, sir.
Like, is there any other tool you are going to reach out as?
Sir, there is another tool we can say that say it as a electronic tongue or electronic nose. With we can find by which we can find the amount of 2AP present in that genotypes, or which we can use for detection of volatile compounds. That electronic nose
You
or
are particularly looking to the volatile compounds. You are not going for any peptides, those molecules, right?
Yes, sir.
Okay, fine.
Thank you, sir.
It's done. Next person, yeah.
Okay, sir. Thank you. The next presenter is Sushma N.
Hello.
Yes, ma'am. You're audible.
Okay. Can I share my screen now?
Yes, please.
Is my screen visible?
Your screen is visible, but please share the presentation, your PPT.
I have shared it already. Is it not visible?
No, ma'am, it's not.
Ma'am, I think you can unshare it and share it again.
Okay. Now is it visible?
Can you please put it to slideshow mode? Yes, ma'am.
Yeah.
It's visible. Your time starts now.
Okay. Okay. Good afternoon, everyone. I'm Sushma, and my presentation is about study of yoga in Indian psychology. Indian psychology encompasses the profound intellect, knowledge, thought, spiritual, psychological understanding, and underlying philosophy rooted for centuries in the Indian subcontinent. Centrality of consciousness is the core concept of Indian psychology, and it emphasizes the integral connection between mind, body, and consciousness. It states that mind, when it is attached to the sensory organs, we experience phenomenal awareness, and when it is attached to the consciousness, we experience transcendental realization. Mr. Rao K.R. states that Indian psychology refers to a system of school or school of psychology derived from classical Indian thought and rooted in the psychologically relevant practices such as yoga prevalent in the Indian subcontinent for centuries. Indian psychology is not just a theoretical assumption, but it suggests some practical ways to remove the blocks that blocks the person from experiencing the true happiness. Many paths have been suggested, like path of gnana, that is knowledge, path of bhakti, that is celestial love, path of karma, that is altruistic action, and also varied meditational practices are suggested to serve this purpose. Yoga is one such ancient psychologically relevant practice as suggested by Mr. K.R. Rao, and yoga is native Indian in its origin, and explicit reference of yoga dates back to pre-Vedic period, that is about 26,000 years ago. For the purpose of study, history of yoga has been classified into four important periods. That is Vedic yoga, where rituals and sacrifices were used as means of connecting with the higher spirit, and pre-classical yoga, when inner vision, knowledge, wisdom were documented in texts like Upanishads and Bhagavad Gita. Classical period is when Patanjali Yoga Sutra was introduced. Post-classical yoga period is when Vedanta, Hatha Yoga school, Tantra Yoga schools all were introduced. Currently, we live in modern yoga period where blending of traditional asanas and with modern exercise is happening. We see a lot of schools around us like Patanjali School of Yoga, Hatha Yoga, Kriya Yoga schools, and so on, where such experimentation is going on. While modern yoga focuses mainly on the physical aspect and limits it to the physical activity, traditional yoga is holistic in its approach. It takes into account a person's physical, mental, spiritual well-being, and also it is linked with metaphysical and parapsychological phenomena. Yoga is clearly understood as one of the six orthodox philosophical schools and practices that aims at personal transformation. Nyaya philosophy, Sankhya, Yoga, Vaisheshika, Purva Mimamsa, and Uttara Mimamsa are these six orthodox schools. In that, yoga is closely linked with Sankhya philosophy. Where Sankhya is the theoretical aspect of yoga, it is like a roadmap, and it states that through attention and meditation, a person can reach self-actualization. Yoga brings this philosophy of Sankhya into the realm of experience or practice. In traditional yoga, specific and systematized approach to both theory and practice has been postulated through sutras and shastras. Patanjali Yoga Sutra is one of the very important contribution to the Indian yoga system. And Patanjali says that some practices systematically designed to free humans from various levels of disturbance like social, environmental, cognitive, and so on. He believes in purity of soul, and he says that separation, that is emancipation of Purusha, that is pure consciousness, from Prakriti, that is material basis of the universe, is the key, and it is done by self-awareness, and it can be practiced through disciplining our body and mind. So to attain self-realization, Patanjali put forth some 195 aphorisms or sutras that makes eightfold path of yoga. It is also called Ashtangas of yoga, and they are Yama, Niyama, Asana, Pranayama, Pratyahara, Dharana, Dhyana, and Samadhi. First five of these are called as external aspect of yoga, and it is about disciplining our body and mind. And last three are called as internal aspect of yoga, and it is about traveling in the path of self-actualization. Yoga and its relationship with mental suffering. The objective of Indian yoga is attainment of moksha or kaivalya, but traveling in this path is not so easy. Psychological sufferings, kleshas and vrittis, all this come in the way. Psychological sufferings or kleshas are hindrances. Vrittis are called mental fluctuations. So kleshas can be something very subtle, or like in the form of everyday challenges and disturbances, or it can be very profound that affects the person greatly, causing him great pain. Traditional yoga identifies some 5 types of kleshas. They are Avidya, that is spiritual ignorance, like inability to judge between right and wrong. Asmita, that is egoism, too much of I, me, and myself, that is a very narrow perspective of the world. Raga, that is attachment or greed. Dvesha, that is aversion from anything that is not pleasurable. Abhinivesha is fear of death, too much of love towards life, and it is like clinging on to life, and then inability to accept death as a natural cycle of life. So, what are chitta vrittis? Chitta vrittis are waves that arise in the mind due to thinking or inputs from sensory organs. These vrittis can either bring pleasure or pain, but whatever it brings, it itself is a roadblock. It blocks us from seeing the true knowledge. So, traditional yoga classifies some 5 types of chitta vrittis. They are pramana, viparyaya, vikalpa, nidra, and smriti. Correct, incorrect knowledge, fantasy, sleep, memory. Whether it is pleasurable or painful, a sadhaka or a practitioner has to overcome this kind of chitta vrittis. Afflicted by vrittis and kleshas, our chitta or our mind manifests some 5 levels of functional or attentional variation. And these ranges from a lot of fluctuation in attention, that is kshipta, to a stage of unwavering attention or nirodha. As practitioner travels ahead, he will be able to overcome these kleshas as well as chitta vrittis, and he reaches a stage called chitta vritti nirodha, or calming of the mind. Once he reaches this stage, he can advance in the path of self-realization. On the whole, unification of the mind, consciousness, and bringing a sense of oneness, harmony, and salvation from human miseries is achieved through the practice of yoga. And also, Indian psychological view not only limits to the conscious efforts of achieving the physical benefits, mental benefits, cognitive benefits, etc., from yoga practice, it also addresses a lot of unconscious factors affecting our conscious self, our conscious mind, and behavior. And also yoga, it does all this by addressing the very root of the human agony. And a lot of negative mental blockages, suffocations, emotional baggages, clingings, and unreal attachments and sorrows, all these are addressed. They are removed and replaced by the light of and self-attainment. And then some implications and applications of yoga practice. Of late, integrating and incorporating all these ancient yoga principles with that of modern yoga practice as well as modern yoga therapies are happening, and that is the focus of research interest. And many areas like medicine, counseling, and therapies, spiritual guidance, they are increasingly exploring the benefits of meditational and yoga practices. Biofeedback, psychophysiological, parapsychological researchers, mysticism, transpersonal psychology, and consciousness studies, etc., are examining the practice and principles of yoga psychology. So, on the whole, an in-depth comprehension of tradition of yoga principle and yoga psychology enables a better understanding of ourselves, to gain a self-knowledge and know our philosophical roots better. Thank you very much for this opportunity. Hello.
Thank you, ma'am.
Thank you.
Over to you, judges.
Are you a student of cognitive psychology or something?
I'm a counseling psychologist, sir.
Okay, fine. So have you heard of you trying to modernize yoga, studying it in modern scientific method? It's a big task.
Sorry?
There is a research funding recently available to do that from Department of Science and Technology, cognitive technologies. But there is something called a new area of domain which you may look into. It is called quantum neurophysics. Okay. So these are the modern tools, because it's a metaphysical subject. It's related to your mindfulness, because we know sodium, potassium, calcium, and chlorine, and the microtubules. These are the things which are present in the neurons, which are actually help in conductivity. You know, it is a positive, it's ionic conductivity, right?
Definitely.
And how to correlate it to metaphysical tasks like thought process? Then you have to combine fMRI studies with quantum models. There are new models. You can refer a couple of. You can just Google quantum neurophysics. You'll get a lot of models. It's a new area. It's only a couple of years old, but that can be one technique to make some. For example, when you're doing a specific posture in yoga, how to correlate it with your thought process, which are the active regions of the brain and which are the non-active regions of the brain, where is the more conductivity possible. So those kind of work can be done, but you need to work with a lot of fMRI studies, and you have to do a lot of modeling in quantum physics, basically applying quantum physics models to neurobiological thought process. So it's neurophysiology combined with quantum physics. You can just Google. I can even put a couple of papers in the group so that you can go through and understand how to analyze these kind of metaphysical concepts. It's not only in yoga. There are many studies across the world, but there are new group of research I'm sharing with everyone. You can go through certain papers there. So that will be interesting because it is from Elsevier. So you can just go through the things, and whatever work you are going to work further on it, just a study paper.
I'm thinking to go further with this.
Yeah, so then you should start working on fMRI studies. So that's the way you can correlate both of them.
Definitely, sir.
Yeah. Okay, that's my observation. There is not much about it. Yeah, thank you.
Thank you very much. Thank you very much for the input. Thank you.
Ma'am, you have any questions? Yes, ma'am.
No, ma'am. You can go with the next presenter.
Okay, ma'am. The next presenter is Shahnawaz Ahmed.
Yes, ma'am. It is visible, ma'am?
Yes, sir.
Thank you. Thank you so much. Hello to everyone. Myself is Shahnawaz Ahmed. I am from IFT University. The title of my work is I'm presenting today is QSR modeling, molecular docking, DFT, scaffold hopping, and it's based on design of the newly designed tetrahydrobital compounds of ABDC2 of breast cancer. Now, coming to the introduction. Cancer is term used for disease in which the normal cells divide without control and are able to invade to other tissues. It can be affected almost any part of the body. Cancer is considered one of the deadliest diseases throughout the world, and most common cancer is the breast cancer in female. According to WHO, breast cancer becomes the most common cancer globally as of the 2020-21. According for 12% of the new annual cancer cases worldwide, ABDC2 is a ATP binding cascade subfamily G. Member 2 is a member of the white subfamily of the transporter, and 48 members are occurred, and seven subfamilies, and most
Excuse me,
occur
sir.
in the
I'm really sorry to interrupt. Can you
Yeah.
please put it in slideshow mode?
yeah exactly.
Can you please put your presentation
Yeah,
in?
yes, yeah.
Yeah.
It's visible?
Yes, sir. Yes. Thank you, sir.
Oh, sorry.
Thank you, sir.
The most occurred in the placenta and breast cancer and testes. The protein is identified as the most important therapeutic targets as it is highly expressed in the cancer, which is presented, affect the efflux mechanisms, metabolics, and signaling pathways. Other thing is we are selected the tetrahydrobic compound, which is widely expressed in many natural products and synthetic compounds, which shows the variety of the pharmacological activities. Tetrahydro-beta carbonyl is a rigid indole ring and flexible piperidine motifs. That means the receptor is easily bind with the flexible piperidine rings, and piperidine rings have many scaffold part, and such rigid structure tetrahydrobeta carbonyl such shows the preferable affinities against the different receptors and could be used for hydrogen bond receptors. And in this study, we utilize the tetrahydrobitanol compounds, form the QSR, molecular docking, scaffold hopping, newly designed 3409 compounds, and selected top rank 4 compounds for DFT calculation. Now, this is the flow chart of my study. I use the flare module of the CRISset software for QSR docking, and for scaffold hopping, we use the Spark module of the CRISset, and we got 3,409 newly designed tetrahydrobeta carbonyl compounds I use glide module for Schrodinger for the STS high throughput screening and quick prop module for the Schrodinger. Used for admit properties and drug likeness properties. And finally, we do the DFT calculation, means density functional theory calculation by the software of SPATN 20. Now, this is the alignment of the our 30 compounds of tetra beta hydrogen compounds, and this ring structure shows the core structure of QSR modeling. Dotted red ring indicates the core part, and this shows the 3D QSR results. We choose randomly 80% training sets and 20% in the test set. The fitness plot plotted between the predicted and actual activity, showing a separate data series for the training in blue color and test for purple color. And R square is 0.99, means correlation coefficient is 0.99, and cross validation correlation coefficient is 0.92. Here in the slides, we are discussing about the different effects. Different effects means molecular representations of the electrostatic variance, static variance, and favorable and unfavorable electrostatic regions of the best biological active molecule. Now here, this is the part of active atlas. Active atlas means the structure activity relationship of a group of chemicals is used for calculations of the accounting for their shapes, electrostatics, steric, and hydrophobic features. Now the favorable hydrophobics are depicted in the green color, you can see here, and the magenta color shows the unfavorable condition, and the red color shows the positive, and cyan color shows, you can say cyan or a blue, both you can say, it's for negative effects. This is the docking part. It is my protein, which is the chain A, and this is the chain B in the binding part. This is the newly designed tetrahydrobeta compounds 1, 2, 3, and this is the 2D graph, 3D pictures you can say. And I'm discussing about here the newly designed compound tetrahydrobeta carbon 1, forming the hydrogen bond interaction with the threonine 402, glutamine is 437, and asparagine is 436. And newly designed compound second means B, in B part, form the hydrogen bond interactions with the residue. Glutamine is 398, glutamic acid 446, and residue threonine 542. This as it is, we can see this is the 2D graph, and this is the 3D of the docking part. Now this is the tetra, this is the metoxytrone. It is our drug control, and this is the earlier synthesized compound number 27. Now come into the my illustration of the different glide score, smile score, smile files, and hydrogen bond interaction. Different hydrogen bond interaction you can see here, and this is the hydrophobic interactive residues. XPG score, IFD. IFD means induced fit docking. First we do the docking by the glide with the help of Schrodinger, and then after I redock by the glide score. We select the 30 compounds for glide score. This is the in this slide I'm discussing about scaffold hopping. Spark module of CRISSET is used for scaffold hopping. We select earlier synthesized compound number 27, which have lowest binding affinity in the data set of tetrahydrobeta-carbonyl. Earlier synthesized compound 27 have different frame modification at the position substituted R1, R2, R3, and R4, and forms the 409 newly designed compounds. And as it is, you can see this is the this slide shows the basically the scaffold hopping. What is the scaffold hopping logic behind? Basically, we select the reference compound on the basis of the lower binding affinity, and different inbuilt databases used in the Campbell, like Campbell, commercial, research reagents, e-vehicles, software, different databases we use, and got about the 3,409 newly designed compound of the tetrabeta hydrobeta carbonyl compounds. We selected on only the topmost 4 compound lead compound, which have here you can see the red color. So I will the positive electrostatic effects in red color, negative effects in
Hello, sir.
scan color. Yeah, ma'am.
Very sorry to interrupt.
Yeah.
You have another 1 minute, sir.
Yeah. This is our control, and this is the compound number 27. And this is the 2D graph of the our newly designed compound is 1, 2, and 3. This is the pink color shows the hydrogen bond interaction, and this is the earlier synthesized compound, and this is our drug control. This table shows the drug likeness properties, all the values of the range of the drug likeness and admit properties like TPSA, topological surface area, not greater than 140, molecular weight is 130 to 725, and octanol partition coefficient bond not greater than 5. All the values are in under the and DFT calculation usually DFT calculation possesses the higher chemical reactivity, while those are large band gaps. Basically here we can see the difference between the HOMO and LOMO energy, and negative mu indicates the nature of the stable compound, while the omega related with the stabilization energy. We noted that except all the compound shows HOMO levels with a slight variation in LOMO energy. This is the compound number 1, newly designed 1, 2, and 3. And this is the basically it is the electrostatic potential map, and this blue color shows the stability and red color shows instability. The QSR study of reported suggested that have R square is equal to 0.9299 and Q square is 0.92. The molecular docking study shows that the newly designed compound tetrahydrobeta carbonyl 1, 2, and 4, and 3 have binding affinities and greater number of the hydrogen bond interaction, and earlier synthesized compound and methoxytrone drug. Further, all shortlisted molecules shows comparative similar HOMO levels with the slight variations in HOMO LUMO levels energy. Significantly, molecules are stable. On the basis of computational studies, we concluded that QSR, molecular docking, scaffold hopping, and DFT calculations newly designed compound 1, 2, 3, and 4. This is the references of our research work. And I acknowledge to ICBDS 2022 and ICPMN and Vasistha Genomics Research Lab, and Dr. Azimul Islam, Dr. Dinesh Gupta, Dr. Tanzeel Ahmad. Thank you.
Thank you, sir.
Yeah.
Judges, over to you.
Yeah, it's a nice work.
Yes.
Can you tell about it from where you are find out the components?
Sorry, ma'am.
Where you are getting the components?
This is I take a paper which have a 30 compound, which is earlier synthesized paper.
Okay.
Yeah, ma'am.
What I'm asking now, like any natural product you are trying to extract the component based on this, if you are utilizing or what I'm asking.
Can you tell me again?
Hello.
Yeah, ma'am. Yeah, I listen.
Yeah. So I'm asking about that, like you are extracting from any components or like from natural sources.
Natural sources, actually, this is the tetrahydrobutyren compounds. This is occurred in natural products like the coca and chocolates and coffee and smoke of cigarettes. Tetrahydrobutyren compounds is occurred in many places.
Yeah, that's why I'm asking.
Yeah.
Okay, why you are choosing the component instead of extracting? I'm asking. Okay. Yeah.
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 Actually, yeah. Thank you so much. And any other question, ma'am?
No questions from my side. Thank you.
Ravinder sir, do we have any questions?
No,
Yes,
no,
ma'am.
no.
Okay,
No.
sir. Thank you. The next presenter is Charai Jolly.
Yes, am I audible?
Yes, ma'am, you're audible.
Thank you. Is my screen visible?
Yes, ma'am. Your time starts now.
Good afternoon, everyone. Good afternoon, respected judges. My name is Cheryl Jolly. I am a PhD candidate at Noida International University, and I have written this paper under the guidance of my co-author, Dr. Ashu Kumari, who is an assistant professor at Noida International University. So my topic for today is understanding the role of social media use on self-esteem among senior secondary school students in Delhi and National Capital Region. So with the advent of social media, it has been commonly observed that the younger crowd has been drawn to it in a vast majority. Accompanying this increased engagement with various social media platforms is an incumbent need to study the varied effects that the use of social media can have on a young mind. So one of the effects that we're currently studying is the effect that social media use can have on one's self-esteem. So these are the objectives of our study. First is to find the relationship between social media use and self-esteem among senior secondary school students of Delhi NCR. Second is to compare the gender differences in social media use. And lastly, to compare the gender differences in self-esteem. So these are the hypotheses that we have designed. First is there is a significant relationship between social media use and self-esteem. Second hypothesis is that there is a significant gender difference in social media use. And the last hypothesis is that females would have lower self-esteem than male school students. And the last hypothesis was designed based on previous literature review. So this is the review of the literature. So as found by Rosenthal and Tobin in 2022, those suffering from depressive symptoms or low self-esteem may benefit from reducing the social media use. Another study by Ma in 2022 established that the associations between online activities and self-esteem were not significant. On the contrary, Zhao et al. In 2022 illustrated that the levels of self-esteem were found to be negatively correlated with social media addiction. In a similar note, True et al. Found that more time one spends on looking at the post and scrolling through more of it, the lesser will be one's self-esteem. Pop, Ayerga, and Irkau in 2022 found that predominantly image sharing platforms like Snapchat strongly positively correlated with self-esteem. In a significant study, Anderson et al. Found that the influence of self-esteem, the use of social media on one's self-esteem is regulated by one's emotions. And some protective factors against getting addicted to social media is self-esteem. Lee and Wei found that the effects of social media use depends on who uses it. Mann and Bloomberg suggested that teenage females tend to concentrate more on interpersonal relationships as they develop their self-esteem. Saini, 2021, found that there is no significant correlation in self-esteem and social media. Sagar, 2021, found that there is a negative correlation. Agarwal found that males showed lower self-esteem, and Bhuvaneshwari found that the sample used social media for 1 or 2 hours. So this now we're coming to the method and the tools used. So this is a non-experimental correlational cohort study. The sample size consisted of 100 school students, which consisted of 50 males and 50 female school students. The data was collected through Google Form. The data that's collected was analyzed through JASP, which is an open access statistical software. The tools used were informed consent and socio-demographic form, Rosenberg self-esteem scale, and social networking time use scale. The statistical tests which were conducted on the data were Pearson's correlation and t-test for independent samples. So these are the results that were obtained. The first table shows the social networking time use scale. This is the correlation value that is obtained, and this is the p-value. The second table shows the independent sample t-test of social networking use, and this is the t-value that is obtained. And the last table shows the independent sample t-test for the self-esteem scale. This is the t-value that is obtained. So now we come to the discussion. So firstly, very interestingly, we found that there is no significant relationship between social media use and self-esteem for the selected sample. And for the second hypothesis, it has been accepted, which indicates that there is a significant gender difference in social media use among the sample. And lastly, we found that females do have a lower self-esteem as compared to males in the selected sample. Lastly, I come to the limitations. So this sample did not include a wider spectrum of gender. We could have included transgender folks and non-binary folks also in the sample. And secondly, more mediating variables need to be explored, such as sleep quality, loneliness, subjective well-being, etc. And the sample size we have selected is only 100, which is very low. So there is a very less chance of generalizability of the results obtained. Some of the implications of the study is that we can design certain intervention programs for the school students, especially for those who have a higher usage of social networking sites. Secondly, school curriculum can be modified to include subjects such as cyber awareness and safety lessons for the students, how they can remain safe while using various social media platforms. And lastly, the students can be encouraged to make use of certain self-monitoring techniques, such as usage tracking apps, which can monitor the level of social media use and hence moderate one's usage. These are the references. Thank you.
Thank you, ma'am. Over to you, judges.
Yeah, good idea it was. Then no questions because you are clearly explaining about your limitations and your implementations also. So there is no questions. Yeah, thank you.
So are you going to expand the study or are you going to stop it here? Because the sample size is
I definitely do plan to expand the study by incorporating all the limitations that I've discussed.
So you are going to correlate the screen time with the implications, or it's only about the number of samples? Or you're going to increase the number of variables?
So yes, sir, I would incorporate more variables such as sleep quality and academic achievement and subjective well-being. All these variables also I intend to study, sir.
And you still know that there is no standardization of the mental health related to cyber. So are you referring the standards, UN standards?
So there have been many surveys that are conducted regarding the usage metrics, but as you said, sir, rightly, that there are no standardization as of yet.
There is no classification in the ICD also, but there are only a couple of classifications, but that are related to psychological aspects.
Yes, yes.
So I think you need more sample size and more variables to bring those conclusions also.
Yes.
So that's my only observation. Other than that, very nice. Thank you. Best
Thank
wishes. Thank you.
Thank you, ma'am. Thank you, sir. We have the next presenter, Ms. Pooja Renuka Das.
Yeah, hello, I'm back.
You're audible, ma'am.
Yeah, I'm just trying to share my screen. Let me know when it comes up.
Ma'am, your screen is visible? Yes, ma'am.
Is it in the slideshow mode?
Yes, ma'am. You can start, ma'am.
Okay. So I'm back again with another topic of linking graphology with 7 chakras. So the title is Graphology, Ultimate Healer of 7 Chakras. And the topics that I'll be covering is, we've already seen about what graphology is and how it is interlinked with personality. So I'll take it to my advantage since we've already discussed it in the earlier one. I will spend more time on chakra, their characteristics, their benefits, and how we are linking chakra with graphology and what can we conclude out of it. So graphology we've already seen. It is the study of handwriting to reveal a character and a personality of an individual. And it actually, I mean, it is coming from the conscious and the subconscious mind by a collective activity of the brain, muscles, and a coordination between the nerves too.
And there are multiple aspects of personality which we can find out in graphology, which helps in on many aspects such as energy and vitality, feelings, moodiness, emotions, problem solving ability, etc. So zones is what I had not covered in my previous presentation, and I would like to spend a little bit more time on this. So basically, zones in handwriting, there are 3 zones in handwriting, which is the upper zone, middle zone, and lower zone. The upper zone is the one which reveals or displays or discloses more about the conscience or the intellectual, philosophical values, the spiritual aspects that the person thinks about and thinks and implements. So these two actually become different fallouts when we are seeing it in handwriting as a trait. So it also, I mean, the upper zone also talks about his imaginative powers, his creativity or creative ideas. And it's only about ideas here since it is the upper zone, and his religious aspirations also. Now, the second zone is the middle zone, which is the zone wherein the person, how he spends his daily routine, his practical life is what we can find out. So the upper zone would only talk about his thought process or what his thinking is, but the middle zone would actually mean how much of that has he actually doing it in his practical life. So it also talks about the mundane tasks that he executes on a daily basis, also his likes, dislikes, his habits, his emotional attitudes and reactions, and his adaptability to reality and his everyday activities. The third zone is the lower zone, which reveals the subconscious and the material wants. So it also talks about the actions that he takes. So what he thinks can be seen from the upper zone, and how he implements the zeal that is seen in his activity can be seen from the lower zone. Lower zone is also it actually also correlates his business interest, his instincts, his physical life, his sensuality, sex drives, etc. So a balance between these 3 zones actually indicates how balanced a person is and how integrated he is. A disproportion, such as, it could be one particular zone could be exaggerated. For example, the upper zone is too tall, which means that it is exaggerated and it is done at the expense of the other zone. So he will be more into thinking about spirituality or philosophical aspects, intellectual. But if his lower zone is not supporting, or it displays, the lower zone says that he's not more action oriented, so his philosophical thought process will remain only as a thought process and will not be implemented into his actions. So that's what we can see through zones. So zones become a very important part, especially when we are dealing in this topic of chakras. The rest of the handwriting traits we've already covered: slant, garland, connections and thinking patterns, as well as size, space, and speed also depict a lot of things about the personality, and they are termed as handwriting traits. Now, moving on to the next slide, it is about chakras. So what is a chakra? We actually live in the realm of energy, whether we acknowledge it or not, but that is the fact. Human beings are more than a physical body. We are a complete energy system that unites at all levels of being into an energetic totality. So basically, chakra is a vortex of moving energy which are always active in our body. These energy vortices, they actually transport energy from the universe around us into our aura and our physical body. So becoming acquainted with it and accepting its effect on our body, mind, and spirit, it actually helps us in giving us a mastery over our actions, thoughts, and attitudes. And this is not just restricted to human beings. Even animals have chakras, but I mean, the number of chakras differ from what humans have. Chakras are also psychic centers as they're connected to our thoughts and feelings and attitudes about life. And they're actually, they stimulate the various endocrine glands or the ductless glands in our body, which secrete hormones in the blood system. That's what makes it an important aspect with respect to our existence and our consciousness. Using chakras, we can actually track and sort emotional, mental, spiritual components of illness through physical symptoms. We can get to the root cause of an issue and we can unlock the energy blocks. We can also nurture and heal our own inner child whenever required. And we can understand why we are stuck in life, what are our harmful habits, how they affect our own progress, and what should be an effective solution for this. So working on 7 chakras, actually, we can have 2-point awareness where, first we identify and acknowledge our positive traits. And the second one could be we can uncover and understand and change our self-destructive beliefs and patterns and programs. So basically, there are 7 chakras. And there are a lot of details that goes around chakras, which could be like, I've listed just a few of them. There are many more that can be incorporated here, but the basic ones is like the location of the chakra, what glands it affects, the functionality that is covered at a physical level, as well as at a spiritual level, as well as a mind level, and the energy blocks that it gets us into. So the first 3 chakras are the Mooladhara, Swadhisthana, and Manipur. The next 3 is Anahat, Vishuddhi, and third eye. And the 7th chakra is the crown chakra or the Sahasrara chakra. They are all related to very different functionalities, and they start from the basic one, such as grounding and security. The second one, or the Swadhisthana, is relationship and emotions. The solar plexus or the Manipura chakra is related to energy and vitality. The Anahat chakra is related to balance and love and connections or relationships. Vishuddhi is related to communication and healing. The third eye or the Ajna chakra is related to intuition and understanding. And the Sahasrara is related to enlightenment and transcendence. Now let us correlate handwriting in 7 chakras.
Sorry to interrupt, ma'am. Your time is up.
Yeah, I'll just finish it another 1 or 2 minutes. Chakra chart can be, thank you so much. Chakra chart can be considered as an arrangement of 7 chakras in its natural sequence indicated by their color in a vertical stack. So this is a chakra chart, and here I have placed those all the alphabets, English alphabets, based on these 7 different zones of chakras. I have mapped the 3 zones of English alphabets. And here we have, I've listed the associated letters with respect to all these 7 chakras. So here we see the first 3, the Mooladhara, Swadhisthana, and Manipura chakras. They're all related to letters which only fall in the middle zone and the lower zone. Whereas as we proceed ahead with the Anahat and Vishuddhi, here again, they are still only in the middle zone. The lower zone is not accounted for in these 2 chakras. Whereas the Ajna chakra and the following, the Sahasrara chakra, they have letters only with the upper zone and the middle zone. So here we can conclude that 7 chakras and graphology, since they're working on the same concept of emotions and thoughts, feelings and attitudes, we can combine both these studies so that we are able to understand better and overcome our limiting beliefs. And that way we can evolve and transform on all spiritual as well as practical aspects, achieving a greater success as well as consciousness in life. And that gets us to the end of this presentation.
Thank you, ma'am.
Thank you so much.
Over to you, judges.
Yeah, Pooja ma'am, I have a question.
Yeah, sure.
Based on your graphology, Is it possible to recover, cure or recover from that hormonal disorders?
Yes, very much. Because as I said, these letters fall into different chakra zones, different chakras, and the basic concept is working on the ductless gland. So when we practice those letters in the correct form, they actually open the neural pathways. New neural pathways are created, which indirectly or rather directly gets associated with all these ductless glands, which could be the pineal or the thyroid, parathyroid, adrenal, and all these other ductless glands. And that's how they work on different hormones. So on a physical level, many of the medical aspects actually get covered if we are practicing those letters in the right way, in the right formations.
Okay.
And that science is called as graphotherapy.
Okay. So any age groups or any particular durations?
Yes, so we can start this only after the age of about 12 to 13 years old. And the upper limit is, there is no actually upper limit. Anybody who can hold the pen. So usually about the age of 80, senior citizens, usually their hands shake. So over there, it becomes a little bit difficult. But in cases where where the hands are not shaky, a person can definitely, even after the age of 80, he can practice it. And the time limit, we can say, so usually to form a habit, we need 21 days. But to actually get it implemented in our system, it takes roughly about 99 days. So it's a 3-month kind of an activity which we can do. And after using or doing it for 3 months, it actually becomes a usual activity for us. So it becomes an ongoing thing, and that definitely helps. It goes a long way.
Okay, thank you.
Yep.
I don't have any questions. You can go ahead. I don't have. Thank you.
Okay, sir. Thank you.
Yeah.
Thank you, ma'am.
Yeah, thank you.
Our next presenter is Subha S.
Hello, can you hear me?
Yes, ma'am.
Is my screen visible?
Yes, ma'am, your screen is visible.
Okay, so good afternoon all. My name is Shubha, and the title of my presentation is Evaluating Generalizability of Deep Learning Models Using Indian COVID-19 CT Dataset. I work under Dr. Neeta Parikh at CCNSP in IIIT Hyderabad. This is a brief overview of my presentation. I'd be talking about COVID-19 and the dataset that we have constructed and the model architectures, what we have used in the study, followed by the results and discussion. Well, our objectives were initially to build an image data repository for COVID-19 and then to evaluate CNN-based deep learning models generalizability to classify COVID-19 CTs. Before I get into the details, I would like to show some of the typical radiographic features of COVID-19 patients. The image that you see on the left side, which is showing some hazy shadows in the lung region, which is called ground glass opacities, which is seen in the earlier stages of the infection, followed by denser shadows, which are called consolidations, and also a web-like pattern, which you see on the right side image, which is called the crazy paving pattern. These are a few very typical image features seen in COVID-19 patients, and a couple of more of them. Our question was whether we need very deep learning architectures to identify these patterns, because there are only a handful of patterns and they don't constitute not much of variations like any other non-medical image. So this was our motivation. Roberts et al. Had reported that none of the models published so far had any potential clinical use for identifying COVID-19. Because of various reasons, maybe they might have trained their model using a very small sample size, or they might have used only a part of the training set for their evaluation of the model, so that the model fails to generalize on an unseen data, or they might have used unverified data for the training and validation of their models. And most importantly, they might have used very deep learning architectures that might overfit the data. So in order to avoid these biases and to build or to test the performance of the generalizability of models, we have tried to take up the study. And these are the data sets that we have used for training and testing our models. We have used the largest benchmark data set available for the CT images of COVID-19 patients, which is called COVID-X CT, which at the time of our study, when we took up the data, involved roughly 2 lakh CT images from 3,745 patients. And this data set had been split into training, validation, and test set in the ratio of 60:20:20. And as you can see, we have used 2 different test sets. The first one, the part of this training data set itself, which is the internal test data, and an external test data, which we have constructed, which we call Indian COVID-19 CT data set, and which constitutes of 12,096 images from 288 patients. Since we didn't have normal and pneumonia images, we had taken the normal and pneumonia images from the internal test set itself, as our focus was on identifying the model's capability in identifying COVID-19 images. So this slide shows the process that we had followed in constructing the Indian COVID-19 CT dataset. We had received 288 patient data from Gandhi Hospital during the period of April to September 2020. And they were all CT scans, plain CT scans captured with no contrast, and the slice thicknesses of these scans were 0.6, 1.5, and 5 mm. We had only considered 42 slices from each patient CTs, which covered the broadest lung area and also the clearest lung window without any interfering organs. And this was also done to reduce the dataset size. Finally, we had converted all these slices to PNG format before using them. This slide shows the representative model architectures that we have used in our study. Start from the top. It's a simple CNN model consisting of 6 convolutional blocks, followed by VGG16, which is slightly more complicated than the simple CNN model, again with convolutional blocks, followed by ResNet 50, which is a very deep 50-layered model with convolutional block, identity block, and residual connections, which are used for overcoming the vanishing gradient problems in very deep architectures, followed by Inception V3 network, which is the more recent network. And in terms of number of parameters, what you can see on the left-hand side, the CNN has only 2.9 million trainable parameters, whereas VGG16 has 138 million parameters. ResNet 50 and Inception V3 with 23 and 24 million parameters respectively. In terms of complexity from top to bottom, the complexity of the models increases. So we were finding out whether to identify the COVID-19 features in the CT images, do we need a really deep architecture, or a simple model, a simple CNN model could do the task. So here I present the results what we had obtained from all the 4 models, with ResNet 50 topping the chart with 99% accuracy and 0.98 precision and recall. The simple CNN model had a comparable performance with 96% accuracy, 0.99 precision, and a recall of 0.94. This was with the case of the test data from the internal test set, COVID-XCT test data. But when it comes to the external test data, these graphs here show the performance on the external test data, which is the Indian COVID-CT data set. And there are 3 bar charts given here. The one on the top left side is for the COVID-19 class. The first 2 bars gives the precision and recall on the internal test data set, and the next 2 bars gives the precision and recall on the external test data. We have given both the results in the same bar chart here to understand the dip in the performance on this unseen data. You could see there is recall of all the models, all the 4 models, CNN, VGG 16, Reston 50, and Inception V3 had dipped in this unseen data. The most affected one was ResNet 50. What we could understand from this was that ResNet 50 could have been overfitting on the training data. That's why the 99% accuracy that we had seen. This was what happens when the model is very complicated or very deep. Whereas the CNN, the simple CNN model was able to generalize better compared to the other deep learning models. And the other 2 bar charts are on the for the normal and pneumonia classes. And the confusion matrix that is given on the right bottom is for the CNN model. As we can see from the 12,000 images, what I was talking, 9,000 plus images of COVID-19 were classified correctly. You could see 2,000 plus were classified as normal. What we could infer from this was most likely many of those images may be either from patients with no lung infections, or they could be from the initial stages of infections, so the model could have identified them as normal images. A few of them also was identified as these were the incorrectly classified COVID-19 images. 670 of them were classified as pneumonia. So to conclude, we had tried to identify the generalizability of CNN-based deep learning models, and we could figure out that a simple model is better generalizable than very deep learning architectures. And we have tried to avoid the biases which was reported in Roberts et al., as I discussed in the beginning. And we were able to build a data set from Indian population named Indian COVID-CT data set, which could be a valuable resource, which can be a benchmark data set for various ML-based analysis. And as I said, we tried to avoid the biases by using the largest possible benchmark data set available for training the models. And then we used only radiologists verified data for training the models. And we also had done testing using multiple sets, including the external test set also. With that, I would like to thank all of you. I'm open for questions.
Thank you, ma'am. Over to you, judges.
Shubhu, yeah, it is very good work. I have a question from my side.
Yes,
for choosing the sample, what is the inclusion and exclusion criteria for the?
Sorry, ma'am. Can you?
You're collecting the data late, CT scan reports, right?
Yes.
So you're having any inclusions and exclusion criteria for this? Any comorbidity, or they have age groups or very severe conditions in this way?
If you're asking about the data set, what we had collected, we had only looked up on the image data set. We haven't considered the comorbidities or any other criteria for including them.
No. Actually, you are collecting CT reports, it is okay, fine. But you have to check about whether they have any comorbidities, what kind of diseases they have, any serious issues it will be, because these are all the major factors, because you are comparing COVID-19 normal and pneumonia patients, right? So this is the important factor.
Yes, ma'am. Okay. So for all the data that we have with the other clinical features, we would be definitely considering for our further studies.
Yeah, whether the age group, then whether the male or female, which age groups, what about their comorbid diseases also. These are all the important factors. Okay. So then you can compare. Okay, these things of COVID-19 patients having these kind of comparable, no, you can compare. Apart from that, generally you can't able to compare COVID-19 normal or in the case of pneumonia. Okay, because it is also inducing the main cause of pneumonia. Okay.
Yes, ma'am.
That is
In
the
this
thing.
work, we were trying
Yeah.
to compare the generalizability of deep learning models, basically.
Yeah.
Okay. Okay, ma'am.
Okay, fine. Thank you.
Thank you, ma'am.
So what is the working efficiency of your model, final model?
Right now, what we got on the internal test data was the accuracy of 96% and with a precision and recall of 0.98 and 0.94 respectively. And on our external test data, we got an accuracy of 88%.
That means the external data, the percentage was quite less, right?
Yes, because that is an unseen data, and this is bound to happen for deep learning models, that they dip a bit when it comes to an external data. Models have to be trained on very widely distributed data sets in order to make them more and more generalizable, sir.
Are you continuously working on training them, or how is
Yes,
it? Are you
yes, we are right now collecting more data from different parts of the world and more data set from wherever it is available in order to make the more data more generalizable and making the model available for others to test. Also, we'll be soon putting it on our website.
So what kind of ML algorithms you have utilized basically?
Right now, we have only used CNN based deep learning models. So all the models that we have tried uses these convolutional modules there. So CNN based deep learning models are the ones we have used.
Specific reason to choose CNN? Or?
Because we work on only image data right now, and convolutional neural networks are supposed to be the best on analyzing image data.
No, that is true, but still you have converted the image to a PNG format, right? So that's why I asked the question. Because all your images are converted to PNG format. Basically.
Yes, all the images, but CNNs work on any image data formats, sir.
Yeah, that's what I was asking. Why only PNG you chose? Is there any specific reason, or
that is yes, because we need a lot of data for training the models, and the publicly available data sets were all giving the data in either JPG or PNG data formats. DICOMs were available, but only very few of them, not sufficient for enough for training the data. So we had to convert our data also to PNG, but we also have the DICOMs available with us as you get more and more data in DICOM format. Yeah.
You are focusing on the pneumonia aspect, and you are trying to locate the patterns. Have you tried with post-COVID also? There will be similar parameters because you are not worried about the root cause. That's what you're not worried about the other clinical parameters. You are only focusing on the pattern, right?
Yeah, right now we are working only on the
it doesn't matter whether it's
features.
a COVID data or not COVID data. Anybody who has similar patterns can be identified using your model. Am I right?
But the we have trained the model to identify between only, no?
The focus is to develop a CNN model which can identify a specific pattern. That's the point.
Yes.
It doesn't matter whether it is a COVID patient or non-COVID patient, because the focus is only on the image, not on the parameters, background parameters.
No, other clinical parameters have not been considered so far.
Since you are not considering clinical parameters, you can try with any kind of data other than COVID also. So is there any specific reason to choose COVID, or it's a group work in your team you are doing COVID data work?
Yes, we are doing COVID data in our team.
That is the reason you are
we wanted to contribute something towards this COVID-19 efforts to tackle this COVID-19 pandemic for the society.
I think there was an earlier presenter who also worked in the same group. I think he had the same data set or similar data set. Yeah, I don't know. Anyway, that's fine. It's a good work. Hope you continue more, and best wishes. Thank you.
Thank you so much. Yeah. Can I stop sharing?
Thank you. Yes, ma'am.
Yeah.
Thank you, ma'am. The next presentation is by Christina Mariam.
Good evening, everyone. Am I audible?
Yes, ma'am.
Okay. What about the screen? Is it visible?
Yeah, screen is visible. You can start, ma'am.
Yeah, sure. Thank you.
Ma'am, can you please put it onto slideshow mode?
Yeah, it's on. So, good evening, everyone. Respected judges, Dr. Ravindra Henry and Dr. G. Dhanlakshmi, moderators of this afternoon session, dear fellow presenters and my dear attendees of 3rd International Conference on Bioinformatics and Data Science 2022 and 9th International Conference on Public Mental Health and Neuroscience 2022. Myself, Christina Mariam Chaco, research scholar in the Department of Psychology. I have done this research with Dr. Sonia George, Associate Professor in the Department of Psychology, Government College for Women, Thiruvananthapuram, Kerala. Today, I'm here to present a paper on multiple case analysis on work-family conflict and financial stressors among COVID-survived newlyweds. First of all, I would like to thank all the organizers of this conference for giving me this opportunity to present our research paper on this platform. So getting into the major contents of today's presentation, the introduction, which briefly gives a background description on the topic, its need and significance, research objective, and familiarizing the keywords of the current research. The research method where the procedures adopted in the current research is discussed, result and discussion where the semi-structured interview questionnaire that is used in the current study is given, and the discussion of the key areas of the couple's interview are also included here. And finally, the conclusions of the study. Yes. So newlywed couples undergo a high rate of change in the early years of the marriage, both within the relationship and outside, which can promote couple bonding or lead to the disintegration of the marriage. Almost all newlyweds report strong optimism for the relationship and the future at the start of the marriage. However, alarming rates of divorce in Kerala shows that married couples are finding it really hard to adapt to their relationship and maintain it, making them adopt divorce as a primary option for solving their problems. A healthy and stable marriage promotes better mental health and individual development, whereas negative life events reduces mental health. For the past few decades, there has been an increase in the percentage of women entering into the workforce, and a significant proportion of them enter cognitively demanding jobs and work extremely hard alongside men to climb the professional success spiral. The research by Smith and Rasagi in the year 2020 portrays that highly educated women prefer to be with partners who are also highly educated and work full time. When both the domains, work and family, are equally demanding, it builds tension and exhaustion, not only in the individual, but also in their partner, affecting the psychological well being of both partners, impairing their marital satisfaction. The conflict between work and family arises from an individual's lack of time and energy when dividing their resources between the two. If the person is successful in striking the ideal balance between work and family, it gives them a sense of high achievement at work and serves as a motivator for them. Individual and couple financial behaviors, according to the Brit, are one of the primary causes of relationship dissatisfaction and also may influence divorce. Understanding how couples manage their household financial expenses and deal with surplus income can help couples address their core financial conflicts. Researches by Ju et al. Shows that married couples often end up in fight not for how much they earn, but mostly over the financial management practices. These financial arguments sometimes create resentments in the partner, which are more likely to dissolve the relationship. Appropriate financial decisions are frequently the result of financial literacy, which includes a fundamental financial knowledge, such as inflation, compound interest, investment types, and so on. Young married couples appear to be unsure of how to communicate and what important factors to emphasize when discussing their finances. So the marriage becomes unequal when one spouse handles household finances on their own without consulting the other. The less fortunate spouse frequently feels constrained in his or her ability to manage household finances and access to money. However, there is less inequality when the husband and wife jointly manage the finance of the family. Couples often find it difficult to discuss their financial situations with one another because personal finances are considered to be one of the most personal and private facets of the society. Knowing their own financial situation, each other's money personality, and their current debt situation can help spouse reach an agreement on their household finances and also can avoid unnecessary arguments. On the other hand, some couples believe that keeping financial discussions to minimum with the partner helps to maintain relationships by preventing pointless financial conflict. Managing household chores, work-related tensions, financial management, and meeting partners' emotional and physical needs is taxing for many newlyweds. It is in this context that this research tries to assess the coping mechanisms used by COVID-survived newlywed dual-career couples to manage work-family conflict and financial stressors through multiple case analysis. Getting into the keywords and operational definitions, dual-career couples are well-educated couples who are committed to their professions and cope with the conflicting pressures of their career, their spouse's career, and their family responsibilities. Work-family conflict, an inter-role conflict that develops when the responsibilities and demands in one domain makes it difficult to fulfill the responsibilities of the other. Financial stress, which is a stress related to one's financial situation. Newlywed couples who got married within 1 year. COVID survivors, persons who tested with coronavirus positive, who were later confirmed to have had the virus by testing positive for antibodies. Getting into the research objective of the current research. This is to examine the coping mechanisms used by the COVID-survived dual-career newlywed couples to manage work-family conflict and financial stresses. Now let's look into the research method. Multiple case study approach is adopted here. The sample consists of two newlywed dual-career COVID-survived couples who were married in less than 1 year span of time were selected. Couples who are dual career professionals were chosen for the research as to see how being into cognitively demanding jobs might contribute to their work-family conflict, as well as how they deal with their financial matters in terms of savings and expenses. It was decided to focus on the ones who had reported with severe COVID-19 symptoms in order to examine how they handle stressors despite the virus' physical and psychological effects. Informed consent was obtained from all individual participants included in the study. A semi-structured questionnaire was prepared well in advance and expert opinion was taken regarding the nature and content of the questions. Now let's look into the results and discussions. So as I mentioned earlier, two heterosexual dual career couples were chosen for the study. The interview was scheduled as 2 sessions on the same day. Work-family conflict was focused in the first session and financial stressors in the second session. These are the questions that were included in the semi-structured questionnaire. Yeah, so now getting into the summary of the interview transcription got from the two couples. So first couple, PS and SM. PS is a 24-year-old female and SM is a 26-year-old male who are married for 4.5 months and have pursued their professional careers. As newlyweds, they are still getting to know each other and each other's families. They make an effort to communicate honestly with their partner. Differences of opinion are sought more patiently by outlining the reasons why they prefer certain ways of doing things. Even when the partner disagrees with what they propose, they treat things objectively and without taking it personally. In terms of their professional lives, they also respect one another and understand and accept the difference between them. Even if there are family issues, being busy at work makes them less emotionally invested in them. In addition, having a peaceful and supportive home environment helps them unwind after a stressful day. They prefer not to talk about work-related issues at home or home-related issues at workspace. Due to the fact that they prefer
Sorry to interrupt, ma'am.
Yes.
Ma'am, sorry to interrupt. Your time is up.
I'll conclude in a minute.
Okay, ma'am.
So getting into their budget, they're feeling greatly at ease when they're creating a joint budget, and also they haven't started anything yet, but they are talking about their future investment plans. As they're leading a very simple life, they find it really easier to manage their family as well as their career. And getting into their cooking and all these parts, they are handling it. Both are handling together, so they don't find it much worried about it. And getting into the personal and professional lives, they also have the conflicts and disagreements, but they come into general terms in terms of their likes and dislikes in many other areas, which helps them to get away from the deconstructive criticism, even if it happens in between. So getting to the couple two, they are the 29-year-old male and 27-year-old female who are married for 5.5 months and had been acquainted for 4 years. And they are engaging in consulting, teaching, and all other activities together. This couple seem to have a really high amount of work-family conflict because they are doing n number of tasks as a means of income. So they are getting very little amount of time to do their household chores together. So they are having high amount of work-family conflict. And but they're getting some quality time as they're working in the same institution, which helps them to have some common friends in their network. Ego conflicts are also very high in this relationship. As a means of rejuvenation activities, they try to hang out. This couple finds it really difficult to manage the finances because one of the person, the wife among the couple, is not so interested in discussing the personal finances. So it becomes very difficult for them to handle the financial matters. So they are maintaining a joint account to overcome the situation. Getting into the conclusion, couples' personality characteristics and situational factors such as their job environment, family environment have been shown to have a significant influence on how they handle stressors like work-family conflict and financial stress. So summarizing the first couple, even though their marriage was arranged, they can adapt to each other's present in the relationship better because they're both adaptable and not overly emotionally attached to criticisms. Their rejuvenation activities are similar, like watching movies at home or cooking new dishes at home, and it is very simple. Their financial matters are like they're more open about their finances, and since they're minimalist, they don't incur many costs, which makes them less concerned about money issues. And the work-family conflict, their couples maintain a work-family balance in general because the cooking and cleaning chores are largely taken care of while they're staying at their parents' house. The second couple, they are being in a love relationship for almost 3 years and are both in the same profession and workplace. They go through power struggle, which causes them to quarrel over trivial matters and prolong their fights. The rejuvenation activities are different, even though they like travel together, but the husband prefers to drink few drinks to deal with the situation if it is a stressful one, whereas the wife prefers to journal down. The financial matters is like wife believes discussing personal finances is offensive because she considers them to be the most private topics, because of which they are trying to maintain a joint bank account. Work family conflict, it is very difficult for them to find family time and participate in household chores because they live alone and have very busy work schedules. So acknowledgement, we would like to thank all the respondents of this study for their cooperation. These are the references that are used. Thank you so much for your patient listening. I'm open for any queries.
Thank you, ma'am. So you have any question?
No, ma'am, I don't have. My only concern is it's very less sample size. We cannot conclude anything on only 2 couples.
So it is a case analysis. That is the reason why the limited number of samples chosen.
Multiple case analysis, right?
Yes, sir. Multiple case analysis can be done with 2 or more participants. So for the current research, I have included only 2 couples.
We cannot make any generic conclusion based on only 2.
Regarding the case studies, the generalizability is one thing which have to be taken with a caution. Like it is studying 2 couples in their own way. We are not trying to generalize anything. It's studying them. That is the main purpose of a case study too. Right?
Okay, thank you.
Thank you, ma'am. Thank you, sir.
Thank you so much for this opportunity.
Yeah, thank you. The next presentation is by Jayanti Shukla and team.
Am I audible?
Yes, ma'am, you're audible. You can start sharing your screen.
Am I also supposed to turn on my video?
Yes, ma'am.
Okay,
can you please put it onto slideshow mode?
Okay.
Yeah. Thank you.
Is it okay now?
Yes, ma'am. You can start.
Okay. Good afternoon, moderators, organizers, judges, and participants. Welcome to my presentation. My name is Jayanti Shukla and I am a student at Department of Biotech, Graphigera, deemed to be University. As you can see on the screen, my topic for today's presentation is in silico analysis for the identification of differential expressed genes and candidate biomarkers in Kawasaki disease. To begin with, first we need to know what is Kawasaki disease. So Kawasaki disease is an acute febrile systematic vasculitis, which means that it is a chronic autoimmune disorder characterized by inflammation of blood vessels and also characterized by frequent fevers. And it is also a it is a rare and lethal disease, but it is commonly observed in children in age groups of less than or up to 5 years. Now, to move on to our topic of studies, that is the identification of differentially expressed genes, we first need to know what are differentially expressed genes. We all know that in all somatic and germ cells, the content and the amount of genetic material is the same. But in different types of cells, for example, the liver cell expresses different genes, the heart cell expresses different genes because of their various functions. So this is a study to identify which genes are expressed the most in Kawasaki disease, so that we can find out various drug designing mechanisms or drug repurposing mechanisms or a novel treatment for this disease. So in the study, a total of 36 samples were selected, in which 12 were control and 24 were Kawasaki disease samples. And on those samples, various screening strategies have been applied to find out the DEGs expressed in Kawasaki disease. So as I already gave an insight, the disease is common in children and usually accompanied by fever. And in lethal cases of this disease, the coronary artery, which is the main artery of the heart, inflames and collapses, leading to the death of the patient. So our primary goal for the study has been identification of differentially expressed genes and to construct a protein-protein interaction network based on it, which would give us further insight onto the pathways of the disease. To reach our goal, the methodology which we applied for is divided into 4 steps. First step was to retrieve the microarray data. So in this, gene expression omnibus database was selected, and through it, GSE series of data set was extracted, in which we had the information for all Kawasaki disease patients. And a total of 36 samples were selected, in which 24 were Kawasaki disease patients and 12 were control samples. Our next step was the identification of differentially expressed genes. To do this, the tool GEO2R was used to screen DEGs between Kawasaki disease sample and control samples. The next step was to perform a GO and KEGG enrichment pathway analysis. The pathway analysis is to find out functional level gene groups which are expressed in a variety of pathways and are the most commonly expressed gene groups. Our last step was PPI network analysis and to construct the PPI network. And in this, string database was used to construct the PPI network. So for the identification of differentially expressed genes, our first step was to extract the data set from GEO database. Further, GEO2R tool was used to determine the differentially expressed genes between control and KD samples. And this was performed using the online free tools and some other online tools which are popular and very efficient. And the second step was construction of PPI network and identification of hub genes. The PPI network was constructed by the string server, and it is important to understand the proteins which are expressed and proteins which are common in both in GO and KEGG analysis. So after extracting the data sets from GEO database, these are the 36 control samples which were considered on the left side, in which we see the control and KD samples. In the right figure, we see the expression density, which has been obtained as a result of running it on GEO2R tool, and it has various results and profiling based on the samples which were used as query into the data into the tool. Now, p-value count is the count which is responsible and significant in telling us that what significant value count is actually able to be considered for our multiple sequence comparison in a hypothetical data set and a hypothetical query which we are handling. So these adjusted p-value counts were based on the parameter that all p-values should be less than 0.05%, the considered values. Our next step was to get a moderated t statistic. And a moderated t statistic was also found out after performing analysis in the GEO2R tool. Now this is the KEGG pathway analysis and GO pathway analysis, in which we have found out that the KD disease were simultaneously enriched in phagosome pathways, graft host diseases, cytokine reactions, receptor interactions, and various other number of diseases. Similarly, for GO pathway analysis, we have found out that it is enriched in various pathways, for example, interleukin-12 production, innate immune responses, and positive regulation of NF-kappa B transcription factors. Now, this is the last step where a PPI network was made using the string server. And in this, a total of 172 nodes and 349 edges were found. Now, nodes represent the proteins and edges represents the interaction. This is important for further evaluation and study of the disease. In conclusion, we found out a total of 1,354 differentially expressed genes, in which 20 were upregulated and 15 were downregulated. This study has given an insight into a number of DEGs which are essential in further finding out candidate biomarkers, probable or potential biomarkers, which will obviously be used to study the disease further, design a treatment. It could be drug repurposing or a new drug designing, and further research and scholarly studies. These are the references. A lot of references were there, but I have included just a few here. And thank you. I'm open for queries now.
Thank you, ma'am. Over to you, judges.
No questions from my side. You can go ahead.
Yeah, why did you choose this particular disease? Any specific reason?
Yes, sir. So actually this disease, even though there are treatments possible for this disease, the treatments we see are not treatments which are directed at the root cause or at the genetic level. The treatments are to treat the symptoms. For example, the inflammation or edema symptoms or the fever symptoms, but they do not interfere with the root cause or solve the problem at its root. So it is necessary to conduct studies which provide an insight which will be helpful to design a treatment method which is actually going to cure the disease at the most basic and fundamental level, which is the molecular and genetic level. And this was the reason I chose this disease.
Okay, good. Good. Thank you.
Thank you, sir.
Ma'am, we have a question from Dr. Nandini Rao. The question is, which sample is used for data collection?
Which sample is used for data collection?
Yes.
Actually, a total of 36 samples were used, and they were extracted from the GEO database, which is the Gene Expression Omnibus database. And in that, the data profile which we used was GSE 109430. And further, when we extracted the 36 samples, table is also shown here. And these were on the left side, these were all the samples which were extracted from that database. These are all the samples. Any more questions?
Thank you, ma'am. I do have a question.
Yes.
You told that, you have used 19 controls. How, sorry, 12 or 19?
12 controls.
12 controls, okay. How will that make the difference in numbers? Like, is there any criteria, more than one criteria for that?
Yes, because if we are conducting any study on a genetic level, we cannot just take 2 samples or 3 samples against a huge number of samples and hope for it to be very accurate. This the number is 12 against 24 samples, because we need a large number of control samples as well to accurately identify if the differentially expressed genes are just the regular expression or there have been some other variable into the studies. So this helps us in screening exactly which DEGs are normally expressed or there is some other variable factor involved in it. And this is very necessary to screen it. Otherwise, the rest of the process will be performed on an error and the study would lose its precision and correction value, or the correct study and the results will not be obtained.
Okay, ma'am. Thank you.
Thank you. Should I stop sharing now?
Yes, ma'am.
Thank you.
Thank you, ma'am. We have a next presenter, Maheshwari Dhanachari.
Yeah, thank you. I would like to share the screen. I think the previous participant has to stop sharing.
Yes. Ms. Jayanti Shukla, can you please stop sharing the screen?
Actually, I can't find the stop sharing option. It is just resume share or, oh wait, I found it. Sorry.
Yeah, it's okay.
Yeah, am I audible?
Yes, ma'am, you're audible. You can
Is
start,
the screen
ma'am.
visible?
Yes, ma'am.
Yeah. Thank you, and a lovely evening to one and all to the journey of emotional being in parenting. I thank the entire team of 3rd International Conference on Bioinformatics and Data Sciences and the 9th International Conference on Public Mental Health and Neurosciences. And I'm Maheshwari Ganachari, Child Counselor and Parenting Coach from Unit 2. Today I'm here to present the paper on the journey of emotional well-being in parenting. So the role played by emotional well-being in parenting is really very, very important. The emotional well-being of the parents has a greater impact on the children. The emotional stability of the parents from the time of conception till the early 6 years of age can create a very healthy lifestyle of the child. Every parent need to be aware that when they do the parenting according to the uniqueness of the child, creating the right and the positive environment to the child can enhance the self-esteem and the self-respect of the child, wherein the child grows up as an emotionally strong individual. This emotional well-being is directly connected to the mental health. When I talk about emotional well-being, that means the thoughts, feelings, emotions, actions, and behaviors of the parents need to be towards the positive side. How they handle themselves, how they are managing their emotions, everything matters. For this, there has to be an awareness about self. Parents need to be aware of self, their uniqueness, and in the journey of parenting, the parents need to take care that they need to know the knowledge about the child, what is the uniqueness of the child, what environment has to be given. So this is how it is very, very important. And when we talk about the emotional stability of parents, we need to understand why certain individuals or a few class of parents are unable to manage their emotions, why there is an emotional instability in any individual. For this reason, for this again, the lack of self-awareness plays a very important role. They are not aware of themselves and they are not giving importance to the concepts of self-love, and they are not teaching the children that how I feel about myself matters a lot. It is always about what other people think about me. So here we need to understand the root causes for the instability. When I stand as an individual and I as a parent talk about my stability, my emotional stability is taken back to my childhood, what sort of parenting was done by my parents. And every parent is doing parenting based on their experience, what they have experienced consciously or unconsciously. Whatever the information they have grabbed through their 5 sensory organs from self, parents, or the society from the time they were in the mother's womb till their early 6 years of age. That period of my, time as an individual decides my emotional instability or stability, positive or a negative side. This in turn, I decide my child's emotional stability. So emotional stability here is very important and it is directly connected to my childhood. When we talk about this, when we see that, when I say I am unstable or any individual is unstable, we see that lot of behavior or characteristics like this in adults, like somebody is lazy or we exhibit unhealthy habits. There is addiction to TV from the, when we talk about adults also, there is lot of imbalanced feelings. Even adults have lot of fears. For example, fear of darkness, there is lot of negative thinking. We are unable to handle stress. We are unable to take the responsibility. Many people have trouble sleeping. There is a lot of guilt feeling in parenting. There is lack of proper communication. There is back answering. All these kind of behaviors we are exhibiting as an adult. When I as a parent have all these qualities and I am doing parenting, and we are aware that children are replica of parents in terms of their habits, behavior, and attitude. When I, as a parent, have all these qualities and unconsciously I'm doing everything, and my children are copying everything, everything is being recorded in the child's subconscious and unconscious mind. And child, at a certain interval, they start exhibiting these behaviors. When we see this, we can see that there are a lot of behavioral challenges in the children, like lack of concentration, fear, bedwetting, anger, poor memory, stubborn nature, ugly handwriting, disrespect to elders, rote learning, indiscipline, learning difficulties, eating challenges, slow reading, crying, unnecessarily crying, shy nature, feeling low again and again without any reason, and not a good handwriting. Parents come to us keeping all these behavioral challenges and looking for the direct solutions to these behavioral challenges. In the counseling session, we see that when parents come up with these kind of behavioral challenges, when we take the case history, we understand that the root cause is again in the parenting pattern. We understand everything in detail, that if we need to work on these behavioral challenges in the children, the emotional stability of the parent need to be balanced. We need to work with the parents first and then the child. So for example, parents come with an example that the 2-year-old child is getting anger. When we understand the entire case history, we see that there is a lot of disturbance in the environment. Even when we study a lot of autistic children, we came to know that during the pregnancy period, the mother had a very disturbed emotions. When in the entire case history, when we study, we see that the emotional instability has caused a lot of imbalance in the child's nature, actually. So to work on this, we need to work on the emotional stability of the parents. So ultimately, what I would like to present here is the emotional well-being of the parents leads to the right thinking, right communication, right actions, and the right associations, and encourage the right things. So thank you, one and all.
Thank you, ma'am. So you have any questions?
No, ma'am, no questions from my side.
Okay, sir. Thank you, sir.
Thank you.
Thank you, ma'am.
Thank you.
So we have come to the final presentation, that is by Murali B.
Once again.
Yes, sir. So your video is on.
Yeah.
You can start presenting, sir.
Yes.
So your screen is visible.
Yeah. But
Please put it onto slideshow mode, sir.
yeah, is it visible now?
Yes, sir.
Am I audible?
Yes, sir.
Yeah, thank you. Welcome to all unique brains who are present here to know about fingerprint analysis and the brain mechanism. We all know that our fingerprints are very, very unique in this world. No two fingerprints are same across the world. So these fingerprints are directly connected to our brain mechanism. In this presentation, I'm going to explain this, how these fingerprints are directly connected to our brain mechanism. Basically, fingerprint analysis is one of the concept. It's a purely scientific concept. Here, I will get to know about a person's personality, intelligence, and the areas need to be developed. Every individual has two faces, basically. The first one is personality, and the another one is intelligence. So basically, personality means it's going to tell about how a person think, how he feels, and how he behaves. And the same thing, intelligence means how I learn things and how I implement and how I share my experience, what I have got through. Basically, when we understand both the personality and the intelligence of each individuals, it's really help us to live a healthy life and successful life. But most of the time, we are failed to understand our original personality as well as our intelligences. We are aware that there are 8 intelligences. This fingerprint analysis is going to tell about the personality aspects as well as the intelligences. Along with that, we will get to know the weaknesses of a person also. So now let me talk about how this fingerprint analysis comes in the picture. So how exactly this evolvement happened. The Francis Galton is the scientist who is a far cousin of Charles Darwin. So here he introduced the basic concept of fingerprints, how exactly the person's behaviors are connected to his unique nature. And he has written many books like the fingerprints and the fingerprints directories. This book is going to give us a complete clarity and understanding about the each fingerprint patterns. I'm going to tell you how these patterns works. Basically, this fingerprint analysis works in analyzing these basic 3 patterns. The first one is arch pattern, second one is whorl and the loop. Here, there are 20 plus subfamilies in each one of us. So by studying all these fingerprints in detail to the core level, we will get to know that how exactly person's personality is. And I would like to show that there are the stages for this fingerprint analysis. There are mainly 4 stages. The first stage of the fingerprint would be like this. We call it as it's an arch family. And the second one is a loop family. Here, the third one is it's a whole family, double loop family. The fourth one is the whole family. Here, there are 4 stages. Based on these fingerprint analysis, by reading the ridge counters and the pattern of each fingertips, we are going to conclude that what is a person's personality. So here in the next way, we are aware of the brain pattern. So this is how our brain looks. So here we will get to know that there are 10 lobes in our brain. The 10 lobes are indicating through these fingertips. Basically, we know that there is a right hemisphere and the left hemisphere. In the both left and right hemisphere, we'll get to know the prefrontal lobe, frontal lobe, and parietal lobe, temporal lobe, and the occipital lobe. See, we know that our left hemisphere is controlling the right part of the body and the right brain is controlling the left part of the body. It means that my left brain is going to indicate through my right fingertips. The same way, my right brain is going to indicate many of the patterns through my left fingertips. I would like to explain each lobe. Here we will get to know that what are the qualities we would have in each brain lobes. So in the right brain, we can see that there is a right prefrontal lobe, which talks about our self-esteem, determination, and the intuitions. The same way with the right brain only, I would like to explain each things. And the second one is frontal lobe, which is connected to our thoughts and ideas. The third one is parietal lobe, which is connected to our entire motor skills, gross motor skill, which is connected to sports activity, dance, swimming, jogging kind of things. And the temporal lobe is going to talk about a person's feelings, emotions. And the occipital lobe is going to talk about the visual aspirations and which is connected to art. The left brain dominance is going to talk about the left parietal lobe, which is connected to self-achievement, behaviors, executions, and planning aspects. And the computation, logical reasoning comes under the frontal lobe. Parietal lobe is with the fine motor skills, and the temporal lobe is connected to communication and the language, and the left occipital is going to talk about the reading aspect and the interpretations. So basically, our brain lobes are directly connected to the fingertips. By analyzing the each fingertips based on the ridge counts as well as the brain, fingerprint patterns, we will come to know that which lobe has a high neuron density. We know that every brain lobe has 100 billion neuron cells. How many neuron cells are activated in each brain lobe? Accordingly, we will get to know the potential of each brain lobe. Accordingly, we understand a person's personality and the intelligence through some of the percentages also. We could see here that there are some percentages. Out of 100%, we will get a categorized and divided each lobe's density. And I would like to tell that, so every brain pattern are different. How exactly these brain patterns are different? Let us see this. So every person senses, listens, perceives, understands and analyzes and execute each activity which is called as unique brain pattern. So this is how every fingertip is unique. The same way everybody's natures, the senses natures are very, very different. So that we call it as every brain is a unique and it has a different brain pattern. So when we talk about the brain mechanism, it includes all hidden combination of a person's skills, passion, strengths, intelligence, interest, potential, weaknesses, and blind spots called as brain mechanism. So if I am not succeeding in my life, if I'm getting more failures in my life, I would not be aware of my brain mechanism. That is how I would like to follow everybody's input. My brain get confusion so that it unable to execute the activities or it cannot take actions. So here, through this fingerprint analysis, we will get to know the brain mechanism. And we would all know that we all are unique and we all have this unique nature. So it is called as uniqueness. One's personality in combination with their specific intelligence highlights the essence of uniqueness within them. And I would like to conclude this concept by, through this fingerprint analysis, we will get to know all these qualities of a person. Personality, thought process, perceptions, behavior, skills, qualities, intelligence, hobbies, grasping level, interest, concentration, memory, learning method, love languages, likes and dislikes, core values, passion, success path, blind spots, career choices, preferences. These are the main things we will get to know through fingertips itself, that is directly connected to the brain mechanism. The thing is that fingerprint analysis is the only scientific concept where we will get to know our inborn, hidden, latent qualities and the brain mechanism. Thank you.
Thank you, sir.
Welcome.
So, any questions?
No, ma'am, not from my side.
Okay, sir. Thank you.
Thank you, ma'am. Thank you.
Well, that was the last presentation for the day. I would like to thank all the presenters for their kind cooperation. I would also like to thank the judges, Dr. Rabinder Henry and Dr. G. Dhanalakshmi ma'am. Thank you, sir. Thank you, ma'am. Over to you,
Thank you.
Ms. Jovita.
Thank you, Ms. Anusha. Research is to see what everybody else has seen and to think what nobody else has thought. As a representation of the court, I would like to take the privilege to invite Dr. Kongfat Wonglin. He is a reader at the Intelligent System Research Center, School of Computing, Engineering and Intelligent System, and the Ulster University, UK. His research interest lies at an interface of computational modeling, mathematical analysis of system, cognitive neuroscience, psychology, brain disorder, and data science. He is an editorial member for the Journal of Neuroscience Method and an associate editor for the Frontiers in Integrative Neuroscience. He even received the 2011 IJCNN Best Paper Award and the 2016 Ulster University Distinguished Research Fellowship Award. He even obtained the Moray Institute Visiting Research Fellowship at the National University of Ireland, Galway. And in the year 2020, he received another award for Visiting Fellowship at the University of Oxford. We welcome you, sir. Yes, sir. So your video is visible.
Can you see my video?
Yes, sir, your video is visible.
Okay, good.
You're audible too, sir.
I'm trying to share screen.
Okay.
Let's see. Are you able to see my screen?
Yes, sir, we are able to see your sheet.
Okay, thank you so much.
Thank you, sir.
All right, so let's see when I can use this. Okay. All right, so thank you for the kind introduction. Before I begin, I would like to thank the organizers, especially Dr. Prenan Bachi for the invitation. In this talk, I hope to provide a flavor of modeling cognition, particularly decision making. For those who are familiar with artificial intelligence, AI, and machine learning, decision making forms a central tenet or feature of these algorithms. Here's a list of AI approaches used. However, the limitations of these algorithms include decisions being made based on statistical correlations only, and the algorithms lack a time component. But we live in a universe of space and time. Importantly, this type of algorithms lack self-awareness. So I will touch upon the AI algorithms later on at the end of my talk. So how about humans or animals in general? It has been said that an average person makes about 35,000 decisions a day. Some decisions are complex and difficult, while others are more like reflexes. However, there often lies a common important ingredient, which is the integration of uncertain sensory information, and then how a choice is being informed. Surprisingly, for simple-based decisions, we still know very little on how information is actually processed. In a more specific form of decisions called perceptual decisions, scientists investigate how uncertain sensory information is processed and integrated, and how a choice or action is selected based on the presented information. Often, responses to report choices have to be done very quickly and accurately in the form of a modal output. For example, in this case here, imagine you're driving a car and trying to make a decision on whether to turn the steering wheel left or right. depending on the visual information you receive. And obviously, having heavy rain can make the perceptual decision-making process much harder than on a sunny day. Some highly studied cognitive experiments often involve making sensory discrimination between two or more alternative choices. For example, determining which motion direction here of some visual stimulus. Importantly, psychologists or cognitive scientists can manipulate and control the stimulus parameters and also how the participants report the choices, often through some modal responses. For instance, participants may be asked to discriminate the overall motion direction here of a cloud of randomly moving dots, a fraction of which are moving coherently in the same direction, either leftward or rightward. The participant is instructed to move his gaze towards one of the two choices on the left or on the right of the screen. The participant makes a correct choice when the direction of the gaze moves in the same direction as the coherent motion of the dots. The task can get easier when the percentage of dots moving in the same directions is higher. Over the years, neuronal correlates of neural integration that actually integrate sensory evidence over time, very much in the calculus sense, during perceptual decision-making, have been found across different brain regions. A highly studied brain region called the area LIP was one of the earliest regions found. Here. In particular, a notable work on non-human primates or monkeys was studied by Jamie Reutemann and Mike Shetland. Here, neurons were recorded while the monkeys performed the motion discrimination task described earlier. They found that the slope of the ramping of neuronal activities here over time depends on the task difficulty. The harder the task, for example, with less signal, then the shallower the ramping of the slopes. This was also associated with slower reaction time in reporting a choice. Upon response, the activities are aligned at similar activity level, perhaps suggesting some fixed decision criterion or threshold. Here's a movie to show the recordings of the monkey neurons. I'm not sure whether I can play it, probably not. Let's see. Oh, okay. All right, you can see the random dots in the middle, and the sound, the bleeping sound comes from the conversion of the spiking of the neuron activity into sound. So as you can see, let me see whether I can play again. Oop. Yeah. So this is the correct error choice, and then followed by the correct choice. Right. Similar type of work have been done by my colleagues on humans using EEG and other neuroimaging techniques with similar results obtained. You can see the ramping of activity depending on the difficulty of the task. An important probabilistic framework or theoretical framework that was developed in the sequential sampling model framework of decision-making assumes some internal decision variable X over here that accumulate noisy evidence or information over time to some prescribed decision threshold or boundary. Upon reaching this threshold, a choice will be made and the decision time or reaction time can be obtained, which is this. Thus, these are considered evidence accumulated models. However, there are too many mathematical models right now, which are based mainly on the behavioral data. So there's a lot of degenerate models. Hence, accounting for neural data may help eliminate some of these models. Here, biologically based computational models, for example, spiking neural networks, you can see these are modeled by spiking neurons, almost like a heartbeat. And each neuron encoding each neuronal population encoding a different choice. Here are the neuron numbers, and this is time. Each dot over here are the spikes of activity of the neurons. And you can average them here to create population of activities. So eventually one neuronal population will win. And if their average activities surpass some prescribed threshold here, a choice will be committed. So this is the example of a winner-take-all behavior, when one guy wins and the other guy is suppressed. Sometimes further insights can be obtained with a simpler network model, which can not only replicate the biological and psychological observations, but also provides conceptual understanding of the decision-making mechanisms. So here, using mean field approximation borrowed from physics, theoretical physics, here we consider a group of homogeneous population of neurons as one representative neural unit. And also we make use of other approximations. So we can approximate thousands of spiky neural networks into two competing neural units, representing the two choices. With this model, we can explain how false choices can be made through attractors. These are the attractors, and unstable saddle point here. For example, shown on the right is some decision or energy landscape deduced from the neural network model's equation, and in the form of two energy wells or double potential wells, just like in physical sciences and engineering. The ball here represents the state of the decision network model at some point in time. In this specific case, choice 1 is the correct choice. You can see choice 1 is the correct choice because it receives slightly higher sensory input. And the state of the network would drop into choice 1, into the attractor well of choice 1. And then the choice 1 is being made. Of course, in the presence of noise, sometimes the other incorrect choice is selected due to fluctuations. So we can understand this with noise driving the ball to eventually drop into the other well, making choice 2, which is the incorrect choice. With a reduced model, one can also perform phase plane dynamical systems analysis. In the absence of stimulus on the left, there lies a stable steady state or spontaneously active state. But when a stimulus is presented favoring choice 1, then the basis of attraction, which is this orange color region, is biased at the expense of the other choice attractor. So it's larger. Hence, error choices here in red trajectory have to cross this boundary and pass through this unstable saddle steady state, and therefore reducing the speed of the trajectory. This explains why reaction time in error choices are slower than correct choices. And when the task becomes more and more easier, the basis of attraction of this correct choice attractor is increased at the expense of the other, and then more correct choices are increased. And this explains the accuracy of the choice behavior. This attractor network model can also replicate a later work that shows brief motion pulse perturbation on top of the random dot motion stimulus that you have seen earlier. And this has a lasting yet decaying effect on the choice behavior and the neural activity. The model shows that the inability of the late pulse perturbation is due to the runaway energy landscape in a model. Here you can see the choices are made by running away from this unstable saddle point. And therefore, perturbation becomes harder and harder to affect choice behavior at a later time. Such energy landscape can also be understood and visualized in the form of an extended decision landscape in which actual spatial coordinates are involved. For example, computer mouse or walking trajectories can inform the decision landscape of a single individual. And so we can predict the behavior of the person, of an individual. And this can be applied to neuromarketing, e-commerce, and neuroeconomics. Instead of the above handcrafted computational models of perceptual decision-making, data-driven approaches are now becoming more popular. In this work, my PhD student, seen on the left side, Brandon Lanfasti, made use of a recent machine learning algorithm developed for physics and engineering fields to uncover the nonlinear dynamical equations governing decision formation at single trial neuroactivity data. And this will inform choice, this can predict choice behavior. This algorithm made use of sparse regression here with a library of mathematical functions and assuming that the physical world can be described in low dimensions. Here is an example of a simplistic nonlinear bistable decision-making model. And the algorithm shown in orange can readily replicate the single trial activity and choice accuracy and decision time of the simulated data. Some recent challenges facing perceptual decision-making occur when people record more neurons. They found heterogeneity of neuronal responses during decision formation over here. So it's not always a clear-cut winner-take-all behavior. So here is a single neuron recorded in monkeys during perceptual decision-making. This is also found in behaving mice, here with 65 neurons simultaneously recorded. Here, the dimensional reduction technique, so-called PCA or principal component analysis space, shows different trajectories for different choice types. More recently, very advanced neurotechnology, such as neuropixels, can now record thousands of neurons simultaneously and invasively in animals while they're making decisions. In this paper, when choice information is used, certain subsets of neurons in different brain regions are found to exhibit also winner-take-all behavior, even with such high-density recording. One way to explain such large-scale neuronal recording and high-dimensional neuronal data is through using recurrent neural network models. Here an RNN or recurrent neural network is trained on perceptual decision-making tasks, and a subset of the neural units embedded in the trained model show time integration and winner-take-all behavior that encode decision information. Now I would like to shift gear. A reason for studying perceptual decision-making is that it's not just a simple, straightforward sensory to motor information processing, but this pathway can be subjected to influences and controls internally within the brain. In fact, because of such influences, we can view perceptual decision-making as a window to understanding higher forms of brain function or cognition. Some of our previous computational modeling studies, which use perceptual decision as a foundational platform, include the study of proactive inhibition of an impending choice or action, how previous trials can affect current trials choices, how attention-induced gain modulation of neurons can affect choice behavior, and how chemical neuromodulation and pharmacology can affect decision-making. Shown here. Most recently, perceptual decision-making provides a platform to study metacognition. This is cognition of cognition. One type of metacognition is self-awareness. So over recent years, studies have investigated how confident people or animals are when making their decisions, or whether their decisions are going to be correct or wrong. An often used approach is to ask participants, human participants, to rate their confidence after each decision is made. Studies have also investigated changes of mind due to low decision confidence or reciprocally high decision uncertainty. This can be reported or traced based on motor outputs or, for example, the trajectory of a joystick or computer mouse. They found to be more curved under more uncertain decisions or even change of mind. A recent work led by my previous brilliant PhD student, Nadim Attia, extended my mean field model of decision-making by incorporating a metacognitive component that monitors in real time decision uncertainty. This module in turn can provide feedback control to the decision-making neural populations. Peering inside this decision uncertainty monitoring module are inhibitory and excitatory neurons that respond higher with more decision uncertainty. The model was able to account for well-known observations of behavior in humans and animal studies. And because of its feedback control loop, just like in control engineering, our model can also account for changes of mind. Here, the model neural populations in the model are showing a clear change of the winner from an initial winner to a later loser. And a simple linear transformation to model position shows a change of mind model output as observed in experiments from negative spatial position to positive spatial position. Our model explains change of mind by creating a neutral central attractor during high uncertainty. And with the help of noise, it can allow the initial loser to win. And it turns out that there are more eventual correct than error choices during change of mind. Hence, change of mind is overall a good thing. If you remember the excitatory feedback control in our model, it turns out that it can predict that decision uncertainty continues to be integrated in the absence of sensory evidence. So even when the stimulus is turned off. And a change of mind can occur at much later time. And this prediction is validated by our experimental collaborators and very much unlike other proposed models. Previously, I've mentioned that our proposed model involved inhibitory neuronal population in the uncertainty monitoring module, but it is unclear whether this is actually true or whether there's evidence for it. Also in our model, we propose top-down feedback in the neural circuit to be important, and this needs to be tested by understanding neural information flow. Here, we make use of so-called dynamic causal modeling approach, or DCM approach, which provides a closed loop between forward modeling and then inverse problem back to. And so this is a closed iterative loop of a computational model that tries to fit the neuroimaging data. So, for example, the neuroimaging data of EEG could be of this, oops, so I can't really click here, could be of this form. And this work was done by my postdoctoral researcher, Dr. Abdurreza Satpour. And here, different possible connected network models were fitted to fMRI informed EEG data of the same random dot motion discrimination task. We found that when comparing with high and low confidence rating, the best frontal parietal cortical model has modulation on both the feed forward or bottom up and feedback that is top down connectivity. We have also peered inside the connectivity weights of this model and found feedback connectivity or top down processing to be stronger than feed forward or bottom up processing. This is both for low and high confidence conditions. With DCM, we can peer further inside the fitted model's neural population activities. Here is the typical cortical column model. We found inhibitory neural populations in the right superior frontal gyrus and left precuneus, which is the middle part of the brain, encode high uncertainty, that is low confidence, with possible propagation of uncertainty from the frontal to the precuneus. To a lesser extent, the left superior frontal gyrus and the right middle frontal gyrus of the brain encode high confidence. But this cannot be found in other brain regions. So this suggests an important role for inhibitory neurons in encoding decision uncertainty and propagating the information from high-level frontal brain regions to more sensory-based brain regions, as first shown in our computational model. Our model was able to account for empirical confidence judgments by fitting the model parameters to choice accuracy and response time of additional participants. Using multiple regression, self-reported measures of mental health issues were associated with weaker uncertainty monitoring and feedback connections in the model. Here, the negative, you can see the negative correlation and with statistical significance. However, there is no significant association with the stimulus sensitivity. So this is an internal inside the brain mechanism. Finally, I would wish to shift another gear to quickly mention about the application in machine intelligence and neurotechnology. Here is a collaboration with robotics researchers incorporating a simple noisy accumulator decision model into mobile robots. Okay. Using multiple, and this is, we can use theoretical derivation to derive the average trajectory of the noisy mobile robots using the so-called drift diffusion model of decision making. Another application is the implementation of noisy decision model to integrate multimodal sensory information. Here, the mobile robots and human controllers' heartbeats are being monitored and integrated in real time on a neuromorphic hardware. When the heartbeat is too fast, the decision threshold is reached and the robot stops moving. In a deep learning study, my colleagues investigated two tasks. One is a looming pattern detection, and the other is individual gate identification. Under few short learning conditions, reservoir network model added with my mean field decision model outperform state-of-the-art recurrent network model called long short-term memory or LSTM model. Last but not least, as shown by my research institute's colleague, Dr. Saurabh Bhattacharya, decision confidence or uncertainty can be utilized to enhance neurotechnology. For example, here, processed EEG scalp data of decision confidence can be detected in real time to improve the accuracy and response times in group decision making. To summarize, throughout this talk, I've shown how understanding decision-making can be linked to psychology, neuroscience, mathematics, computing, engineering, physical sciences, and psychiatry. But it can also have impact on many other fields of study, some of which are beyond this talk. In conclusion, we have shown mechanistic computational modeling or simulation and mathematical theory are important in providing deeper understanding of decision-making, which is a key component of cognition, and also even metacognition, which is cognition of a cognition. With advancements in neurotechnology and leading to big, large data sets and complex data sets, data-driven and machine learning approaches can complement such mechanistic modeling. But mechanistic models can still provide valuable insights, as I've shown earlier, even with complex data. If you look inside, you can see mechanistic models still arise. And here, understanding brain mechanisms of decision-making can lead to or inspire novel computational algorithms and intelligent technologies that can surpass state-of-the-art AI technology. To end, I would wish to thank the people who have helped or influenced some of this work and the funding agencies. I also wish to announce that a fully funded PhD studentship is currently available in the area discussed in this talk. So anyone who is interested may contact me for further information. The successful candidate will be based at our research institute, which is recently rated as one of the best research facilities and resources in the whole UK. This is our research center. And finally, I will end with this slide, and I'm happy to take any questions. Thank you.
Thank you, sir, for giving your valuable practical insights. I request the audience to post your questions and queries in the chat box.
Okay, so let's see. Chat box. Should I stop sharing?
Yes, sir.
Okay. Stop sharing. Chat.
I request the audience once again to post your questions and queries in the chat box.
Any questions? No questions.
I guess, yes, so there's no questions.
That's okay. Yeah.
Thank you, sir.
Again, thank you for the.
Thank you for giving your valuable time and sharing your deep knowledge with us. Thank you, sir.
All right, cheers.
Over to you, Ms. Ajita.
Thank you, Ms. Jovita. Next, I call upon the last speaker of the day, Professor Dr. Gustavo Zubaita Kahia. He is the director of IPPA, President of Zubaita University, Vice President of International Society of Chronic Hypoxia. He is a past visiting professor at the University of Copenhagen and currently visiting professor at BLBD University, Lucknow, India, an American university at Barbados. He is a coordinator of research corporation with BLDE University in Vijaypur, India. He is expertised in chronic hypoxia, adaptive response that extend to survival on planet and beyond Earth. His research interest lies in high altitude medicine and physiology adaptation to chronic hypoxia. He has over 126 publications. We welcome you, sir.
Thank you very, very much for such an interesting invitation. I was just very impressed to listen to Professor Kongfat, and I hope he remains and listens to my talk, as it is something completely different, but obviously related, as we all are on planet Earth. I am trying to share my screen. Okay. I believe we are ready. Hello?
Yes, sir, it's visible.
Excellent. Okay, so thank you very much to Dr. Prinong Bakshi for inviting me to participate in such an interesting and important event. I think it is one of the events that is most advanced in the technology and the observation of genetical observation. And my talk relates to life at high altitude, how we live, and how we, in chronic hypoxia, have chronic hypoxia is the name given to a low exposure to a permanent exposure to low oxygen levels that we live at high altitude as high altitude residents, and I will show you here in the city of La Paz. And we have a medal, an award that we give every 2 years that's called Science, Honor, and Truth, because we have seen that around the world there is an alteration of the concepts of science, honor, and truth in many ways. And let me mention that I have some awardees from India in our science awards, and one among them, Professor Tupil Venkatesh, Professor Kushal Das from BLDE University, and some others, but very few have entered such concept in life, because it's very, very important. This is the city of La Paz, with over 2.5 million inhabitants living between 4,100 meters to 3,000 meters. So we live in a chronic hypoxia environment. As you can see, life is completely normal in the activities, in the everyday activities, and many beautiful tourist places to visit, of course, and many sky-high buildings, very high buildings all around, a lot of traffic, of course, and we are located on a mountain. And you see over 2 million people carry out perfectly normal lives all around. There you will see some of the surrounding mountains and a beautiful view of some of the part of the city. It is growing day by day, bigger and bigger. And what happens when a subject goes from sea level with these values of arterial oxygen tension, 98 millimeters of mercury, PaCO2, 40 millimeters of mercury, and an average hematocrit of 42%. When he goes to high altitude, to a city like La Paz, it's different when people, because they just go temporarily and they're outside in the wilderness. But here you come to a city, to a very active, normal city, a metropolis actually. It's actually two cities that are joined, and the highest part of the city is the 4,100 meters, as I said. So what happens when a person flies to La Paz? He's going to this altitude, and as he arrives to La Paz, if he comes to La Paz, his PaO2 will be 60 millimeters of mercury, PaCO2 30 millimeters of mercury. You see, both have lowered because it's related to the pressure of the atmosphere. We have a slightly less atmosphere here because we're at a higher in the mountain. And so if you go up very fast, you can have diseases like acute mountain sickness, which is headache and sometimes vomiting and so on. Or you can have some more serious diseases like HAPE or HACE, high altitude pulmonary edema or high altitude cerebral edema. But if you come up slowly, take it easy, you arrive, you go to a comfortable hotel, you're in a warm environment and so on, and you don't overdo it running around, and you adapt very well. And after some time, if you remain here in La Paz, which I will explain later, your hematocrit will rise as a mechanism to compensate for this low oxygen pressure. And so there is a remarkable adaptation to chronic hypoxia. This is one of our chambers of, it's called a hyperoxic hypoxic adaptation chamber, where we study by modifying the oxygen concentration. We can put a person inside down at sea level, or we can put them on Mount Everest. We have created, based on our experience over many, many years, the high altitude adaptation formula, which we propose is time over altitude change. This has to do that for a fixed altitude, the only significant variable is time, not like in mountain climbing where you are changing altitudes permanently. Here, if you arrive to a fixed altitude like La Paz, then the only variable that will allow for adaptation is the time factor. And how long does it take to adapt to high altitude? And what is adaptation to high altitude? Well, it's having a quality of life, just as like at sea level, with some differences, but nevertheless, even with some advantages. The adaptation time can be calculated by putting the kilometers of altitude, multiplying it by 11.4. In that case, for instance, for La Paz, it is 41 days when your red blood cells would increase to the optimal level for a normal life with a activity just like at sea level, except in extreme exercise. For Everest, it would be around 100 days. That means you would have to, in theory, arrive to Everest and take 100 days to fully adapt to be on the summit of Mount Everest. Now, this is something that has been proposed by my father, whom I will mention later on, but it's true. We observed that people can adapt to extreme hypoxia and survive. For instance, I was a visiting professor at the University of Copenhagen in Denmark, and after the first 3 months I was there, I came back to our lab to work some on high altitude. And as I returned to La Paz, my hematocrit, the day I arrived, we analyzed it, and it was 32%. Normally, my hematocrit in La Paz is 50%. It has been all my life, practically. And so I studied myself what happens, and I did this repeatedly over several years. And what happens is that my hematocrit gradually increased to reach the highest point of my normal hematocrit, where a plateau is reached in 41 days. And so we have an acute adaptation, an intermediate adaptation, and also a full adaptation that is achieved in the later stages. And in the first stages, you can have the first couple of days, you can have the acute mountain sickness diseases, and you can have, if you force yourself physically, you can have a subacute mountain sickness because your organism is not fully adapted, your hearts and your lungs and all your cells are not adapted. So if you force it very well, very much during the first stage, 30 days, you can still have some subacute mountain sickness, which generally is not very severe. And some people that have some lung or heart malfunctions can have an increased level of hematocrit above those living normally at high altitude, which we know as chronic mountain sickness or polyerythrocytemia. In one of our latest publications, we made this graph to show these mechanisms of adaptation, because as you see in the bottom here, the normal blood gases is pH 7.4, PaCO2 40, PaO2 98 millimeters of mercury. And as we see, we have 3 stages here, the first 2 days, then the intermediate stage, and then beyond 40 days. What happens is that as here on the left, as barometric pressure goes down, you can see that the inspired oxygen tension, just a minute. Yes. The inspired oxygen tension goes down, the arterial oxygen tension and saturation in your blood goes down, and ventilation increases and cardiac function increase. Those are the 2 pumps that I will talk about in a few minutes. And your carbon dioxide goes down and the pH goes up. And this is described in medicine as acute respiratory alkalosis. And so what happens is that you cannot live in permanent alkalosis because cells need to function in an optimal pH. pH is a crucial, it's essential. And so the kidneys start functioning to excrete some bicarbonate, so you have a balance in the Henderson-Hasselbalch equation. But some people have, some C-level scientists have talked about a chronic respiratory alkalosis. We are in complete disagreement with this. We don't live in chronic respiratory alkalosis. We live in a perfect, normal pH, perfect acid-base balance at high altitude. But our values are different, yet our pH is normal. pH is 7.4, PaCO2 30, PaO2 60 millimeters of mercury. And so as we go into the intermediate here section in the middle, you will see that the vascular endothelial growth factor is stimulated. There's an increase of nitrous oxide. The erythropoietin in that red line increases because we have to have an increase as shown in that graph curved line of hemoglobin going up to the normal levels. But we also have an increased pulmonary artery pressure and a right ventricle dilatation. This evolves to be a right ventricle hypertrophy with time. In the right third of the image here, you will see pulmonary hypertension, which is perfectly normal. We have a normal at sea level, pulmonary artery pressure is around 12 millimeters of mercury. Here in La Paz, almost everybody has 25 millimeters of mercury, and it's perfectly normal. We have an increased hemoglobin, as I said, a hematocrit of 50% or a hemoglobin of 16 grams per cent is perfectly normal for me. But there's also a increased mitochondrial efficiency. This is very interesting. When I wrote this paper, this produced a rejection from some sea-level experts saying that, oh no, there's no mitochondrial efficiency at altitude, but there is. There are several articles that show this. And there are multiple molecular changes, some of which probably are being mentioned in your symposium. And so the oxygen transport mechanisms is what we call the pneumodynamic pump, which are the lungs, which are a vacuum pump. The diaphragms go down when you breathe, and that makes a vacuum in your lungs, and air goes into your alveoli. The hemodynamic pump, in the contrary, works not with gas, but with a liquid, which is blood, and that's a compression pump, and that's the heart and all the vessels. And respiration and circulation have common traits because they are both pumps, and they both work in conjunction. As you can see, there's a respiratory rate, which corresponds to a heart rate, and a tidal volume, the amount of air moved per breathing, that corresponds to a systolic volume, stroke volume at sea level, or at high altitude. And both of these depend on one crucial factor of transportation of oxygen, which is hemoglobin. For us, hemoglobin is the magnificent cell that allows our survival at high altitude in chronic hypoxia. This is from another publication that we recently made with my daughter, Natalia Subieta de Oroste. She is also an MD, has studied with us here in Bolivia, and now she's doing a neuroscience PhD at the University of Calgary. So the oxygen transport triad is what we call it: the pneumodynamic pump, the hemodynamic pump, and hemoglobin. Those three factors are crucial for survival at high altitude. They are the macro physiology essential factors. And this oxygen transport triad is related to the laws of physics, of course. And that's why we made this graph, this interesting graph, where you can see that first the Gay-Lussac law, the relation of pressure and temperature, play a role when gases enter to the lungs by the vacuum system. And once there, the Boyle's law plays a role, which is related to the pressure and volume. And so the air is spread through the alveoli and then reaches the alveolo-capillary membrane and has to be transferred. The oxygen has to be transferred to the red blood cells, and that's the diffusion of Fick's law. And Henry's law plays a role throughout all the mechanism. And later in the capillaries, they, like a tree, like an inverted tree, like we see here, a slanted tree, it gathers all the vessels to form, to go to the heart again, and that the hydraulic laws play a role with the Fick's law and Pascal's law. And so finally, diffusion is the mechanism by which oxygen reaches the mitochondria. What we have observed over many years, this is a resume. I'm not showing a full extensive data, but this is what a graph we developed from permanent living high altitude populations at different altitudes and what their hemoglobin was. As you can see, it begins to rise at about 2,000 meters. And here in La Paz, where we are at 3,000, it's around 16, the average, of course. And it continues to rise up in populations living at 5,300 meters, around 20 millimeters. The population there has 20 grams per cent of hemoglobin in their blood, and they are perfectly adapted. The estimated Mount Everest would be 26, and this, whenever I mention that, most sea level physiologists are shocked, but it's true. And this has been proved before by Professor František Kolar from the Czech Republic. He has put rats living at Mount Everest, and in fact, they did increase to those levels the hemoglobin. The other interesting factor that happens when going up to high altitude, this is from the paper, is that carbon dioxide, the pressure of carbon dioxide decreases gradually. And as you can see in the city of La Paz, it's 30, but on the summit of Mount Everest, it's around 8 millimeters of mercury. So these two factors, the hemoglobin and carbon dioxide, are factors that are part of life at high altitude. And that's why we developed the formula called the tolerance to hypoxia formula. The tolerance to hypoxia formula has as a numerator the hemoglobin, and as a denominator the arterial oxygen tension of carbon dioxide. And there's a factor. This is very interesting because, as you can see, to tolerate hypoxia, you need to have a low PaCO2 in the denominator. The lower it is, the better you tolerate hypoxia. And that changes the acid-base status. We have very interesting papers where we have calculated what are the correction factors for the interpretation of acid-base at high altitude. And as time goes by, when you stay longer, as I have shown you, your hemoglobin gradually has to grow up. And this is because there is a limiting capacity of production of millions and millions of red blood cells, extra cells that are needed to transport oxygen. So as the higher the hemoglobin, the higher the tolerance to hypoxia. But there's a limit according to this level of altitude, of course. Now, let me show you that what deficiencies are tolerable and intolerable. If a living organism such as that of a human receives less water, if you don't drink water, you will die from dehydration. If you don't have enough potassium, you can have a cardiac arrest. Less sodium, you'll have neural transmission alteration, lower blood pressure, less chloride infections, less food, you will starve and you will die. If you have less vitamin, if you don't receive vitamin C, for instance, you will have scurvy like they had in the trips in the ships, in the long conquering ships of America, for instance. They suffered scurvy, which is a lethal disease. Less vitamin D is neuritis, less vitamin D, osteoporosis, less immunity, less calcium, osteoporosis, less zinc, less immunity, less magnesium, decreased bone density, altered nerve function, less selenium, altered mood and anxiety, less glucose, hypoglycemia, metabolic dysfunction, less fat, less cortisol, less proteins, less immunity, less essential amino acids, and malnutrition. Extreme exposure to cold, you can have frostbite, lose your fingers or your toes or your nose, or death. So many of these deficiencies are not tolerable and often deadly. What's interesting is that chronic hypoxia, which is less oxygen, can be survived. It allows for survival. The less chronic, the chronic exposure to hypoxia people can survive with lower oxygen. This is what's amazing. This molecule, that's the essential molecule with which you cannot survive, but you cannot stop breathing for 3 or 4 minutes. You die if you don't have oxygen. It's one of the fastest way that person can die, aside from a heart attack, of course. But this is interesting, that if you are exposed to a lower pressure of oxygen and hence lower oxygen levels in your organism, you can survive very well. And what I'll show you is that we even survive better than at sea level. So the types of adaptation is the way we talk about it: genetic adaptation and physiological adaptation. We don't use the word acclimatization for high altitude aspects because it's not a climate change, it's a hypoxic change, which is a completely different thing. So we call it physiological adaptation. And of course, physiological adaptation gives rise to genetic adaptation with time. Darwin's evolution of millions of years gives rise But the physiological adaptation, and it's interesting that we are programmed genetically in our DNAs to survive oxygen levels, even on top of mountains like what you see behind, which is Illimani at 6,400 meters above sea level, right behind and right next to the city of La Paz, that you can see behind me. And but there's a lifetime survival, like what I'm explaining to you, which is can last days or months, and it has to do with epigenetic change, hypoxia-inducible factors, vascular endothelial factors, nitrous oxide, and so on, as I have explained. So all these changes are allowing us to survive. So in this city like La Paz and El Alto, we are defeating hypoxia. You know, hypoxia is not a threat for us. We live in this low oxygen pressure that at sea level would be critical. If someone at sea level presents an arterial oxygen tension of 60 millimeters of mercury, this is absolutely critical. They can go into intensive care. And this has been shown in COVID, and I'll talk a little bit about that later. But for us, this oxygen pressure is normoxia. And sea level for us is hyperoxia. And my father even said that man can adapt to any altitude on planet Earth, even the summit of Mount Everest. He passed away several years ago. The organic systems of human beings and all other species tend towards adaptation to any environmental change within an optimal period of time and never tend towards regression. In other words, there cannot be loss of adaptation or de-adaptation, because that leads inevitably to death. What we did is we studied how long do people in the city of La Paz, in El Alto, the highlands, live here in Bolivia in relation to the lowlands in Bolivia, because as you know, we are located in the center of South America, and we have Amazon, we have tropical lands, and we have the second largest city, and maybe now the first largest city is in the lowlands, Santa Cruz, at 400 meters of altitude. So we studied how long the residents live, and we showed that there is extended longevity at high altitude. What we saw here, for instance, is here dividing by the population, of course, in order to uniform observations, going from the lowest city in Bolivia at 221 meters in the Amazon, going up to Santa Cruz, 416, Tarija, Cochabamba, Oruro, La Paz, where we are located at 3,800 meters, and Potosí at 4,090 meters, which is the highest city in Bolivia, with where the silver was mined during the space, during the history, during the colony, where the Spanish took a lot of silver to Spain and Europe and made Europe wealthy, actually, thanks to that. And so you can see here, there's a tendency that people will live over 100 years old, much more in the highlands than in the lowlands, with the exception of Chuquisaca, which is located at 2,700 meters, where there were more people with older than 100 years of age. And so where is COVID less deadly, at sea level or in the highlands? You know, many scientists around the world believed, oh my God, now COVID comes, the high altitude people will die tremendously. Well, with our experience, I was totally surprised by that affirmation. So I was invited to give a talk, an television interview at the Universidad Mayor de San Andrés, where I did study my medical school, on January 31st, 2020. And there I exposed the first hypothesis that COVID-19 would be less severe at high altitude. And this is the interview. It's all available on the web. And several of our publications followed. One of them was this one: does the pathogenesis of SARS-CoV virus decrease at high altitude? With a lot of Bolivian colleagues, except one that is from Australia. But what we proposed there, that the lower amount of ACE2 receptors in the lungs would probably, that high altitude mice have shown as a mechanism of adaptation, as that is the entering point for viruses, like COVID, then they would have lower risk. But we also talked about higher ultraviolet radiation at high altitude, which would have a protective factor. And several of our papers followed: decreased incidence, virus transmission capacity, and severity of COVID at altitude. And all of them showed this type of pattern, that as you go up in altitude, there were less number of cases of COVID. This confirmed our original postulate, that our hypothesis that we would have lower COVID incidence. And for instance, this is a letter to the editor, because someone affirmed that at high altitude they found that there were more deaths, which is truly not true. As you can see here, in Bolivia, on top, the case fatality rate on the further right is 8.6% in the lowlands, but 3.2 in the highlands of Bolivia above 2,500 meters. The same happened in Peru, 5.9% case fatality rate in the lowlands, below 1,500 meters, yet at high altitude was 3.1. And this is the way it evolved in Bolivia, how the cases increased. And if you can see, the green line is the number of cases in Santa Cruz, located at 400 meters of altitude, and whereas the dotted blue line is La Paz, where it was much less, although we had very similar populations. And so we did a COVID incidence and case fatality rate. I was the director along with Natalia, my daughter Natalia, and we reviewed the populations of Bolivia, Colombia, Ecuador, Peru, Mexico, and we had a total of 161 million people that had been exposed to COVID. And in all of them, we found a difference that you see here in each one of the graphs of each country, where the incidence, the case fatality rate, was much lower at high altitude than at sea level. We also observed in another one of our papers that those with a higher erythropoietin at high altitude survive much better the COVID aggression, even in intensive care units. You see here, higher EPO were survivors, much more survivors, those that higher EPO, than those that had a lower EPO levels, or higher hemoglobin and lower hemoglobin. You see, those with lower hemoglobins died more. And in conclusion for that study, exposure to chronic hypobaric hypoxia is mostly beneficial even during the COVID pandemic. Now, many people truly don't understand our pathologies very well. Many scientists from sea level that study high altitude but live at sea level, they have a focus about what high altitude medicine is and how they interpreted it, particularly during mountain climbing. And so the diseases, acute mountain sickness, high altitude pulmonary edema, and high altitude cerebral, a view from the high Andes, is a paper that we published with Natalia, and this was in May 2021. And this paper was 18 sea-level high-altitude experts wrote to the chief editor asking that the article be withdrawn. They made around 24 observations to the article, and they were really trivial. And we asked some people what they thought, and we had a letter of support from great scientists like Professor Tupil Venkatesh, Professor Kushal Das, and many from India, and many great researchers also from Latin America, from the high altitude medical centers here that were treating patients all the time. All these people wrote and gave support to this article that we have written. Nevertheless, it was retracted from the journal. They removed it. And this happens because they don't understand the pathology the way we understand it. That's why we wrote 2 more articles which have to do with the responses to their observations. And this is the oxygen transport triad in high altitude pulmonary, a perspective in the high Andes, and also this other published online by De Gruyter in the reviews on environmental health. They are both high-ranking articles where we rewrote the article, high altitude pulmonary edema, high altitude cerebral edema, and acute mountain sickness, and enhanced of vision from the high Andes, La Paz, Bolivia, 3,500 meters. Unfortunately, the original article that we wrote was above their head. They did not understand it because they don't live here as we do. We treated patients during 52 years, and no one has ever died at our institution from those diseases because we know how to handle it very well. And one of the observations was, is mitochondrial efficiency increased at high altitude? They criticized this strongly, that we had written that in that original article. But here are many articles that are written now that, well, they're very recent, that appear in different areas, showing that mitochondrial efficiency is indeed increased during adaptation to high altitude. Our mitochondria are stronger. They are more effective. They are able to produce ATP, generate energy, even with low oxygen levels. If life is so successful at high altitudes in the city of La Paz and Cerro de Pasco in Peru, at those altitudes, and the human being in his quest for instinctive survival will leave planet Earth and travel through space, is it important that all humans adapt to chronic hypoxia? Is the question we are postulating since many years back. I wrote, when I was at the University of Copenhagen, a small book called Human Adaptation and to High Altitude and to Sea Level. It was published by a German press, written in 2007. And there I talked about this new concept that I am now explaining to you. You see, the extravehicular unit is the smallest space, it's the smallest space vehicle, because in the organism of the human has to survive in this small environment, and to stay alive in space, the pressure vessel around the astronaut has to be adequate. There has to be protection from extreme environment, radiation, micrometeor, etc. Human performance gloves have to be used, but there's also thermal control to maintain the body temperature, and the backpack, the portable life support system, has to supply the oxygen, the pressure control, carbon dioxide and trace has to be removed, the humidity control, thermal control, power communications, and data displays. But you see, the original space flights were in a pure oxygen environment, 100% oxygen, and 1/3 the sea level pressure, because they were afraid of the pressure changes. But this was a big mistake, as the pressure was so low in the capsule, and one of the first flights to go to the moon was Apollo the first. With these conditions, they unfortunately suffered a terrible accident with the death of 3 great astronauts, Grissom, White, and Chaffee, who unfortunately burned in the cabin because the oxygen was 100%, with the idea to maintain sea level pressures of oxygen for the organism. It was then decided that they would maintain the sea level pressure and oxygen and the sea level pressures with a barometric pressure of 760 millimeters of mercury and a PiO2 of 150 millimeters. So then the actual, every spaceship right now traveling in space uses the inspired oxygen tension of 150 millimeters and the barometric pressure of 760 millimeters of mercury. Now, we think this is a mistake. We think that IPA, our institution, has proposed that the pressure should be the same as in the city of La Paz. You see, going from 150 to 94 millimeters, which is our environmental oxygen pressure in this city behind me, with a barometric pressure of 495. The reasoning behind this is that there's a faster preparation for extravehicular activity. You see, normally it takes several hours during the preparation to put on the spacesuits in space, because they had to reduce the pressure from sea level to around the pressure of Mount Everest in order to enter the suits, to survive in the suits. And so if we use our concept of having astronauts adapt to high altitude, like city of La Paz, we would have an oxygen economy, and we would have greater possibility of survival, not only during space flights, but also even in Mars, where humans plan to go next with Elon Musk. You see, these are some of the suits that have been used by NASA. Look how big and strong they have to be in order to sustain the pressure of Mount Everest, because outside in space you have no pressure. So inside they're like in Mount Everest, but they increase the oxygen pressure when they have to go out to try to maintain sea level oxygen pressures. And you see how complex the articulations are, and it's very, very, very uncomfortable for the astronauts and very limiting. These are a little more modern models, and they have tried different models to produce this. This is one of the final models, but I understand Elon Musk is also preparing different, more advanced models of these extravehicular mobility units, which are extraordinary indeed. But still, look how thick the clothes are in order to maintain the pressure. And so the proposal is to use, as I said, in all these vehicles in space, inspired oxygen pressure like La Paz, with a barometric pressure of 495, which is our normal right now. Where I am right now, I am with that pressure. And why would we do that? Because, for instance, if you can see here, the extra mobility unit in space has to have this minimum pressure, 160 millimeters of mercury, plus CO2 40, plus water vapor. It has to have a pressure of 240 millimeters of mercury, which is Mount Everest pressure. Whereas in our proposal, if the astronauts were adapted to our life at high altitude, they could use these lower levels, not the sea level of the inspired oxygen tension, but these lower levels and lower carbon dioxide and the same water vapor pressure, which is the same. But you see how this reduces from 240 millimeters of mercury to 171 millimeters of mercury. This corresponds to an economy of 70 millimeters of mercury, which would make suits much lighter, much more feasible to use in space and also to survive. Why do we propose this? Because the astronauts suffer anemia during spaceflight, but we don't suffer anemia. We increase our red blood cells because we have lower oxygen levels. That way, astronauts, when they return from space flights, if they would be living in a chronic hypoxia environment, they would not suffer anemia when they return to sea level. There will be an energy efficiency and economy are improved in space. In order to prepare for EVA, the preparation time is reduced and facilitated. They would be able to get out of the spaceship much faster because they don't have to go from sea level to the pressure of Mount Everest in the suits. A much more flexible suit, allowing for more comfort and precision. So here in the future, man will face lack of oxygen in space. Physics can be aided by biology. Physics would be the change in pressures in space, but biology is the adaptation of the organism to the oxygen pressure changes. So these laws of physics can be wisely surpassed by biology, thanks to the extraordinary adaptation of living beings to their environment. Adaptation can be taken to the limits with adequate time, as expressed in our formula of adaptation. Here is a graph to show you. This is sea level, the red line at the bottom, and the top of Mount Everest there at the right. So at sea level, you have a barometric pressure of 760 millimeters of mercury, whereas at Mount Everest at 8,842 millimeters, you have 250 millimeters of mercury of pressure. And that's the pressure that's used in space at 330 kilometers of mercury, where there is 0 millimeters of mercury in space. There is no pressure. There's an emptiness sort of in space. So inside this suit, the pressure that can be used, that is used currently, is 250 millimeters of mercury. But why is that? Because you have to reduce from sea level, this right arrow, to 250 millimeters of mercury is is a difference of 510 millimeters that have to be reduced. And this is done in a special lock in space where they put the astronauts inside this lock and they start reducing the pressure gradually over hours. They have to breathe a lot of oxygen to try to not to suffer decompression sickness. And so it's a complex process. And it's a big change. 510 millimeters of mercury is a big pressure change. Whereas if they would use the altitude of La Paz barometric pressure of 495 millimeters shown here, the change to put on the spacesuit would be only 245 millimeters of mercury. So that would be reducing the pressure change only to 1/2 what it currently is. So chronic hypoxia has to go into space because that way biology can help reduce the starting pressure of change. And that way, we believe, is the way to go in the future. High altitude residents live because of their circumstances closer to space, 1/3 less barometric pressure. We have adapted to this lower pressure, and we are perfectly adapted with 1/3 less oxygen pressure than at sea level. Oxygen in space is scarce, we know that. Although oxygen is abundant in the universe, but to have an atmosphere is quite unique for planet Earth that has 21% oxygen. Why not accept the fact that humans living at high altitude are more tolerant to hypoxia? Whereas sea level residents have one crucial disability, poor tolerance to hypoxia. If there's anything women will have to face in space, it is hypoxia. And this can be the greatest threat to survival. I created a word called biospace forming. What is this? Bio means life, space is the universe, and forming is adaptation. So it is the adaptation of all living beings on Earth to space. But this not only applies to humans, it's animals, plants, and all microbiota and everything, all bacteria and everything have to adapt to space. So I created the term biospace forming. This is very important because they're talking about terraforming, changing whole Mars to become like Earth over many, many years. But nobody has talked about the biological adaptation to space, and that's why I created this term. And incidentally, it's published in the BLDE journal in India for the first time. This is my father, the mountain guru. He was named the mountain guru at the University of St. John's in Bangalore in 2009. And this is my daughter, Natalia, and the 3 generations that have been dedicated to working at high altitude and observing these things over 52 years in our institution, as we said. But obviously, my father lived here in Bolivia during most of his 90 years of age. He passed away at 90 years of age, having lived in high altitude all his life. And so this is finally the formula of adaptation, time over altitude. This is the mountain of Sajama, the highest mountain in Bolivia, located at the peak is at 6,542 meters. We then carried out a soccer game at that altitude at 6,542 meters, directed by my father. We were his support team. And they played a game of, these are reporters, and they played a game at that altitude, having gone up during the same day, all the way up from the base of Mount Sajama at 4,300 meters, all the way to 6,542 meters. They set up the goalposts and put on their suits and played 20 minutes per side, tied 2-2, and this proved that the remarkable adaptation of human beings to play even at such high altitude. And as you can see, they're running quite actively and playing. Nobody died, of course. They told us when we were going to carry out this feat that that would be very dangerous, somebody could die, but they played 20 minutes per side, and the balls were painted red because it was played in snow. And then one occasion, one of the balls went down after the goal, went down the mountain, and one of the went all the way down. And one of the reporters said that it had gone down to sea level, but not really to sea level, only to the base of the mountain, of course. That was an extraordinary feat that has not been able to be repeated again. It took a lot of logistics and very complicated times. And anyway, we have already celebrated, I believe, 15 years from this event. And it is good to show you the extraordinary adaptation of human beings to 6,542 meters to explain that. Now, one final comment. You see, Earth became habitable, and it has evolved over time needed for the development of our intelligence, of us humans, 4.4 billion years. And that is incredible time it took for us to be sitting here to communicate from this side of the world in Bolivia, where it's almost 9 a.m., to your side of the world, where it's, I suppose, around 6-something p.m. And it took this long for intelligence to arrive so this could be carried out. And the Professor David Kipping has postulated that probably we are alone in the universe. They're talking about exoplanets, finding life in other planets that may have a similar environment, because there's billions of planets and galaxies. But you know, I always ask myself, if we are able to hear the Big Bang, we still have hear the noise of the Big Bang, apparently, to what has been said, but we don't hear one message from life outside. We don't have messages. Some people affirm they do, but truly scientific, proven radio waves or some kind of mechanism, we have not received. And so I believe the Big Bang is an interesting theory, but it's also like what has happened throughout history all the time. When people could only see the sun, they thought that we were the center of the universe. The Earth was the center of the universe. Then they found out that there was the Earth that was going around the sun, and there were 9 planets around. And so suddenly it started getting a little more clear. But then they found out that we were in a galaxy. And then they found out that there are many galaxies. And now they say that the Big Bang created from nothing, which is extremely strange, the whole universe. And I believe they are, again, doing the same mistake. In beyond, probably the Big Bang has occurred, but it's only a small part of the true universe, which is immense, which is infinite, which is not only what we are seeing with the great James Webb telescope, the Hubble. It's way beyond. So we humans tend to believe that we are extremely intelligent, which we are without doubt, I believe, but we think we can be above nature. And I think this is a huge mistake. I think the DNA has evolved by trial and error over these 4.4 billion years. Trial and error, many, many generations. And the most efficient ways it has worked with survival of the fittest, of course, which is reasonable. So that DNA that has evolved, we have to keep it preciously and not change it too much with our intelligence, because I am certain that we believe we are extremely intelligent, but the changes we make can be extremely deleterious for life, can be dangerous, like a time bomb, like a nuclear explosion at the genetic level. So I'm always urging scientists to be careful with genetic manipulations, to play godlike in science can be extremely dangerous and can have terrible side effects. And we cannot risk that life because we probably are alone in the universe. Thank you very much. Thank you.
Thank you, sir. Thank you for sharing your deep knowledge with us. We request the audience to post your questions and queries in the chat box. Sir, there are some questions in the chat box. Can I take them one by one?
Please.
Okay. So Dr. Sumant have asked, is there any effect of infection like COVID-19 and adaptations?
Interesting question, doctor. Very interesting. You know, one of the things I postulated is that our immune systems are different at high altitude. And that I postulated because llamas have nanobodies like antibodies, but they are smaller antibodies. They're called nanobodies. And there are several studies showing that these nanobodies have great capacities for immunity. And I had postulated originally that we probably have a higher immunity that has allowed us to survive the COVID. And a professor, some work from Argentina showed that when they made an ELISA test to test immunity in patients during the COVID times, they showed that at high altitude cities in Argentina, there were higher levels of immunity to COVID as compared to sea level. They were surprised, and it's published now. So yes, I agree with you.
Thank you, sir. We have another question from Dr. Nandini Rao. Is the incidence of macular degeneration high at high altitudes due to increased VEGF?
Challenging question. Very nice, doctor. We carried out our 8th chronic symposium of chronic hypoxia symposium just recently in October. One of the doctors I invited was one of the ophthalmologists that works here in La Paz, Dr. Vania Garcia. She studied the retina of high altitude residents as part of her normal studies in a multinational study throughout South America to see the differences of the vascularity of the retina at high altitude, I mean, in all their patients, in all cities. And to their surprise, they found that the people living in La Paz, Bolivia, had much higher vascularity. That means our retinas have more vessels as a form of adaptation to transport oxygen. We can see very well, of course, very clearly. And I believe we don't have true statistics, but I strongly believe that we probably have less macular degeneration than those living at sea level. I strongly believe that, but because of this more vascularity. So, but that needs to be tested. That's why your question is very appropriate for the moment. Thank you.
Thank you, sir. We have another question by Dr. Nandini Rao. What is the molecular mechanism behind the increased efficiency of mitochondria at high altitudes?
Ooh, you got me on that one, Dr. Landevin. I am not an expert in that, but I would be happy to give you the citations of those articles of those studies. Some of those studies are being carried out now in La Paz, in some of the laboratories here with some of our friends from Laval University, Jorge Solís, Christian Arias Reyes, and several others are working, and they have shown, and Dr. Edith from the University of Zurich also, they are showing that the mitochondrial efficiency is a fact. But obviously this will carry on with much more research, and I cannot tell you the exact response, what of the molecular level precisely it is.
Thank you, sir. Is the work qualitative or quantitative?
I'm sorry?
Is the work, the same question, sir, regarding mitochondria, is it qualitative or quantitative?
Yes. Originally, some publications talked about quantitative increase of mitochondria, but the work currently now is showing that there is qualitative. This is a big field. That was originally proposed as actually like a theory that since all cells have to adapt, all our organisms adapt. Why not also the mitochondria? And so it is also a qualitative improvement, sir.
Thank you, sir. Thank you so much. That's all for the questions, sir.
Excellent.
Thank you, sir.
Thank you very much.
Thank you, sir. Thank you one and all for your valuable participation. We have almost come to an end of this program. Thank you for attending this 3rd International Conference on Bioinformatics and Data Science and the 9th International Conference on Public Mental Health and Neuroscience. There are more interesting and brainstorming sessions tomorrow. See you all tomorrow. Thank you.
Now 9 o'clock. Thank you. Good night.